Good morning and welcome to this very interesting session on protective factors for dementia and other neurodegenerative disorders. I think this is an exciting area because evidence is growing of effectiveness of this approach, non-pharmacological approach, to protect the brain and also to slow down disease progression. So we are very happy to have experts, worldwide experts on this field. And the first one is Diego Centonce, who is professor of neurology at Vergata University. He's an expert of neuroinflammatory disorders and brain plasticity. Please, Diego.
Thank you, Vincenzo. Thank you, Professor Comi, for inviting me to present data trying to summarize some neurobiological underpinnings at the basis of the effects of environmental manipulations like physical exercise and diet in the brain. I will essentially present data in a model of neuroinflammatory disorders such as experimental autoimmune encephalomyelitis, which is a well-recognized model of multiple sclerosis. I will start presenting some data in where we wanted to identify the mechanism by which voluntary exercise could affect not only motor function, but also cognitive abilities in EAE mice. You see here in this model, we immunized the animals with the MOG to induce the disease. A group of these mice had the chance to go on a running wheel, and this was associated to improvement in the motor score of the animal. But even before the appearance of the motor deficits, animals showed some improvement in their cognitive abilities. You see, at day post-immunization 8, we testing the animals which still had not motor disabilities in a test, in a cognitive test, in a novel object recognition test, where the animals were had to identify the new environment, and you see that EAE was associated with a loss of keeping track of previous exposure to objects, while physical exercise was able to improve the ability of animals to keep memory of the previous exposure. According to an effect in the hippocampus, we also found that in another test, essentially dedicated to explore hippocampal function, which is the nest building test, where the animals is an ecological test where the animals generally build their own nest, and this requires, of course, some good attitude. Is also a model of depression, and also some cognitive abilities. You see that EAE is associated with a poor score, which is recovered after exercise. So again, exercise is able to prevent some damages in this hippocampal test. According to the idea that the hippocampus was a critical area where exercise could cause some beneficial effects, we also found that synaptic plasticity, LTP-like plasticity, was abnormal in EAE mice because it was, you see, exacerbated potentiation. We could not depotentiate. It's a form of abnormal potentiation, which was again restored by exercise. Exercise was therefore able to prevent some synaptic deficits in the hippocampus. And according to the idea that synaptic transmission and plasticity is a main site of action of exercise, synapsin-1, a marker of synaptic transmission, was abnormal in EAE, was recovered following exercise. And we found by counting the number of specific interneuron in the hippocampus, which is the GABAergic parvalbumin positive interneuron, which you know is essential for cognitive abilities, giving rise to the gamma rhythm, which is a rhythm involved in cognitive abilities. EAE, inflammatory neuroinflammation in the brain, was associated with a significant reduction of these parvalbumin interneurons, which are again positively responding to exercise. So this also provides us a possible explanation for increased LTP found in EAE, because of course the reduction, a possible reduction of GABAergic transmission could explain the potentiation of LTP. And in fact, by incubating with pyrcotoxin, which is a blocker of GABA receptor, we could mimic in exercise mice the effects of EAE alone. So again, confirmed the involvement of GABAergic interneurons in the abnormal synaptic plasticity and motor deficits occurring in EAE mice. And a number of markers of inflammation was altered in the hippocampus. This was true for IBA1, indicating, of course, microgliosis, TNF-alpha production, interleukin-1 beta, interleukin-6. All these inflammatory markers were recovered following physical exercise. There was also a tight association between parvalbumin depletion and microgliosis. Areas enriched, particularly enriched with microglia, were those where the parvalbumin interneurons were selectively degenerating. And yes, analyzing in more detail the macroglia in EAE mice and the effects of exercise in these mice, we found that the density of IBA1 microglial neurons were increased, following EAE, indicating the activated microgliosis. And again, exercise was able to reverse this inflammatory reaction of the brain to neuroinflammation. And not only the density, meaning the number of microglial cells, but also their morphology, that microglial cells react to inflammation by showing some morphological phenotypes involved in the production in inflammatory activities such as synaptic pruning and so on, and also the morphology, which were measured in terms of microglia total area and the cell area, you see is altered following EAE and reversed by physical exercise. So inflammation, neuroinflammation causes some synaptic inflammatory activity, abnormalities, and microglial cells seem to be one important element in this reaction. And according to this, with some other experiments, we found that microglial cells were increased in number, had altered morphology, and also produced higher levels of pro-inflammatory cytokines, such as TNF-alpha, the co-localization of the marker of IBA1 and TNF-alpha indicates the ability of microglial cells in EAE mice to produce a TNF-alpha, and the same was also true for interleukin beta. But exercise is not improving EAE curves only by affecting the inflammatory milieu, synaptic plasticity and transmission of the hippocampus. In other experiments in this striatum, we found that synaptic transmission and morphology and the functioning of synapses were recovered following physical exercise. These are Golgi immunostaining showing dendritic branch with a number of dendritic spines which are dramatically reduced following EAE. EAE, even before the appearance of motor deficits and inflammatory reaction in the striatum, causes a dramatic loss of dendrites and a dramatic loss of synapses. Exercise was again able to recover this abnormality, this morphological abnormality. And in another model of non-inflammatory demyelination, which is essentially caused by cuprizone feeding on animals, cuprizone causes a demyelination which is not associated to inflammation. We wanted to test whether inflammation, the reduction in inflammation, was the primary event to explain the effect of exercise, and so we also studied this model. What we found is that exercise, even in the absence of inflammation, was able to produce some improvement in the motor defects associated to Cuprizone, to demyelination, and the effect of exercise was also seen at the level of the density of myelin in the corpus callosum, in the striatum, in other brain areas. And exercise again was able to recover these abnormalities, as also shown here in terms of increased MBP, Western blot. And what happened in humans? In humans, essentially, again, physical exercise was found to exert a number of beneficial effects. We wanted to focus our attention on the effects of exercise, voluntary exercise, on the inflammatory reaction in the brain. And so we simply patients which were hospitalized for the diagnosis of multiple sclerosis were asked to fill a form where they, simply gave us information on their lifestyle. And so by focusing on their spontaneous activity in the 6 months before the hospitalization, we divided patients in terms of exercise, of lifestyle, physical activity. What we found is that in comparison to sedentary MS patients, the levels of some pro-inflammatory cytokines, such as interleukin-2 and interleukin-6, were reduced in individuals normally doing some physical activity, even as a lifestyle, and not exercising regularly in a gym, for example. And in line with the idea that physical exercise reduces inflammation and produces some beneficial effects, we observed in these individuals which had not motor deficits. They were at the diagnosis of MS, and so, but we found some effects of physical exercise on mood. You see that not only the prevalence, but also the severity of trait anxiety, state anxiety, and depression were significantly reduced in individuals that had the physical activity and exercise as a lifestyle. And same experiments, same studies were also run in another study where we measured synaptic plasticity assessed by TMS to measure the amplitude of LTP-like plasticity, which is associated to some brain resilience. And we confirmed that in comparison to healthy individuals, newly diagnosed patients with MS had a reduced LTP in the brain, which was somehow recovered, or which was higher in individuals who spontaneously had, as a lifestyle activity, had the attitude of doing some physical activity. So exercise was able to recover LTP defects of patients. Also in progressive MS patients, the effects of LTP is even more pronounced, and hospitalization and the rehabilitation program was able to recover LTP amplitude in the cortex, and the amplitude of LTP nicely correlated also with improvement of some motor tests, such as the 9-hole pegboard test, which is a measure of hand ability. And not only physiologically, but also morphologically, we found the effect of physical exercise on hippocampal volume and cortical thickness in these patients. Now we want to, go further in this line of research. We identified recently that T lymphocytes extracted, taken from the blood of progressive MS patients, but not from healthy individuals, have the ability, when placed onto brain slices during neurophysiological recordings of synaptic activity. So these lymphocytes alter synaptic transmission. You see, this is just a neurophysiological experiment where these downward deflections reflect a spontaneous glutamate release. You see that this is recording was run in the presence of T lymphocytes from healthy individuals, in the presence of T lymphocytes from progressive MS patients. Even in the absence of any sign of inflammation in the brain, you see there is this dramatic increase in the frequency and also in the kinetic properties of synaptic transmission. This effect, the synaptic effect of T lymphocytes of progressive MS patients was blocked by physical exercise, by rehabilitation programs in patients. So rehabilitation and physical exercise interacts also with the ability of T lymphocytes to cause some synaptopathy in the brain of MS patients. We are now trying to see whether the effects of physical exercise on the ability of T lymphocytes to produce brain damage could be mediated by communication between the brain and the lymphocytes through the vagus nerve. And so here we wanted to see whether vagotomy could interfere with the ability of physical exercise to cause synaptic protection in the brain of EI mice. And what we found that cervical vagotomy was indeed able to fully block the protective effects of physical exercise on synaptopathy associated to inflammation in model of multiple sclerosis. Interestingly, we found that selective splenic vagotomy, the idea is that the vagus nerve is innervating the spleen, and the spleen, the lymphocytes released from the spleen enter the brain with less ability to produce damage in response to exercise. But vagotomy was not able to replicate, to mimic the effects of cervical vagotomy, implying probably that the effect of vagotomy is not mediated through the effect on the spleen, but on the intestine. And so we're now addressing the hypothesis that the vagus nerve causes some changes in the microbiota in the intestine, which in turn produces some inflammatory reaction which could affect the brain. Just moving to the other topic, which is diet. Yesterday we listened to wonderful presentations on the effects of diet in brain health in several diseases, including multiple sclerosis. This is just to recap some of this information. And so we, again, wanted to see whether a fat diet, which is, of course, produced in experimental models, simply increasing the percentage of energy coming from fat, was able to produce some synaptic effects. The focus was, again, synaptic transmission and connectivity. These are experiments not in model of multiple sclerosis, but in normal mice. And what we found is just feeding the animals with high fat was able to cause a number of alterations in the brain of mice, phenocopying again the effects of EI, of inflammation. So there is an effect of fat diet of interleukin-1 beta, TNF-alpha, and some other markers of inflammation, and an associated alteration of synaptic transmission, which was blocked by antagonists of TNF-alpha, interleukin-1 beta. So fat diet per se causes an inflammatory milieu affecting brain functioning and synaptic transmission through the production of pro-inflammatory cytokines. Of course, the idea is that the fat diet is affecting the brain, again, passing through gut microbiota. We found, in fact, that EAE per se, even without changing the diet, was able to produce some alterations in the microbiota of EAE mice, and this effect was exacerbated by fat diet. Also, patients with multiple sclerosis with unbalanced diet had some alteration in this index of gut dysbiosis, which is measured by measuring the concentration of some microbiota molecules in the urine. And we wanted to, again, continue our exploration of the effects of diet on brain functioning. And what we found is the fat diet was associated to altered blood-brain barrier permeability. This was an experiment done with the Evans blue, which normally doesn't pass the brain through the blood-brain barrier. In case of fat diet in control animals, again, you see there is an increased permeability of the brain associated to increased infiltration of T lymphocytes in the brain per se. And our experiments now are going in another direction to try to correct these abnormalities by trying to feed the animals, the high-fat diet mice, with some pre-probiotic supplementation. What we found is that even fat diet mice, the administration of pre-probiotics was able to recover some effects on synaptic abnormality caused by high-fat diet, and also to somehow correct the mobilization of T lymphocytes from the gut to the peripheral blood and the brain. You see here that in the gut, the percentage of T cells was increased in response to a pre-probiotic supplementation, indicating less release of inflammatory cells from the gut, and in fact there was less concentration, less number of T cells in the peripheral blood, and also in the brain. So just to conclude, the lifestyle, such as physical exercise and diet, is able to influence brain health, regulating gut microbiota, inflammatory and neurodegenerative processes in the CNS. Synaptopathy is a key and very sensitive marker of this activity. Physical exercise and diet, again, reduce the inflammatory neurodegenerative processes and promote synaptic plasticity and repair. And thank you, these are the members of the group working on this topic in the clinics and the laboratory. Thank you.
Many thanks, Diego, for this fantastic overview and also for sharing with us the ongoing research and the published data. Now we have the next speaker, Margaret Feinstock, Professor of Psychiatry at McMaster University in Canada. She's an expert of neurotrophins in neurodegenerative disorders and the modulation of neurotrophin production. Please, Margaret.
There we go. Thank you, Professor Comi, and thank you to the Prada, to the Fondazione Prada for the opportunity to attend this wonderful meeting and to speak to you today. I want to start by acknowledging the lovely talk yesterday by Chiara Chiarelli about synapses, because I'm going to start with the synapse. She made it very clear that synaptic dysfunction is a common feature across neurodegenerative diseases, and that this synaptic dysfunction occurs very early, often before any symptoms or protein deposition. I want to start, first of all, I have to figure out, there we go. I want to start by getting back to the synapse and making the point that communication across synapses is bidirectional. So we often think about the synapse in terms of its neurotransmitter. There are many different neurotransmitters, as you know. Many of them are very specific to different diseases, acetylcholine for Alzheimer's disease, dopamine for Parkinson's disease, et cetera. So the synaptic loss, the synaptic dysfunction across the brain is not global. It varies according to the area of the brain and the different diseases. But what's often not recognized is that equally important is the communication from the other side of the synapse, which is usually via a neurotrophic factor. This can be NGF, GDNF, or BDNF, for example. So BDNF is brain-derived neurotrophic factor, and that's my favorite neurotrophic factor, and you're going to hear a lot about it today. It's a small protein. You see here the crystal structure. It's dimeric. It has a molecular weight of about 32,000 Daltons. The colored areas are the parts of the molecule that bind to receptors. But most important are the biological functions of BDNF. And they include synapse maturation, synaptic function, synaptic plasticity, which we heard about yesterday, and the regulation of neurotransmitter synthesis. So for example, in Alzheimer's disease, you need BDNF at the synapse to potentiate and to mature the synapses, to strengthen them, and also to regulate the amount of acetylcholine that those cholinergic neurons of the basal forebrain need to produce to maintain our cognitive function. I should also mention that in addition to synapse dysfunction being a common feature of neurodegenerative diseases, it also contributes to age-induced cognitive dysfunction. So all of these actions of BDNF are necessary for learning and memory, and that's why maintaining BDNF levels is so important for cognitive dysfunction, both in aging and in neurodegenerative diseases. So we discovered many, many years ago that BDNF levels are decreased in Alzheimer's, and they're decreased very early in the disease. So what you see is the amount of BDNF in normal control subjects. This is postmortem human brain study. It's reduced significantly in mild cognitive impairment and even further in Alzheimer's disease. And this loss is correlated with poor learning and memory. So in this graph, you see people with normal cognition in blue squares. They have fairly high levels of BDNF, and they have good cognitive scores, whereas people with Alzheimer's disease, shown in gray circles, have lower BDNF levels and poorer cognitive scores. And the pink triangles in the middle are people with MCI, some of whom will progress to Alzheimer's disease and some of whom will not. So one of the important take-home messages, I think, about Alzheimer's disease and some of these other neurodegenerative diseases we're talking about in this meeting is that they're multifactorial. And there are many risk factors, and there are a few of them that I've shown here in this slide in yellow. Of course, aging is the biggest risk factor for Alzheimer's disease. Also, other comorbid diseases such as cardiovascular, obesity, diabetes, and of course, there are genetic risk factors as well. Most of these risk factors end up causing inflammation, oxidative stress, and energy deficits, which are at the core of these neurodegenerative diseases. And these can increase the production of amyloid beta and hyperphosphorylated tau in Alzheimer's disease, but they can also cause directly loss of neurotrophic support. And in this case, for example, BDNF is reduced by oxidative stress. So the reduction of neurotrophic factors happens in Alzheimer's disease both directly through these contributors and also directly through the production of these misfolded proteins. Of course, the loss of trophic support, as I hope you understand, leads to synaptic loss and also to neurodegeneration, both of which cause cognitive impairment. So there are things that will mitigate the loss of neurotrophic support: exercise, diet, and environmental enrichment. We've already heard about some of these and the effects of exercise, which are mediated via BDNF. So loss of BDNF in aging and in Alzheimer's disease leads to synaptic dysfunction and to memory loss. So in my lab, we ask, how can we raise brain BDNF levels? And we know we can't take BDNF as a pill because it doesn't get across the blood-brain barrier. We can't inject it directly into the brain because if you inject a bolus of BDNF, that causes excitability of neurons and you get seizures. But we know we can raise BDNF levels by getting plenty of exercise, which we already heard about, getting lots of social activity, eating a healthy diet, which we heard about yesterday and today, and getting plenty of sleep, which we also heard about yesterday, and reducing stress. So some years ago, I did a study in collaboration with Carl Kottman at UC Irvine, and he used a colony of beagle dogs that were aged. And beagle dogs, like people, they're very good models because they get cognitive decline as they get old. Many dogs do, and like people, some are cognitively impaired and some are not. But dogs, as they get older, also get plaques, and there's some evidence that they get tangles as well. So they're a fairly good model for Alzheimer's disease, but an even better model for age-induced cognitive decline. So in this study, we had a group of young dogs that I measured the BDNF levels in, and you can see that they're pretty high compared to the aged control dogs, which had about 40% of the amount of BDNF as the young dogs. And this is very comparable to that picture I showed you at the beginning, where in Alzheimer's disease, people get down to about 40% of the level of BDNF as in normal. In this study, which lasted 2 years, we had 3 groups of dogs. One was given an enriched environment. That meant that they were group housed. They had lots of interaction with trainers. They went on walks. They played games. Another group of dogs was given an antioxidant diet. And the third group of dogs was given both the antioxidant diet and the enriched environment. And as you can see from this graph, which shows BDNF levels in the brain, the dogs that were given both the diet and the exercise had levels that were indistinguishable from the young dogs. And to show that this BDNF increase made a difference, we tested the dogs, and this graph shows the number of errors in a memory test. And here are the control dogs, the old ones which had no intervention, made a lot of errors, but the dogs that were given enriched environment and antioxidant diet made many less errors on these memory tests. So then we moved to a mouse model. This is a standard model of chronic unpredictable stress. And when you stress mice, just like when you stress humans, they have reduced BDNF in the brain. And so these are the stressed mice, groups of stressed mice, and you can see they had a lot less BDNF in their brains than the control mice. And the only mice that recovered their BDNF levels had both voluntary running wheel access and a high antioxidant diet. And you can see that those mice had levels the same as the controls. So then we moved to a human study, and this was done in university students. And this, of course, we can't control the diet in people like we can in mice or dogs. So in this study, we only used exercise as an intervention, and we used high-intensity interval training, also known as HIIT, or in this paper that we published, HIIE, high-intensity interval exercise. So in this type of exercise, it's a very short, intense exercise, and after a brief warm-up, you do 5 different bouts of 1 minute on an exercise bike at maximum effort, and then this is interspersed with 1.5 minutes at 30% of your peak effort, and then there's a cool down, and then there's a rest period of 10 minutes. And the control group didn't do any exercise, but they sat for the same amount of time. Now, these students started as, we only recruited sedentary students. These were students that had less than 1 hour of exercise per week. And we subjected them to this HIIT protocol 3 times a week for 6 weeks. And at the end, we measured their memory performance and their BDNF levels. And I just want you to focus on the light gray bars, because the dark gray bars had HIIT plus cognitive training, which was like essentially computer memory games, and that didn't make a difference in this study. But basically, the HIIT 6-week protocol of exercise increased memory performance, and this was in a high interference task compared to the control sedentary students. And at the same time, we didn't find any difference in the BDNF levels, and this puzzled us a bit until we separated out the students who actually responded with an increase in fitness compared to those who didn't. So the students here had an increase in their VO2 max, a measure of fitness, and these, after the 6 weeks of HIT, didn't have this response. And so the ones that responded with increased fitness had increased BDNF, and those that didn't did not increase their BDNF. So we took serum samples both before and after the exercise, and this is after a single session of HIT. The first session, we took blood before that session and then after the 17.5-minute HIT session. And you can see that even a single session of exercise raises your BDNF levels. And this is really good news, except that the bad news is that the levels drop again quite quickly. So hence the 3 times a week, because by the time in another couple of days after this first increase, the BDNF levels go back to normal and you have to exercise again to get it back up. Anyway, this was very interesting to us that a single HIT session would do this. So then we moved to the question of how does the exercise raise those BDNF levels? And the model is that exercise stimulates your peripheral organs, such as bone, skeletal muscle, and liver, to secrete various factors and hormones into the blood. So for example, the bone secretes osteocalcin, the skeletal muscle secretes factors such as lactate, irisin, and cathepsin B, and the liver, IGF-1. And these factors go into the circulation, and they eventually either cross the blood-brain barrier or end up as metabolites or downstream signaling factors that cross the blood-brain barrier, and these increase BDNF levels in the brain. So to examine this further, we first looked at osteocalcin in the serum in the same samples where we'd seen a rise in BDNF levels after a single HIT session. And in fact, we also found an increase in osteocalcin. So that to us meant that osteocalcin could be one of the factors that is raising BDNF levels immediately after a single HIT session. Then we looked for a skeletal muscle protein, in this case, FNDC5 irisin. FNDC5 is the precursor, and then irisin is cleaved from that, and it's the irisin supposedly that goes to the brain. But we didn't see any change in the serum of irisin after either a single HIT session or 6 weeks of HIT. Oops, oh, I lost a slide. Okay, my next slide was supposed to be a graph showing you that in, thank you, if you can come back to my second to last slide. I don't know what happened to that slide. Okay, it shows that if we harvest extracellular vesicles from the blood, instead of just assaying the serum, we can find an elevation of irisin after 6 weeks of HIT. So our current hypothesis is that the short-term increase in BDNF is due to osteocalcin, and the long-term rise is due to irisin. What we're studying now is we're using a cell culture system to try to figure out which isoforms of these various hormones are the active ones, which ones cross the blood-brain barrier, which ones are the active ones in terms of upregulating BDNF. And the hope at the end of all this is that we can come up with some kind of treatment or pill or whatever to increase BDNF levels in those who cannot exercise for whatever reason, like frailty or injury, or so hopefully if we can find a way of raising BDNF levels other than exercise and diet and, you know, all these other lifestyle changes, then we'll have a pharmacological approach. So I want to thank my past and present lab people, in particular the 4 graduate students at the bottom. And unfortunately, this is an older slide, so I don't have Sama Jabari's picture on this slide, but she's my current PhD student, and she's here at the meeting, and she presented a poster. So if you want to talk more to her about these kind of studies or some of the other ones that she's doing, I'm very lucky to have a really great lab full of good students. So thank you very much.
Thank you, Margaret, for this wonderful presentation. Now we have the next speaker, Andrew Summerlett. He's professor of psychiatry at the University College of London. He's member of the Lancet panel for dementia prevention. And his presentation is about a very complex and interesting area of brain protection. Please.
Okay, thank you very much. Thank you for the invitation to come and speak at this really interesting and important conference. I'm Andrew Summerlad, so I'm a psychiatrist. I work in dementia services in London, and I do research on the interplay between social participation and dementia. So just to explain what I'll be talking about today, I'm going to recap the evolving dementia risk that we've seen over time, suggest perhaps that social participation is also changing over time, and then summarize evidence from observational studies and the potential limitations of those. Think a little bit about mechanisms, although I feel that's been extensively covered this morning, and also think about interventions and the future potential for prevention of dementia through social participation. So I think we can agree that dementia is a major global health challenge. Dementia itself is one of the most feared diseases for older people, huge societal cost as well. And to recap, that there are increasing numbers of people with dementia globally, and that's driven by demographic changes. So at the moment, there are 50 million people worldwide with dementia, and we are expecting that to more than double over the next 20 or so years, particularly driven by changes in low and middle income countries. But the really interesting and important for us observation that's been made in the past 10 or 15 years is that dementia risk appears to be evolving over time. So studies have found that incidence rates, age-standardized incidence rates, have decreased by around 20% over the past 20 years. I've listed some countries here, the US, the UK, France, Holland, Sweden, where this change has been observed. And what this means in real terms is that someone who's 80 now probably has around a 20% lower chance of having dementia than someone who was 80 20 years ago. And that's demonstrated in the figure on the right, that comparing older cohorts of individuals to more modern cohorts, and all the plots on the left-hand side of the line here show studies where more recent studies of older people have shown lower rates of dementia. And that's echoing the talk yesterday, showing that that's been shown in the Rotterdam cohort to some extent, also Framingham as well. So it's difficult to have a very clear understanding of why these changes have been observed, but the supposition is that these changes are due to lifestyle changes over time. So people have received far more education as education has been mandatory, and better treatment of cardiovascular health is also likely to be one of the main drivers of this. But that might suggest, well, this is just a problem that's resolving itself, but clearly there's the possibility that these trends could reverse. So there are increased rates of obesity and diabetes seen over the last 40 or 50 years. And there's, to turn to social participation, there is a very clear kind of popular sense that societies are becoming more disconnected, more fragmented, and people are becoming more socially isolated and lonely as a result. And this is exemplified by the US Surgeon General's report last year, which is sort of catchily titled An Epidemic of Loneliness. And they show some empirical evidence from studies over the last 20 or so years showing that people report higher levels of social isolation now compared to 20 years ago. People have less social engagement with their friends. So this evidence has not been seen consistently across the world. There's plenty of studies that suggest that actually levels of loneliness have remained stable, but there's certainly a potential sense that actually as society is changing, people are at higher risk of reduced social participation. And of course, we saw another epidemic that had a huge impact on social participation in the last few years, with COVID requiring us all to keep distance from other people, and brings further attention to the possibility, particularly in a kind of post-COVID world, that we are less socially connected. So all this suggests that social participation is one of these factors that might be going in the wrong direction and requires further study to understand how we might be able to improve it. So most of the evidence that I'm going to be talking about today comes from observational cohort studies. Ideally, we would want randomized controlled trials to be able to be clear about the causality, but these are impractical, unfeasible, probably often unethical. So instead, most of the evidence comes from studies of this nature, where cohorts of healthy, middle-aged or older people have their social participation assessed and are assessed at a later time point, hopefully many years later, for the presence of dementia. And just to flag up some of the particular kind of methodologically important factors, which I'll come back to during the talk today, there are various factors that give us stronger confidence in a causal association, one of which is having this arrow should be as long as possible. We should have long follow-up. We ideally would want to find dose-response relationships, so more social isolation being more of a problem. Considering the mechanisms, we want this to be a biologically plausible relationship. Ideally, evidence that changing social contact makes a difference, and generalizable evidence, so consistent evidence across different settings. All of this gives more confidence and is often ignored in studies that and in the popular media that talks about dementia risk. All of these stories are from one British newspaper, which you can see is low cost and quite low quality as well. Within here, for example, there is one suggestion of kind of greater social participation: stay married to halt dementia. So we'll kind of focus really in a little bit more detail about the strength of the evidence for this. So I want to just establish some terminology. Social participation is a term that we and others use as quite a broad term that encompasses various different aspects of social relationships. But terms are often used interchangeably, and it obscures quite a lot of the research evidence. So by social participation, I'm talking about engagement in social activities with other people and contact with social networks. And within that, we might be able to pull out different components, like the amount of social contact, a quantitative measure of how often people see or communicate with their family or friends. Social networks tends to describe a sort of the structural nature of the spread of individual social relationships. Social isolation, a term I've already used today as well, refers to the counterpart, the inadequate quality and quantity of social relationships. And then at the bottom there are terms that more closely relate to experienced or subjective components of relationships like loneliness and social support. So taking an approach to look at all of these, we did a review of systematic reviews, essentially, where quite a lot of the evidence that I'll be talking about today comes from. And you can see here that dividing social participation into those different components, we see some fairly consistent relationships from systematic reviews of observational studies. So again, to orientate us, any of the points on the right are systematic reviews where the combined result suggested that having lower level of social participation was associated with an increased risk of dementia. And you can see that in the top panel, participation in social activities seemed to be associated, or less participation in social activities was associated with higher dementia risk, as was less social contact, being unmarried, or being lonely. All of those seem to be linked to higher dementia risk. And then there are some components like the size of your social network, having less social support, or being unsatisfied with your social networks, where there was less consistent evidence. But I want to really focus on scrutinizing this in a little bit more detail, and particularly with a focus on the potential for, I mean, there are many potential biases and limitations in this research related to measurement error, to confounding, and I'll address some of those in talking about some of these studies individually, but I'm going to focus particularly on the potential for reverse causation to underlie some of these findings. So as we've heard in several of the talks, well, study of dementia risk is particularly prone to reverse causation bias, and that's because there is a very long prodrome for the development of dementia, with neuropathology being evident at least 10, probably quite a lot longer, many more years before the onset of clinical dementia. So what that also is likely to suggest is that some social and behavioral changes are also occurring in the period prior to dementia. So it raises this particularly important question that if we find an association between a low level of social participation in someone who is, say, for example, 75, and we find that that individual is more likely to go on and develop dementia 5 years later, it is unclear to what extent that low social participation has caused dementia, or to what extent the prodromal symptoms of dementia in their early development are causing that individual to withdraw from their previous social participation. My other strand of research is examining the underlying social functioning changes in people with early Alzheimer's disease, and focusing on social cognition deficits that occur. And it's clear that people with dementia experience early changes in social cognition, which make it more difficult for them to continue to engage socially with other people. And it's also very clear that some of those changes precede dementia. And also there are other factors as well that are likely to be changing, like memory difficulties and language, that all might cause this, the lower part of this relationship to be the case. So I hope that, well, I'll talk more about whether we can pick out whether the studies that have been conducted might reflect, well, which direction of this association, or perhaps both. So to focus on the first group of studies at the top that looked at social activity participation, many of these studies that have previously been conducted have measured social activity participation only a few years preceding dementia. So they've noticed that people are reducing activities perhaps during the prodromal phase. So specifically, I'll show a figure from one of these studies. Here are 7 studies of social activity participation and dementia risk, and 5 of them reported a positive association on the left here. But all of those studies had less than 6 years of follow-up, so less than 6 years interval between assessing social activities and dementia risk. And only two had 9 and 12 years respectively of follow-up, and those are the two that didn't find an association. So it suggests that when you have a longer period of assessment and you try and avoid reverse causation, then you're more likely to find negative results. And since that review, 2 studies have had similar findings, really. So looking at, when they look at longer than 10 years follow-up, found negative results. So one is a study of the Million Women study and found a negative association. And our study of the Whitehall participants had a similar finding. So we were able, the Whitehall study has serial measurements of various different social domains. And so we could look at relationships across different periods of the lifespan. And when we looked with the longest follow-up, so we looked at people aged around 50 and looked 18 years later, we didn't find an association. And it was only when the duration of follow-up was less than 10 years, was down at 8.3 years, that was when a positive association was found. So all of this suggests very clearly that social activities are declining in the prodromal phase of dementia, and it is not clear that taking part in social activities reduce dementia risk. It doesn't obscure it. It's possible that there is still an association and that there is a bidirectional association, but it's not entirely clear. Moving on to social contact with other people. Again, previous reviews have conflated studies where there is generally quite short duration of follow-up. So most of the studies had less than 4 years follow-up. That's one of the studies included in my figure earlier on. But some studies have tried to find ways to address that. One is by looking at marital status. Being married is to some extent a marker of lifetime social contact, because you're more likely to have lived with someone and had frequent social contact as a result of that. That's likely to have happened many years before the onset of dementia. It probably is something that's less prone to reverse causation. And there's quite clear evidence that being married and being a parent is associated with lower dementia risk. So you're probably having more frequent contact. You've also got people who might help you and help you to maintain better health. But also studies conducted by us and others have found that with more than 10 years follow-up, there is a fairly consistent link between more frequent social contact with other people and dementia risk. And this is a figure from our study, again, in the Whitehall cohort, where this time we did find a link with very long follow-up. And from the figure here, it suggests to me, to some extent, that there's a dose-response effect. More frequent contact on the right was associated with a lower risk, and less frequent contact, a higher risk of dementia. So I think, taken together, all of this evidence more strongly suggests that social contact leads to lower dementia risk. And finally, focusing on the component of loneliness, systematic reviews have found loneliness associated with future dementia. There are difficulties with these studies, partly relate to measurements, potential for measurement error, and the confounding effect of depression and also social contact. And what I'd say is that we feel less certain about this. Some studies do account for social contact and find, even when you take into account the amount of social contact that people are having, that loneliness is still associated with dementia risk. But it's unclear exactly how this should be teased out and whether it's important too. So I want to flag up that most of the studies that have been conducted were studies from Europe and the US, and clearly social relationships and behavior are really closely linked to culture. So some cultures are more closely integrated and they have better support for people into old age. And an example from a study that our colleague at UCL, Nahid McAdam, did showed a huge variation in the prevalence of social isolation from in the 1066 study, which used consistent methods to assess this across different settings. So a very small proportion in Latin America, a much higher proportion of social isolation in India. So all of that suggests that this risk factor has a different weight of importance in different settings. As I said, I think others have probably covered this in better detail than I will be able to, but thinking about mechanisms, I think the most convincing argument is that social participation helps to build cognitive reserve, and that's supported by this really nice study from a post-mortem study which suggests that social contact modifies the relationship between pathology and cognition. So here, in people who had high level of social contact, having more pathology had less of an impact on cognitive function than in those people who had low social network contact. It seems likely that it defies the having more social contact defies the kind of importance of dementia pathology, and that's certainly consistent with I think what we can potentially tell people. I think there's nothing. Socializing with other people is the best exercise for your brain. It's something that requires you to utilize multiple different cognitive domains: memory, language, humor, social cognition. So all of that suggests that it's likely to have a positive effect. Other potential mechanisms, as I said, might be reducing stress and inflammation, cardiovascular risk, because perhaps you're more likely to eat healthily and take exercise, and lowering depression, which might all have a benefit. In the last few moments, I'm going to talk about the interventional evidence. So I think it's quite unclear how best we should intervene. Again, for anyone who has elderly relatives, let alone seeing patients, it's difficult to persuade people to change their socializing behavior. It's difficult for any of us to modify this. So social changes might be impractical. It's clearly inappropriate to experimentally manipulate some aspects of socializing, and they would need to be culturally appropriate, and follow-up in all of these studies is a challenge. There have been studies looking at social interventions for cognitive function, and summarized in a review, 3 RCTs with which tended to focus on interventions where people were put together in group activities led by a facilitator. 2 of these showed a positive effect on some component of cognition, and 1 found a non-significant effect. All of these were too small and short to show any effect on the risk of incident mild cognitive impairment or dementia. But it suggests one potential approach. And clearly the direction of travel in preventative interventions are multi-domain interventions, and looking at the Cochrane review of these, 2 of the included studies and 1 subsequent study that's been done contain social elements within their multi-domain interventions. So those are largely addressing physical health components, but there were social elements through group meetings or promoting social activities, and all of these have had a small effect on cognitive function, but the interventions themselves are quite time-consuming and intensive and costly. So it's difficult to think exactly about how to expand these at a population level. And I suppose that moves me to my last slide here, which is that individuals have limited ability to change their own social participation, and it's difficult to do that. And perhaps individual approaches have the potential for perpetuating inequity. So thinking about policy approaches, we think is likely to be the most important way to do this. So approaches to try and create environments which support people to socialize more into their old age, particularly during retirement, which is a potentially important transition point. So examples are shown on the right of the slide here, providing socially connected housing, provision of volunteering and activities for older people, and really emphasizing a return to pre-COVID levels of social participation is likely to be a really important and most beneficial approach to increasing social participation. So with that, that's my last slide. Thank you everyone for their attention, and look forward to answering questions. Thanks.
Many thanks, Andrew. I invite you, all the speakers, to join me for the discussion. So, are there questions? There.
Thank you. Hi, Carly Tanner from UCSF. Thanks for some fantastic talks. I have a question for Dr. Fonnerstock. So do you have any comments on, there's a phase 1 study, the second round of AAV-associated BDNF that's starting in the States, I think targeting entorhinal cortex for Alzheimer's, compared to your sort of much more holistic approach of, changing behavior of physical activity and increasing BDNF. How do you think about those things?
I think it's a valid approach. It's better than a simple injection, a bolus injection, which, as I mentioned, can cause seizures, because the AAV vector allows kind of a slow release of BDNF, which is good. On the other hand, it's not very physiological. I believe things like exercise and diet and social activity, for example, provide BDNF as it's needed in the right amounts in the right place. And I think that's probably much better for brain function than simply, adding a lot of BDNF.
Yes, there's a question here, and Lice.
Wonderful talks. Two questions for everybody. One is about the role of lactate. Lactate, BDNF is a great promoter of plasticity, but there is some evidence that lactate can act not just as a fuel for energy and so on, but directly as a factor promoting plasticity. Some comment about that. And the second question is about depression and social activity. Depression is present earlier on in all the neurodegenerative diseases, is associated with lack of plasticity. And what triggers what? Is depression triggering social, decreased social interaction, or vice versa, or both? Is there a way to districate the three scenarios? Thank you.
So I can answer the first part about the lactate. Lactate can also increase BDNF levels. So you're correct, it doesn't simply act as a fuel. And there's studies that are, including in our lab, that are trying to figure out what the pathways are by which lactate and BDNF can regulate each other. Regarding depression, I should also mention that BDNF is dramatically reduced in depression. And so possibly that's a prodrome for neurodegenerative diseases.
Diego, do you want to?
Yes. Yes, for lactate, I would probably say that is so interesting how now the metabolism of the cell and their function in terms of synaptic activity and so on is increasingly recognized to interact closely. And for decades we have been probably considering metabolism as just what the cell needs to do their activities, but without influencing them. Now we know that there is a shift between metabolism based on the specific activity. This is true for the immune system, but also for neurons. 2 days ago, just reading the table of contents of Journal Neuroscience, there was a paper demonstrating a role of a key enzyme of metabolism of the Krebs cycle, probably, I don't remember, and in other neuronal functions. So I think that this is an interesting area of research, and lactate, the example that you did for lactate, is in this type of evidence. For the relationship between BDNF and depression, what causes what, is not that simple. I think that probably some information could come from studying some individuals where BDNF defects are for sure primary. I'm mentioning the Valmet, for example, polymorphisms of BDNF, which are well recognized to have a reduced activity and the metabolism of BDNF. They for sure have reduced plasticity, reduced hippocampal volumes. They respond poor to rehabilitation after an ischemic events. They are also so. I think that even in this case, apparently BDNF comes first because there is also an association. This was first, but you are an expert on that, demonstrated in depression in comorbidity with Parkinson's disease and other diseases, where it was clearly shown that the polymorphism of BDNF influences the mood in these neurodegenerative diseases, but now evidence is also exists for primary depression.
More comments on depression?
Yeah, thank you. Yeah, so I mean, I agree absolutely that there's likely to be a bidirectional, a circular relationship between social isolation and depression, and I think that there's quite consistent evidence that looks at the interplay between isolation and loneliness and depression, and clearly I think it's important differentiating those three elements: depression, dementia, social isolation. It's important from a diagnostic point of view to try and understand on an individual level, when you're seeing people in clinic, how best to approach and diagnose. I think it matters less from the point of view of intervention, because I think if there is this relationship, then addressing social isolation, addressing depression, those are all going to be important elements, and that's what we would say. I think if we see people in clinic, try and manage depression, try and promote social activities and other sorts of social interventions, because if you can have some impact on that circle and that relationship, then it's likely to be beneficial.
Alessandro?
Well, first of all, thank you, all of you, for the presentation. I think you went through three important factors: social relationship, diet in some way, and exercise. Now, the point is, do we have a dose-dependent effect? So how much of exercise? What kind of diet? When you talk about fat diet or fat-based diet, then I remember that ketogenic is based on fat, and actually it increases our resilience to some disease. And when we talk about isolation, it's also true that sometimes isolation is something that we want to. So I think that some all these complex aspects need to be probably object of a more complex analysis. And there are some data that are arguing that isolation is a prodromal, let's say, prodromal aspect of what I call, a sort of a neurosenescence. So neurosenescence has to manifest with cognitive decline, behavioral decline, even exercise and motor decline. So dementia, when we talk about Alzheimer's, is a completely different issue. But neurosenescence means that the brain is not doing properly the action that it's doing for. So we have probably to separate what is resilience, what is resistant to neurosenescence, and what is the effect of different other diseases. But I thank you for showing how complex it is, even understanding big data. And I think that probably we will be able to give a sense of all this data, but I think the reality is really, really complex.
Thank you. Yeah, no, I mean, I think those are really important points. I agree that none of this research takes into account individual preferences for socializing, and that's incredibly important when seeing individuals. It's only going to be more stressful and presumably more harmful to push people into activities that they might struggle in and might not choose. I think it remains the case that more, a more enriching environment is likely to be beneficial for cognition. That's borne out by the evidence that the fellow speakers have suggested, as well as, there's the kind of evolutionary approach that suggests that actually, clearly there is a social benefit to being with other people. So I agree. I think on an individual level, it's difficult to those are all factors that need to be considered.
Thank you, Alessandro. You're perfectly right, of course. For exercise, what exactly means, which is the threshold where exercise is beneficial and the other threshold where it can be detrimental. Well, as you know, there are some consortia around the Europe and the States trying to define what exactly exercise means. And for multiple sclerosis, I can say that there are some published indications indicating 3 hours per week aerobic activity as the minimum, and also probably the maximum is just around 3 hours, as those recommended to cause beneficial effects. And of course, there are some assumption behind this indication that needed to be further elaborated and probably leading to future revisions of the indications. But I would probably agree with this, even in our experience, where individuals are asked to give us information what kind of exercise they do. Essentially, physical activity means not simply, stepping just one floor every day or something like that. It's something that should be run with some leisure and rewarding also effect on that. And for diet, yes, of course, we also agree on that. I'm not an expert, but as far as I know, for ketogenic, it's true that most of the caloric income from fat, but there should be a completely abolish reduction of carbohydrates, and this makes the difference because, of course, if you force the system to use only fatty acids for metabolic purposes, you have an anti-inflammatory environment, as has been demonstrated in several works, preclinical and clinical data. Adding fat, as we did in our experiments, trying to mimic what we do when you go to the McDonald's, is not just exactly ketogenic activity.
Thank you. It's very important, the dose response comment you made. Major gaps in our knowledge in exactly with BDNF as an endpoint, raising BDNF levels as an endpoint. Very little information is available on how much, at what interval, what kind of intervention, what kind of exercise is needed. There's evidence in the literature, pretty solid evidence, that aerobic exercise is better than non, but the time courses haven't been established, and they need to be.
More incidence of ALS in soccer players. So maybe also that above certain dose of exercise, it not be so beneficial. So it's an open question, the amount of exercise you need. More questions?
Oh, okay. Thank you, everybody, for a great session this morning. I just had a question for all three of you. Did any of you see any sex differences in your studies at all? And if so, what it was?
We see differences in BDNF levels. Women have higher BDNF levels than men.
So did you see that did you see that difference in your exercise studies?
No, we didn't have we did have a big enough sample size. No, we didn't see any differences, sex differences in the exercise.
No.
Yeah, and similarly, I'm not aware of evidence in relation to social participation. It's a really interesting point because clearly there are different cultural expectations, particularly in older people. So yeah, it's a great point.
I'm sorry, actually, for Dr. Summerland, I was wondering whether you think online social networks can be beneficial or would they be different than participating in social networks in person?
So again, a great point, and one that I think we don't know the answer to, because these online social networks are relatively new. I think it probably comes partly back to the dose response point that, you know, the remote communication with other people has an element of the social relationship. You know, it certainly requires deployment of some of those kind of cognitive domains, but it's a watered down version, really. And certainly when we've looked at the link between social contact and other aspects like mental health, it appeared, so this is evidence during COVID, that remote contact by telephone and other approaches, computer, had some benefit on mental health, but not the level of benefit that face-to-face contact had. So I would expect that you'd see a similar relationship in relation to cognition, that there's likely to be more benefit of face-to-face contact, but something is better than nothing.
Giancarlo?
First of all, I also want to join to congratulate a really very highly scientific session. A bit a provocative question, Diego, to you. I think you have shown how toxic could be the inflammatory activity at the level of the synapses. So if we want to promote a reshape of the synapses, then we probably should also consider how we may take under control this inflammatory activity. In MS, we have a drug, natalizumab, that is able to block the trafficking. Should then we expect that if we use a combination of the two strategies, for example, exercise, and at the same time we try to limit the pro-inflammatory activity, we can pick up something that is difficult in other conditions.
That's a great idea. And yes, I think the point is well taken. We are trying to address this issue in these days, these weeks, because we found some differences in the responsiveness of synapses of male mice compared to female mice, and apparently the reason is the inflammatory milieu, which is in normal condition, is delivered to the brain by T lymphocytes in male only. So we want to somehow feminize this synaptic by using natalizumab to prevent, because the reason is not inside the brain, but from the circulating T cells. So I think this is just a way to answer your question, and I really believe that natalizumab and other adding trafficking inhibiting drugs could yes, be helpful. And it's not surprising that not for natalizumab, but to my knowledge, but for fingolimod, there is evidence that there is an effect of fingolimod increasing brain plasticity, LTP-like plasticity. Treatment with fingolimod, and you know better than me that there is literature showing that under fingolimod and also under natalizumab is higher the probability to experience inflammatory activity at the MRI without the clinical counterpart, meaning that synaptic activity is able to compensate the lesion. And this again could suggest your hypothesis that if you reduce inflammation by reducing the trafficking of inflammatory cells into the brains, you generate a pro-plastic environment in the brain that could be effective in increasing brain resilience to damage.
Increases BDNF levels.
More questions?
Thank you for a wonderful session as part of a wonderful meeting. I'm Eugénie Soutre. I work at Roche. And when I think about interventions, I have a question for Dr. Fenstock. I tend to think about or care a lot about responder, non-responder situations. And you had a slide where you addressed that there were VO2 responders, non-responders with your intensive exercise intervention. What percent in that or in similar analyses were non-responders?
That's a good question. I don't think I can answer that offhand. I'm going to have to get back to you on that. I'm very sorry.
Follow up?
Okay, please
I
do.
raised it
I apologize.
also
I don't
for-
have the numbers
Thank
in
you.
my head.
I raised it also for the general idea of thinking about that sort of influence when we look at studies that do and do not show correlations with exercise, for instance. Maybe we're dealing with a subset of responders or non-responders.
I should make my pitch then, which is that I think that it's really important to measure BDNF levels in the serum or plasma of people who are undergoing some of these interventions, because I think that can really tell you, a lot about the particular person's response in any kind of an intervention test.
But can we trust the BDNF plasma level? Does it reflect faithfully the level in the brain?
That's very controversial. There's some literature that shows that yes, it does. So what's very controversial is whether the BDNF in the brain can actually pass the blood-brain barrier either in or out. BDNF is made in the periphery as well. And so exercise may be increasing BDNF levels in the serum by increasing BDNF expression in the vasculature. So one's never quite sure, or in the lymphocytes, one's never quite sure exactly what the source of the BDNF is. But I think it's fairly well established that when BDNF goes up in the brain, it also goes up in the periphery. So yes, we use that as a proxy for brain levels. Thank you for that question.
Thank you. There is a question? Yes, please.
Okay, my question, thanks, is to Andrew Sommerlad. So it's regarding the reverse causation bias. So a paper was recently published that examined the Lancet Commission risk factors on dementia using Mendelian randomization. So my question is there Mendelian randomization studies investigating the link between social deprivation and dementia? And do they support your findings that when you increase the follow-up, you do not find significant associations between social deprivation and the risk of dementia?
Yeah, thank you. So, I'm not sure actually about whether there have been any MR studies of social isolation in particular. I'm aware of some of the studies that have been conducted thinking about other risk factors, and I know that they find quite a variety of different findings, some that seem slightly counterintuitive. So I think it's a really important question. It's a really important approach to try and think about some of these risk factors. But I think it's not the only answer at the moment, and one that we should think more about.
Okay. One more question.
Do you hear me? Yeah. I wanted to ask you if you think there may be some connection between low social participation and the probability of having Alzheimer's disease in the future, because I thought like people with low social participation may have a variated diet, so they may have diabetes, and that's a risk factor of Alzheimer's disease. So do you think there may be some connection? Thank you.
Yeah, thank you. So I guess I didn't go into it in detail, but I would think many of these studies, our studies have considered, have thought about some of these potential confounding factors in relation to physical health. Again, I think they're complex relationships, because one would think that socializing more, as that would have a beneficial effect on your diet, on your level of exercise. these are potential pathways through which socializing is beneficial. So we don't want to completely obscure those potential pathways. So I think clearly being unwell, physically unwell, limits our socializing, but it's also quite possible that these are some of the pathways through which it's beneficial.
Okay, many thanks for this such a great discussion. No, we don't know if the coffee is protective factor for the brain, but I think we can go to our coffee break.
Can you hear me? Yeah? Okay. So good morning. We will continue with this session. I'm Jean-Christophe Corvol from Paris. I'm very pleased to chair this session on gene and other risk factors of neurodegenerative disease. And it's my pleasure to introduce the first speaker, Alberto Ascario, who's from here, actually. He was graduated in medicine at the University of Milan, but now he's a professor of epidemiology and nutrition at Harvard. And among his most notable contribution is the discovery of the relationship between Epstein-Barr virus and multiple sclerosis.
Thank you. And thank you to the Prada Foundation for inviting me to this exciting symposium. This is a topic that may be relatively new for many of you. The idea that infection have a role in Alzheimer's disease has been gaining momentum in the past few years. The idea is not new. It goes back several decades to the 1980s, but it's been sort of in the backyard until recently. And I think in part our work on Epstein-Barr virus and MS has contributed to reviving interest in this idea. This was a database, a large electronic database study last year, suggesting that infection can even, flu with pneumonia could even increase the risk of Alzheimer's disease by 4-fold. This seems sensational news, but the purpose of this paper is recognized by the author, in spite of the many limitations, just to raise interest on the possibilities. And recently, this is Science in August 2024, referring to the small group meeting in parallel to the main Alzheimer section of a small group of investigators that for years have been proposing that infection has a role in dementia. Now, I will focus only on one example because of time and also because of interest. So my voice tends to fade, but today should be okay. Herpes simplex virus. The herpes virus has established latent infection in the peripheral nervous system, the herpes simplex virus. But it can reactivate periodically and cause cold sore. The idea that it, as you know, can cause encephalitis, which is a rare, severe complication, but it's very sensitive. But it can, from the peripheral nervous system, can in fact reach the brain through several different routes, as shown on the top here. Do you see the, yeah. So there are several different routes. Can be through the peripheral nerves, to the blood-brain barrier, to the olfactory nerve. And it is, in fact, the virus has been found in the brain of people with Alzheimer's disease in the region that are more affected by beta-amyloid deposition and tau pathology. And once in the brain, conceivably can, even low level of viral reproduction could cause inflammatory reaction that could lead to neuroinflammation, increase the deposition of amyloid beta, the phosphorylation of tau, and neurodegeneration. Now, the problem with this type of study that are very intriguing, so you will find a pretty broad literature, and they seem very convincing, but the question is, you cannot really exclude reverse causation in any of these investigations that in humans are based on autoptic material. So the neuroinflammation and the beta-tau deposition, the beta-amyloid and the tau phosphorylation could themselves promote the invasion of the CNS or reactivation of the virus. Now, the direction of causation can be established in animal models, of course, and this has been shown, and these are a couple of examples here. On the top left is a mouse model in which recurrent HSV-1 infection seems to be due to the hallmark of neurodegeneration. And even in a three-dimensional human brain-like tissue, there you can demonstrate that the infection come first and cause the beta amyloid deposition. The problem with the animal models is that, this is just an example shown here, that the result of the infection can depend on the strain of the virus on the left, and also by the genetic of the host. So generalizing from these studies in mice to human is obviously not possible. So we are left with the task of answering this question, that is the key question in terms of causality. So that comparable in orange, hides an encyclopedia of complex issues. So it's just a little word there, but it's one of the key to everything. In some, to me, this is almost the definition of causality. If we say that the virus, that HSV-1, causes Alzheimer's disease, what we mean, if you have a person with Alzheimer's who has the virus, you're saying, well, if this person had not been infected with HSV-1, then they would not have Alzheimer's. Obviously, the individual level is counterfactual. Even in a population, you say, well, if these people had not been infected, they would have fewer cases of Alzheimer's. So the way we address this in science, usually do a randomized study, which you can do in mice, but not in humans. So what we are left with is a very challenging question that can only be answered by the most rigorous study. And I think, our study on EBV and MS has provided sort of a blueprint that we could think, can we do something similar in the case of HSV-1 and Alzheimer's? And this was conceptually very, very simple study, but it took 20 years to carry out. And it was made possible by a population of 10 million young men and women. You can see on the left, 7% were EBV negative at the beginning of the study. They were followed for over 10 years with repeated blood sample on average every 2 years. And the focus that we had enough people, because of the large population, were not infected with EBV. And we could demonstrate that those who remain EBV negative throughout the follow-up of the study have virtually no MS as compared with those who were EBV negative and became infected on the study. Now, the study was challenging for many reasons, but also was facilitated by the unique population source. Now, I just want to highlight a few details of this study because they transfer in some way to what we could do on Alzheimer's and neurodegeneration. So these are some obvious details, but very often they are forgotten, and they're really critical. We need to distinguish what is in elevation of antibody titers and what is infection. In diseases where we have in the case of Alzheimer's would be immunosenescence, in the case of MS, immune dysregulation, you can have an increase in antibody titer, and that may change the sensitivity, specificity of your test for infection. What we are trying to do here is to say who is infected and who is not. In the case of EBV, we were able to do this by combining the ELISA for VCA with a very specific Western blot. Another aspect to have a negative control, in the case of a MS, we had EBV as our exposure of interest, a CMV, a virus transmitted in a similar way as a control virus. As you see on the left, the seroconversion rate in individual for MS was much higher for EBV than in controls, but there was no difference for CMV. And the other useful aspect here is that the association of EBV with MS is extremely strong, was over 30-fold increase in risk comparing people who became infected to those who remain EBV negative. Again, on the right, you see CMV with no association. In the case of Alzheimer, you heard yesterday about the burden of Alzheimer. Even a 20% increase would be huge. You know, if infection with a common virus increased the risk of Alzheimer by 20%, that would have transcending implications for public health, particularly considering the possibility of a vaccine or an antiviral treatment. Finally, we could establish temporality by comparing the time of EBV infection here with the time of elevation of neurofilament. So referring back to the discussion yesterday on defining the disease in biological terms, the equivalent here for Alzheimer would be to demonstrate that herpes simplex virus infection precede, say, an increase on phosphorylated tau or precede any evidence of Alzheimer time pathology. And finally, this is looking at a broader range of viruses in case of MS to establish the specificity of the association with EBV. Okay, let's get back to herpes simplex and Alzheimer. There are some similarities here. HSV, as you see, this is the prevalence of infection with herpes simplex in the US. By age, as you see, the virus is very common. There are very few people who are not infected. By the age you become at risk with Alzheimer's disease. This is similar to the MS situation. Now, we have to distinguish seropositivity from being infected with the virus. So the issue that I mentioned before, we need very sensitive and specific serology. And then there is a sort of small aspect that sometimes gets forgotten. There have been several studies, and I will show you the results in a moment, looking at people who are HSV negative versus those HSV positive. And this study has been done with follow-up of 10, 15 years, but they don't have a follow-up blood sample. So they compare the two groups according to their serology at the beginning of the study. And during the follow-up, you can, imagine some of the people are negative will become positive. If you look at this, is 50 to 59, 60 to 69. It seems a small proportion, but your control group of individuals who are not infected, 27% will become infected during the follow-up, which would attenuate the contrast and bias toward the null any comparison between the two groups. So the next few slides, I'll go over some of the studies that have been done so far. In reality, there is a common denominator that is one blood sample at the beginning, a long follow-up, ignoring a new infection. So the limitations are obvious. But the results are somewhat intriguing. This is one of the first studies. 512 people in France over 65 years of age, 77 cases of Alzheimer, no significant association with being IgG positive versus IgG negative. That is the closest you can get to being infected versus non-infected. They reported an association with IgM positive in individuals who were also IgG positive. So interpreting this as a sign of viral reactivation, but as I mentioned before, you cannot build a causality on viral reactivation because the viral reactivation could still be a consequence of the disease. This is an expansion of the same study in the southwest of France, not the high proportion of IgG positive. In this case, they didn't find any overall association based on an expanded population with 156 cases of Alzheimer. So really close to one hazard ratio with the caveat I said before that some people may become infected during the follow-up. But they reported for the first time a significant interaction with ApoE. So in the ApoE group, 178 people with 43 cases of dementia, there was a marginal non-significant association with IgG, but a significant association with IgM. Separate study, this is a nested case control study in Sweden, 360 Alzheimer cases matched with controls. This is a truly prospective study in the sense it was nested in a cohort, non-significant odd ratio, but still a 60% increase in risk. No association in this case with IgM positivity, so in conflict with what I've seen before. ApoE was reported in a later paper that is shown here. And here also in this separate population, there is an interaction with ApoE4 that's shown here at the bottom. Still in Sweden, but a completely independent study. This is a cohort, 3,400 people, mean age of 63, followed for 11 years. Again, only one blood sample at baseline, 245 incident cases of EBV, 88% HSV1 positive at baseline. And in this case, there is no association with being positive versus being negative as a ratio of 0.99, but a twofold increase in risk on reactivation, quotes, because I'm not sure we know what IgM means. After adjustment is marginal significance. And this is the same study. Results are stratified by ApoE, again showing a possible interaction. And this is looking at, in the same study, looking at the cognitive decline. In cross-sectional study, individuals who are HSV positive seem to have a faster cognitive decline with aging if they are ApoE positive, but not if they are ApoE negative. But in IgM, in this case, surprisingly, were associated with a decreased risk of episodic memory decline. Finally, I think this is the last study in this setting. This is the most recent small study in Sweden, independent from the previous one. Only 36 incident Alzheimer cases, suggesting a twofold increase in risk for Alzheimer or dementia. So I cannot reach any conclusion. I wish I could give you a convincing presentation. To me, these results are intriguing and provoking, but not really reaching any conclusion other that we need to find an answer to this question. Now I'm showing some separate investigation here, just very briefly, just to sort of increase the mystery here. This is the Framingham cohort that you heard yesterday. So this is looking at people with history of cold sore, labial herpes. And those who have a positive history in the first 10 years of follow-up seem to have a lower risk of Alzheimer's disease. But this difference disappears over time. So when you extend the follow-up over 2 years, there is no difference anymore. What does it mean? I don't know. Maybe it's just people with better memory, they remember having had cold sore in the past than people who don't remember. I have no explanation here. Another interesting aspect is that anti-herpetic medication, these are electronic databases, study being done in Taiwan, in Korea, in the US. They show that people who took anti-herpetic medication had a lower risk of developing Alzheimer's than those who didn't. And these medications are given for symptomatic HSV infection, which could be a genital infection in addition to cold sores, and is typically acyclovir or valacyclovir. That was this is a study in the US showing a similar result. Note that here, having a positive history of herpes simplex overall is still associated with a lower risk of Alzheimer's, but it's actually much lower in individuals who take antiherpetic medications when you look with some sort of dose response. And finally, this is a study in Sweden showing a similar finding. The red line here, people with history of herpes who took antiherpetic medication have a lower cumulative incidence of dementia than those with without antiherpetic medication who have a positive history, and the controls are in the middle. Finally, you know, another little piece, you know, I don't know we will be able to answer this. I think it will take 10, 20 years of rigorous study if we want to answer this question, but I think we should answer this question. This is another intriguing case. I'm changing virus here to the herpes zoster. There is quite a cool study showing that since the beginning of vaccination against herpes zoster, there is evidence that being eligible for the vaccine is associated with a lower risk of dementia. The reason this is really cool is that, you know, people who get the vaccine typically have lower risk of everything, lower risk of dementia, lower risk of cardiovascular disease, just because of the health bias, the health vaccine bias. People who are vaccinated tend to be healthier than people who don't get the vaccine. But here is not comparing people who got the vaccine with people who didn't. Here is comparing people who are eligible for the vaccine, whether they got the vaccine or not, to people who are not eligible for the vaccine, exploiting a policy implementation. This data from Wales, that people born, you know, before a certain date were not eligible because we're too old, and those just born the day after were eligible. And this regression discontinuity is difficult to explain other than in causal terms. And this is a related study looking at the two vaccines, and this showed that the red line here is the new recombinant vaccine compared with the old vaccine, showing that the new recombinant vaccines the previous study was based on the old attenuated vaccine, seem to have even lower risk, but that is decreasing over time. You know, the time scale is, is several years. And if you look on the right, the hazard ratio goes down after the new vaccine, but then it seems to climb up over time. I think we have a lot of intriguing results, a lot of complexity, and I wish I could explain it to you. I've been digging in this data in the past few weeks, and the only conclusion I can reach, we do need to invest in studying this. And that's all what I think we can say at this time, and thank you for listening. And just a moment.
Thank you for the great presentation. Alberto told me that he has to leave, unfortunately, just after his talk. So we propose that we have 5 minutes of questions, and then we will have the other talks and the rest of the panel at the end of the session. So is there any questions? Yeah, over there.
Thank you. That was really amazing. Joanne Stratton from McGill. So my question, the military cohorts where you have like 10 million people that you can track over time, it's so powerful and it's done so much for MS research. I'm wondering if anybody's gone back and checked those same patients for whether they develop PD, Alzheimer's, ALS, kind of done a screen, because you have a lot of really strong baseline data.
The same population that we use for the MS study. Those are very young men and women in active duty in the US military. They tend to retire from the military before getting to the age in which those diseases become common, unfortunately.
So a study to continue to collect from that population would be interesting.
Well, in theory, it's possible through the veteran administration. I spent the last 3 years of my life trying to link the VA, convince people to link the VA databases to the DoD. We are making some progress, but still a long way to go.
Thank you, Professor Askerio. I'm René Carvajal from Barcelona for Multiple Sclerosis Center of Catalonia. I have a question for you regarding for the ABV vaccination for prevention for multiple sclerosis. what is your position about the optimal timing for administering this immunization, considering that Epstein-Barr virus exposure is usually in early life? And do you believe that this vaccination would be suitable to preventing MS? Because now there are a few clinical trials running and cover this important topic. Thanks.
Yeah, that's the EBV vaccine is a very complex question. So if we had a vaccine that prevent infection, right, that completely prevent infection, and with lasting immunity, then vaccinating in early childhood would be ideal, right? Preventing even in infancy to preventing infection. But if that only delay the acquiring the infection, in theory, this could backfire. If you get infected then when you are 15, instead when you are 2 or 3 years of age, you could make things worse. But most vaccines don't really prevent infection. They just modify the immune response to the virus. How that will impact and reduce the risk of mononucleosis, conceivably and plausibly will reduce the risk of MS, but I think we need to prove it because it depends on the specificity of the immune response that the vaccine will induce.
Continuing on this, assuming that there is a causal relationship between EBV, typically, and MS, how can we move forward and try to answer the question, is it due to the specificity of the virus or the immune response? Because, yes, vaccine would avoid the virus, but you could also imagine to decrease the immune response when you actually have the infection, like for antiviral herpes.
Yeah, there are vaccines that modify the immune response in people who are already infected with EBV, and some companies are pursuing this way to modulate the immune response to reduce the risk. There is a possibility of using antiviral drugs to prevent the reactivation of the virus. I mean, there are many possibilities, but there is a lot of work to do.
Well, the evidence connecting EBV with MS is pretty clear and is there. Do you have any evidence of a strong association between EBV and other autoimmune disease? Is this something very specific to MS, or should we look at this relation between virus and host as a sort of a trigger that might address different diseases, different autoimmune diseases?
There is pretty strong evidence for lupus, reasonably strong for Sjögren, and I think not for other autoimmune diseases, in particular not for rheumatoid arthritis, even if it is being proposed. I don't think there is evidence there. Hey,
Maria Piamato. Thank you, Alberto, for a great talk. My question deals with pediatric multiple sclerosis. Yeah, because you know that very well that there are some cases that are negative for EBV, cases of multiple sclerosis, and that the diagnosis resists also after excluding misdiagnosis, MOGAD, and other diseases. So do you think that this is something peculiar to the immunological system of children, or do you think that studying this negative case could provide some insights on the link between EBV and MS?
Yeah, well, first, as you know, those children were EBV negative in the early study. When they follow up, they turn out to have, most of them had anti-MOG and other form of demyelination. There will be a few left, and I've always been restraining from saying that EBV is a necessary factor for MS, because when you define a disease clinically, almost inevitably, you will have some cases that have a different causality. Heterogeneity is a common, even if you define, the most typical, if you define measles in terms of the clinical manifestation, you will have some cases of measles that are caused by the measles virus, right? Poliomyelitis, another classical example. Now, with the vaccine, we have case of flaccid paralysis. It would have been, meeting all the diagnostic criteria for poliomyelitis, but they're not caused by the polio virus, they're caused from other enteroviruses. So very rare, but it happens. So unless you define the disease as being consequence of the virus, you will always have a few cases. But there are very few. We are down less than 10% with the new study.
Last short question.
Ross Patterson, UCL. So intriguing association between EBV and AD potentially. Just wonder if you'd share your thoughts about how mechanistically EBV might interact with the earliest stages in the amyloid cascade.
EBV and Alzheimer's disease?
Yeah.
I don't know that there is an association between EBV infection as such and Alzheimer's disease. I don't know that whether it's been rigorously investigated. Probably not, but I'm not aware of an association.
Okay, thank you very much. We will move to the next speaker. So Anne Mikey. She's a neurologist, but a neuropathologist also in the US, and she has working a long time to decipher the chronic traumatic encephalopathy and describe it. So thank you very much.
Thank you. Well, I just want to thank the organizers, Foundation Prada, as well as Professor Comi for inviting me to give this talk. I'm delighted to talk about our work at Boston University and the VA in Boston. I'm going to talk about traumatic brain injury in general and the risk of neurodegenerative disease, and you'll see it's multiple neurodegenerative diseases that occur after trauma. But I'm going to talk much more specifically about a disease called chronic traumatic encephalopathy, which occurs after a particular type of traumatic brain injury, and that's repetitive head impacts, the type of impacts that lead to concussion, but also asymptomatic non-concussive injuries, impacts to the head that occur in contact sports, in military activities, and occur to people exposed to interpersonal violence. So my objective, okay, I can't change these. My objective to the next 20 minutes, I'm just going to start out with what I want to convince you of in the next 20 minutes. CTE is a preventable neurodegeneration. If we limited hits to the head while exercising is beneficial, no question, to brain health. But getting hit in the head while exercising is very bad for brain health. And we can prevent this disease if we limit head contact, head injury in sports and other activities. It is caused by these exposure to repetitive head impacts. There's a lot of misinformation. There's a lot of obfuscation of the scientific data, led in part, or led, I'd say, in the majority, by stakeholders in not wanting this to be true, financial stakeholders. But the overwhelming evidence is that CTE is caused by repetitive head impacts. I'm not going to give you all that data, but there is a very substantial dose response with length of playing career in American football, in ice hockey, in rugby, and risk for CTE. I also want to say that hits to the head don't just cause CTE, a tau-based neurodegeneration. They also increase the risk for Parkinson's disease and Lewy body disease, independent of CTE. They accelerate, in the context of CTE, TDP-43 deposition, A-beta deposition, alpha-synuclein deposition, as well as the neurodegenerations AD, amyotrophic lateral sclerosis, limbic TDP-43 encephalopathy, Lewy body disease, Parkinson's disease, and hippocampal sclerosis. And then lastly, because we know the cause of this disease, the trigger for this disease, and because this disease starts quite early in life in many individuals in their teens and their 20s, this is an opportunity for us to study the lifespan trajectory of this disease in these young people to understand the mechanisms that lead to this heterogeneous neurodegeneration. Oops. So TBI of all severities, very generally, mild, moderate, severe, has a large annual incidence, 70 million worldwide. It is increasing in prevalence as neurocritical care improves. It accounts for 5 to 15% of the attributable risk for dementia. And the primary injuries are heterogeneous. There's a secondary neurodegeneration that's common to neurodegenerative disease, and that includes neuroinflammation, breach of the blood-brain barrier, ischemia, excitotoxicity, oxidative stress, metabolic dysfunction, and others. Very generally speaking, TBI of all severities increases the risk for all-cause dementia. It increases the risk for clinical Parkinsonism. It increases the risk for deposition of neurodegenerative proteins, and that's tau, it's A-beta, it's TDP43, and it's not on the slide, but it's alpha-synuclein as well. We know that very generally speaking, the risk for neurodegeneration increases with the severity of TBI, the cumulative number of TBIs, the age at the time of the TBI, and the frequency or the interval between TBIs. I'm going to talk specifically about repetitive head trauma, and as I said before, this is concussive and non-concussive injury. It occurs in the context of sports as well as military activities and other people who experience hits to the head. And the only known cause of CTE is this exposure to RHI. There are other contributing factors. There's genes, there's other environmental exposures, there's social determinants of health, but the single common denominator is hits to the head. CTE can only be diagnosed after death by postmortem exam, although there's a huge effort underfoot in many centers to make the diagnosis using fluid and imaging biomarkers, and it's following in the footsteps of what's been done in Alzheimer's disease. It has a clinical counterpart that I'm not going to talk much about. It has a mood disorder, personality changes, behavioral changes, as well as memory loss and cognitive changes. And in the end stages of this disease, Parkinsonism is also seen. It was described about 100 years ago by a pathologist who described the clinical symptoms that occur in boxing. My first experiences with this disease was in 2008. I described the clinical and pathologic features of 3 athletes, a boxer and 2 NFL players who had this extraordinary tau pathology. In my experience as a neuropathologist, I was very familiar with tau-based neurodegenerative diseases, and I'd never seen anything like this, and that began my real true passion and interest in this particular neurodegenerative disease. We started a brain bank to study the neurologic and neuropathological consequences of trauma back in 2008, and we now have close to 1,600 brain donors. It's by far the largest brain bank in the world dedicated to the understanding of trauma's effects. The diagnostic features, the hallmarks of CTE that are only found in CTE and no other disease are that the tau is in nerve cells around small blood vessels. So it's a perivascular tau accumulation, and it occurs at the crevices of the brain, at the depths of the cortical sulci. There is a pathognomonic lesion as described by 2 consensus panels of experts, and it consists of tau and nerve cells around a small vessel at the depths of the sulci or at the small crevices of the brain. It has 4 stages of severity, starting with the most mild, which consists of focal deposits of tau in the cerebral cortex, usually the frontal cortex, and it progresses over aging. Aging is the biggest factor for progression into a more widespread disease. In stages 3 and 4, you see medial temporal lobe involvement, you see widespread diencephalic brainstem involvement, and that's when you really see the more pronounced clinical symptoms. This is just another example of showing you low stage CTE with those focal cortical pathologies versus high stage disease when medial temporal lobe, hippocampus, amygdala, entorhinal cortex are involved, and then you get widespread cortical diencephalic brainstem disease. Back in 2019, Michel Godard and colleagues described the unique CTE tau. It's not Alzheimer's tau. It's not like any other tau in other neurodegenerations. It has a unique fold and a hydrophobic molecule at the interior of the fold, and it's unique to CTE. We know from studies using finite element and computational studies that the reason tau is deposited in those focal regions in the brain reflects the mechanics or the physics of the injury. There is greater tissue strain, there's greater tissue deformation around blood vessels and at the crevices of the brain, and that explains this unique deposition of the pathognomonic lesion. Clinically, you see cognitive change, memory loss, executive dysfunction, and dementia. And behaviorally, you see violent behaviors, explosivity, impulsivity, and loss of control. The patient usually knows something is going wrong. And that's a key feature that distinguishes it from many other neurodegenerative diseases. The individual is aware. There's often depression and hopelessness, and Parkinsonism occurs late in the stage. This was one of our papers that came back out in 2017. It was met with very polarized interpretations, but it showed in 202 American football players whose brains had been donated to our brain bank that a very high percentage of NFL players, 99%, had pathologic evidence of CTE. And it was also a high percentage in college athletes, 91%, and in 21% of high school football players. So this was not a prevalence study. This is not the prevalence in the general population of football players. This is a brain bank study with loads of selection bias. But subsequent studies have shown that the lowest possible prevalence of CTE in NFL players is around 10%. That's the lowest possible prevalence, and studies are being done right now to improve on that prevalence rate, to do more sophisticated studies establishing the true prevalence in NFL players. We know that this is a very rare disease in community brain banks. The Framingham Heart Study Brain Bank, which actually I direct, very, very unusual to see a case of CTE, 0.6%. And in those cases, the rare cases in community brain banks where we find CTE, nearly all of them have some history of trauma, either sports or military, whatever. In the cases of CTE with no history of trauma, it's because the methods they didn't ask the question, did this person, or the person wasn't available, did this person play contact sports or other activities? I'm going to show you a case of CTE. This is a pretty well-known case. It's a Netflix documentary. I think there's another series about this individual, Aaron Hernandez. But I want to point out that he's only 27 years old. I know we think of neurodegeneration as affecting people in their older years, but this is a disease that starts in youth. This is Aaron Hernandez, 27 years old. This is his brain when it came into the brain bank. It looked perfectly normal if you looked at it from the exterior surface. It weighed a lot. NFL players are big and they have big brains. I've come to very used to that. But nothing looks abnormal on the outside. But when we started to do the dissection, we could see that he had clear ventricular enlargement. Frontal horns of the lateral ventricles are clearly enlarged. And he had this atrophy of the fornix. And when we looked more carefully, we could see these septal perforations of the septum pellucidum. Very unusual. I'd only seen it before in a 45-year-old. So this was a clue that something was going to be found microscopically. And indeed, we found widespread lesions of CTE, the classic tau around vessels, depths of the sulci. And when you look at his coronal sections of his brain, a wide extent of the brain, you can see he has very substantial frontal lobe pathology, even though he's only 27 when he dies, stage 3 CTE. So we have many studies establishing the dose-response relationship between years of playing a sport, football, ice hockey, rugby, and risk for CTE. This is our study looking at football players where we found that for every 2.6 years of playing football, the risk for developing CTE doubled. And that was accounting for all sorts of simulation and inverse probability weighting. There are other neurodegenerations after RHI, and we've shown, and I don't have time to show you all the data, but there's accelerated A-beta deposition, there's a differential deposition of amyloid angiopathy, there's atypical AD, there's Lewy body disease and Parkinson's disease, there's TDP-43, hippocampal sclerosis, ALS, and late. NFL players have 4 times the risk for developing ALS as an age-matched general control. And not only that, but the ALS that develops in the context of CTE can occur in the 20s, so in some very young people. As the people age, and they tend to get multiple proteinopathies, so similar to all sorts of other neurodegenerative conditions. As individuals enter into their 70s and 80s with CTE, it's often a very heterogeneous, multiple neurodegenerations are found. I want to show this one study, and this is a study from last year where we looked at all the players that had come into the bank who had not reached their 30th birthday. At that point, there were 152 contact sport athletes, and we found when examining their brain that 41% had CTE, and most of that 41% played amateur sports. So they weren't elite athletes. These were kids playing high school sports or college level sports. And we found CTE in 41%. Most of them were low stage CTE, and we even saw an age relationship. Those with CTE were on the average 4 years older than those without CTE, but we're talking about a 25-year-old compared to a 21-year-old. And those who played football tended to play about 3 years longer with CTE than those who did not develop CTE. I'm going to show you something that might be a little disturbing, but this shows what these athletes are experiencing.
So what this is me explaining what's wrong with me. I've been depressed for a long time, and I've attempted suicide twice, and this is gonna be the 3rd time. It is my way out of my head. My head is pretty messed up and damaged. My thoughts never stop racing. I always have racing thoughts, and I can never think straight. But I've been depressed for a long time. Ever since probably freshman year of high school, I've had issues. And I covered it up with activities, going to booths, playing football, hanging out with friends, playing PS4. And over the years it got worse. Sophomore year was pretty bad at the end. And then junior year was easy. Senior year was the easiest. I'll explain that why as well. But the voices and demons in my head just started to take over everything I wanted to do. Hanging out with friends, enjoying time with family, riding my bike, and just daily activities started to become not so interesting for me. I'm really sorry for this. I really am. But I just want you to know that I can rest easy now. That's what's important about this. And I want you all to be happy. And I want you all to come together and bond over this. You know, don't feel guilty, like I said. Don't feel bad. You can be sad. I know you'll be sad. But at the end of the day, be like, you know, Wyatt's in a better place. This is what he wanted. I feel good for Wyatt. I feel happy for him. He suffered, and he's not suffering anymore. And that's what's important. And I want all of you to come together.
So this is Wyatt Bramwell. He was 18 when he died. He had stage 2 CTE. Now, I'm not making a link here between CTE and suicide because we know that for a young person to die, it's almost always an accidental death. So there is no clear link between suicide and CTE. But this is showing us that the individuals are aware of something going wrong with their head, and their only way out of it is to end their life. And that is what is concerning, that our medical profession doesn't take this more seriously, provide care and management to these individuals who are suffering, whose symptoms that can certainly be treated. And this is just the result of this study showing stage 1, 2, and 3, examples of stage 1, 2, and 3 CTE in these young athletes. We also found that they had more cavum, septum pellucidum, more dilatation of the frontal horns, more dilatation of the third ventricle in the context of CTE. And this is just showing you those brains. They not only had the tau-based neurodegeneration, but they had white matter loss, as well as perivascular hemocyanin-laden macrophages in the white matter. And what that means to us is that there was vascular injury with breach of the blood-brain barrier and escape of heme-laden blood cells into the parenchyma. And those were substantially increased in CTE, significantly increased, and they also had a dose relationship with years of playing football. So it's an indication that blood-brain barrier breach and other studies have shown inflammation are key features of the pathogenesis of CTE. This is just showing what those things look like under the microscope. So in conclusion, there are neurodegenerative consequences of RHI. Some of those are acute or subacute. That includes inflammation, vascular injury, breach of the blood-brain barrier, axonal loss, white matter degeneration, and metabolic dysfunction. It's unclear if all of those are progressive, but that remains to be seen, but they most likely are. And the chronic stages of this post-traumatic neurodegeneration are indeed progressive. And what we see are, in the context of tau pathology, it's CTE. We also see beta amyloid deposition as Alzheimer's disease. We see TDP-43 deposition as hippocampal sclerosis late and ALS. And we see alpha-synuclein as Lewy body disease and Parkinson's. So I want to thank all the brain donor families that made this work possible. I want to thank my funding sources. And of course, I was going to thank my team, but it disappeared. Thank you.
Thank you very much. Thank you. We will move to the last speaker. So Alexis Elbaz is a neurologist and also a professor of epidemiology in France, near Paris in Villejuif. He's worked a lot on the epidemiology of Parkinson's disease.
Jean-Christophe. Thank you very much for this wonderful meeting. It's really an honor to be here. So, so we've already heard many times during the past days that the number of patients with neurodegenerative diseases is increasing worldwide, and there's an urgent need to develop interventions that could help reduce the burden of these disorders. So for this, it's important to target modifiable risk factors that play a causal role and to obtain a good estimate of their effect. And by causal role, I mean that if you decrease the prevalence of these risk factors, or if you suppress them, then you will reduce the frequency of the disease. These slides show it's a simulation study done in France, in which the authors tried to examine the impact of completely suppressing three risk factors: hypertension, physical inactivity, and diabetes, on the prevalence of dementia, on the risk of dementia in 2040, so in a few years. And one of their main interests was to look at the effect on the risk of dementia, and also because these risk factors also are associated with mortality, what's the relative effect on prevalence and life expectancy. And what they showed, for example, here in men, is that if you reduce, so if you completely suppress these risk factors, the prevalence rate of dementia after 65 in 2040 would reduce by 34%, but the lifetime probability at 65 years of dementia in 2040 would reduce only by 17%. So this is due to the fact that we reduce the risk of dementia, but also because we're going to live longer, then the risk of dementia, in fact, also increases. So there's a balance between both of them. But the main assumption here behind this sort of risk of study is that the hypothesis that there's a causal effect on the risk factors of dementia. So in my talk, I will try to discuss some of the difficulties that we have in epidemiologic studies of neurodegenerative disorders to identify risk factors of these disorders. We've already heard quite a lot yesterday and today about this. So one of the biases that we discuss is reverse causation bias, to the fact that these diseases are very progressive and develop over many years. Survival bias is due to the fact that these disorders affect mainly old people. Diagnostic misclassification bias is due to the fact that these diseases are mainly, the diagnosis is mainly clinical, and confounding, which is better known, is because they have a complex etiology. There are many others, but I will focus on these ones, and mostly talk about these, give examples in the field of Parkinson's disease, which is my main field of research. So reverse causation refers to situations in which the outcome precedes and affects the exposure instead of the other way around. So we've heard many times that Parkinson's disease is characterized in some patients by a long prodromal phase, characterized by non-motor symptoms such as constipation, REM sleep behavior disorder, dysautonomia, weight loss, some subtle motor signs, and these prodromal symptoms can be present as long as 20 years before diagnosis. So the risk is that these non-motor and subtle motor signs will affect, may influence the exposures we're interested in. Dementia is also preceded by cognitive and motor decline and other symptoms, as we already saw. So for example, this slide shows the frequency of 3 prodromal symptoms of Parkinson's disease at the time of diagnosis and before diagnosis. So here, up to 10 years before, it was a study done in England. So for example, you see that balance problems appear to be more frequent in cases than in controls about 5 to 6 years before the diagnosis, whereas constipation is more frequent in cases and controls even 10 years before, although the difference increases as we get closer to the diagnosis. And these non-motor symptoms can have consequences. So for example, if we're interested in trauma or physical activity, because balance can influence the risk, for example, that someone becomes less active, then we need to take into account this prodromal phase. It's the same for constipation. For example, constipation may affect the diet of the person. They can change diet due to constipation, and so this is also important to take into account when we examine these risk factors. And all of these features can also increase the number of medical contacts. So PD cases may be more likely to see doctors before they're diagnosed and controls. And this is something we addressed in this study, in which we looked at the relation between statins use and the risk of Parkinson's disease in E3N, a cohort of 100,000 women followed since 1990. And we saw that this is the time of diagnosis as it is. This is a retrospective time scale, so going back to the beginning of the follow-up. We see that the cases saw doctors as often as controls until about 5 to 6 years before the diagnosis, where you see there's a difference in the number of medical contacts that PD cases have each year with doctors. And at the same time, we looked at the prescription of statins. So you see that cases were using less often statins than controls until when they started actually seeing more often doctors, where probably they were prescribed more often statins for cardiovascular prevention. And then you see that the rate of prescriptions of statins is similar in cases and controls. So if you want to study the relation between statins and Parkinson's disease, you need to take into account this phase, because otherwise you will get, I think, an unbiased estimate. So we did this by using what we call lagged analysis, in which you include a lag between the time when you start following the patients for Parkinson's disease and the time when you start assessing exposure to statins. We included here a lag of 5 years because this was what we saw. And so what it means is that at each time you look at the risk of Parkinson's disease as a function of statin exposure 5 years earlier. And what we saw is that by doing so, statin users had a 30% decreased risk of developing Parkinson's disease. But had we not included the lag in the analysis, you see that there was no association. So you see that this can have an important impact in the results of the studies. This is another example looking at body mass index. You know that Parkinson's disease lose weight, and this weight loss can start before the diagnosis is made. This is what you see here on the bottom curve, where you see that the frequency of obesity starts to decrease in PD cases compared to controls about 10 years before diagnosis. But you also see that here in our study in E3N, the frequency of obesity was lower in cases than in controls throughout the follow-up. So when we looked at the risk of Parkinson's disease as a function of obesity, you see that if we included a 5-year lag, we saw an inverse association that also was present with a 20-year lag, meaning that if you assess obesity and then you look at the risk of Parkinson's disease 20 years later, you do see an inverse association. This is very different from what we see in dementia. So again, people with dementia patients tend to lose weight, and this can start years before the diagnosis. So if you don't include a lag in the analysis, you see that you see an inverse association. People with patients have already lost weight. But if you include an increasing lag in the analysis, you see that in fact higher weight is associated positively with the risk of dementia. And so this means that midlife obesity, overweight is associated with high risk of dementia, whereas this is not the truth if you assess BMI and dementia at the same time. So because we were puzzled by the findings for Parkinson's disease, this inverse association of obesity with the risk of Parkinson's disease, we performed a Mendelian randomization study. This has been briefly mentioned before. So Mendelian randomization is a form of instrumental variable analysis in which you use genes that are strongly associated with an exposure. And this allows to estimate the unbiased association between the exposure and the outcome, not biased by confounding or reverse causation under some strong assumptions that we can discuss later. So we used 500 genetic polymorphisms associated with body mass index, and we confirmed the same thing that we saw in the observational analysis, that higher BMI was inversely associated with the risk of Parkinson's disease. We used the same approach to study the relation between milk consumption and Parkinson's disease. Again, we were concerned, as I was mentioning earlier, that constipation may have an impact on the diet the persons have. And so this could, because of the reverse causation, this could bias the association between diet and Parkinson's disease. So we confirmed what others have already seen in other studies, that higher milk consumption is associated with an increased risk of Parkinson's disease. But what was more original in the study is that this was true even if we assessed milk consumption milk consumption 10 or 20 years before we started to follow the patients for Parkinson's disease. And then we performed the Mendelian randomization study where we use a polymorphism in the lactase gene that is strongly associated with milk consumption, and we confirmed that genetically predicted milk intake was associated with a higher risk of Parkinson's disease. This is another example with physical activity. I won't go into detail because it's a poster that's presented by Fanny Artaud in the exhibition. This is probably a little bit less well known, I think, is that reverse causation is also an issue for environmental exposure. So there's an increasing interest in environmental exposures as risk factors for brain health. And it's possible that prodromal symptoms such as cognitive decline or physical impairment may lead persons to move more frequently before the diagnosis. And so this could have an effect on the exposures that they are exposed to. And so this is something that this small study in the US, it's a hypothesis that they tested. They were interested in a small number of participants in the relation between episodic memory and vegetation index. And what they saw in their analysis is, in their traditional analysis, is if you had a higher memory, you were less likely to live in a green space. And then they tested this hypothesis using a gene that associated with lower memory, so the ApoE epsilon 4 allele, and they performed the Mendelian randomization analysis, so that which is less likely to be biased by reverse causation, and then confirmed the inverse association between lower memory and living in a green space. So there are some solutions to this issue of reverse causation. We've already heard that longitudinal studies with a long follow-up in which you can assess midlife exposures are very important. You can do lagged analysis, as I tried to illustrate. You can, I think what's also very useful is to have repeated measures of exposures, because in this way you can look at trajectories before diagnosis. You can also adjust for subclinical disease if you have information on prodromal symptoms. You can also use the outcome to predict a future change in the exposure, as we just saw in this study on the relation between memory and green space. And you can use Mendelian randomization or other type of instrumental variable. I think another approach that is interesting is the analysis of gene-environment interactions, because this is another way of using genes to improve causal inference. If you can, because they allow to give some input about the potential mechanisms involved in the relation between an exposure and the outcome. So here are a few examples. Now, survival bias is a form of selection bias. It's due to the fact that the incidence of neurodegenerative disorders increases markedly with age, and older persons at risk of these diseases have survived to other frequent causes of death, so mostly cancer and cardiovascular disease that start to occur earlier in life. And so this can happen if the exposure that we're interested in is associated with survival until inclusion into the study. The survival bias can go in any direction. It can lead to a lower or higher estimate, and it can also affect Mendelian randomization studies performed in older persons. And so this is just an example looking at the relation between fat mass and mortality, where you see the expected association between higher fat mass and mortality in persons who are younger than 70 years. But when you look at younger persons, then you don't see any association at all. And also this means that persons who are older than 70 years have survived until 70, and also these are a selected group of persons. There are some solutions, so you can examine whether correlations between the exposure and other variables change with age, because this is what you expect to happen if you have survival bias. You can also examine whether the association between the exposure and the outcome change with age. So this is some analysis that we did in our Mendelian randomization analysis, so in which we stratified the analysis by the median age. And what you see here is that the inverse associations that we found, so for BMI, also for smoking, which is inversely associated with Parkinson's disease, were stronger in younger than older participants, which means that survival bias may be attenuating a little bit the associations in older participants. It could be also other causes, for example, differences in causes of death of disease by age. There are more sophisticated approaches to handle this, such as inverse probability weighting or simulations. Now, I think diagnostic misclassifications is also an important issue in our studies, because increasingly we're using health claim databases to study neurodegenerative disorders. And in these databases, diagnoses are generally ascertained based on diagnostic codes or drug tracers whose validity is unknown. And this is an example of a validation study done at the Mayo Clinic, where they compared diagnosis of PD based on review of medical records and diagnosis of PD based on two codes in their databases of PD. Although this approach to identify PD had a high sensitivity, well, relatively good sensitivity, you see that it had a very low predictive value, only 46%, and that PD ascertained using ICD codes overestimated the number of diagnoses by 73%. So this is very large. It's probably due to the fact that by using these codes, you include other causes of Parkinsonism. So there's a high risk of misclassification of disease status. And this can have an impact on what we found. So this is, for example, a study that looked at the associations between cerebrovascular risk factors and Parkinson's disease among Medicare beneficiaries. You see that they identified a large number of PD cases using a very similar algorithm as the one used at the Mayo Clinic, and they found that the relative risk of PD in smokers was 1.2, so very contrary to expected inverse association, and also a relative risk for hypertension of 1.4. This is opposite to what you see in studies such as the Nurse Health study, in which there's a careful validation of diagnosis of PD, in which you see the expected inverse association with smoking and no association with hypertension. So I think that what's happening here probably is that there was an inclusion of cases with vascular Parkinsonism, which is probably quite frequent, and misclassified as Parkinson's disease. I think there's also a risk of error on age at onset and diagnosis when you use these health claim databases, especially if diagnoses are ascertained using hospitalization records. And this is because often persons who develop, for example, Parkinson's disease, they're not hospitalized early on the course of the disease, but at a later stage when they start to get complications. So, for example, this is a study that found that only 40% of Parkinson's disease were hospitalized over 5 years. And so this means that if you use the age at hospitalization to determine the age at onset or diagnosis, then you're going to overestimate, and there's a risk of exacerbation, the risk of reverse causation, because you're going to measure exposures after diagnosis, in fact. So how to reduce this problem? I think that validation studies are essential when you're using these algorithms in databases. This is something, for example, a study that we did in France, where we validated diagnosis by a neurologist who examined the patients in persons who were using anti-Parkinsonian drugs, and we found 93% sensitivity, 86% specificity. So not perfect, but the interest of these estimates is that then you can do quantitative bias analysis. I think this is a very interesting and underused approach that allows to estimate the impact of misclassification bias and also confounding on association estimates. So for example, if we go back to the study that I presented earlier with a relative risk of smoking of 1.2, then you can vary a little bit the parameters. But for example, if you assume that 10% of the cases in fact had vascular Parkinsonism and the relative risk of Parkinsonism due to smoking is 2, then in fact you can compute a corrected relative risk, and it becomes 0.7, so the expected relative risk of Parkinson's disease. Now, confounding is also another issue. You're all more aware with this problem. And again, I think it's an issue in health claim databases that are increasingly being used, for example, in pharmacoepidemiologic studies. They have a large sample size and they're allowed to detect small effects with very precise confidence intervals, but many major confounders such as smoking, physical activity, weight, head trauma, etc., are not included, are most often not available in these databases. So there's an important risk of confounding. And again, I think that quantitative bias analysis is a very interesting tool in this context. So to conclude, there's an increasing number of data sources that are available together with major developments in computational tools and methods. And these allow to use a variety of approaches in order to identify modifiable risk factors that play a causal role. So we of course, we can do traditional observational studies. We can use methods such as Mendelian randomization, other forms of instrumental variable analysis. Alberto just showed this example of discontinuity regression, which is related to this. We can do quasi-experimental studies. We can emulate target trials in these databases that have a large sample size. And I think that this is what these authors have called triangulation of evidence. I think it's a very nice way to frame the research we do. And this is using different methods, each one with its own type of biases and limitations and strength, to see if we get comparable results. And if we get comparable results, then we have more confidence that what we found is probably in the right direction. But if they don't, then also the limitations of each of these approaches can help us understand maybe what's going on. Thank you very much.
Thank you, Alexis. And I invite the two speakers to come to the table for questions. So if you have any questions, we have time for a few questions to the two speakers. Maybe I can start for you, Anne. Thank you very much for your talk. I was wondering what is the prevalence of co-pathology with the typical tau CT pathology you have, particularly in older people? And first, and second, which is related, does the co-pathology location correlates with the tau pathology?
Yeah, thank you. Thank you for the question about co-pathology with CTE. So we certainly see many cases of what we call pure CTE. So there's only a tauopathy neurodegeneration. I can't remember the exact number, but let's say it's about 50%. And then there are co-pathologies like Alzheimer's disease, very commonly with aging. The older, the more likely. The TDP43 deposition does occur in the same spots as the tau deposition. So it's unlike any other TDP43 related disease. You see it around blood vessels and you see it at the crevices of the brain, suggesting a shared mechanism. The alpha-synuclein it looks just like garden variety Parkinson's disease or Lewy body disease, nothing that we can see that's unusual about that.
Other questions, yes, in the audience? Please present yourself and ask your question.
Hi, Alana Katzand from New York. Thank you so much. Those were both really excellent talks. My question is for Anne. So first of all, thank you so much, not only for the presentation, but for the work that you do. I have to say, from a personal standpoint, my husband played competitive ice hockey growing up and through college. And when our kids were little, talked about letting them do that. But by that time, your work had started to come out, and I put a hard no on things like football and ice hockey. And so my question is, what do you think about the ethics of parents signing their kids up for these sports, given the work that you and others have published? And what do you think can or should be done in terms of improving education? For example, should parents it's not so much of a problem for us in New York City where there's lots of other things, but for areas like, Texas and then US, places where football is still king, should parents have to watch a video like what you showed us or something else before they sign their kids up to play sports like football?
Pretty loaded questions. Thank you. The ethics of letting kids play these sports, the consents are signed by the parents. There's just inadequate education out there. In highly educated communities, the parents are more aware of the science and the evidence, but there's lots of places in the U.S., the more rural spots, and I don't know about what the rest of the world, where individuals just have no idea that this could be the possible consequence. We certainly have been, I've turned into an advocate. I never intended this to be part of my career path, but you just become an advocate, and we align ourselves with something called the Concussion Legacy Foundation, which is an advocacy and education program, and they provide education all over the U.S., but these sports define our culture. These sports are like a religion all over the world. They're fanatical fans. Nobody wants to hear what I have to say. So they would like me to go away. It's not just me, obviously, but, this is very unwelcome information. And, and that's a difficulty that I've spent 17 years working on this disease, and it's been very hard to overcome. It's an obstacle. No one wants to hear it, and so there's just tremendous denial that comes in many forms. This can't this only happens to professional athletes that played high levels of sports, or this or that or the other thing, but it's just not true. It's a risk for our children, and if this risk, think about what we've done for car safety belts or children's cribs with the width of the slats. If this were happening in something else, we would already have done something as a country, as a world, about this public health issue. But because this is sports and it involves all of us in sort of an emotional way, we haven't. I don't know the answer to the next one. What was it? What can we do to change this? We're trying, but it's been a long uphill battle.
Another questions?
My question is actually connected to the previous one. Do you think there is enough scientific investment in the scientific divulgation about pathologies like CTE and the high risks that they may cause, like depression, that is very high social price, let's say?
Your question better.
Do you think there is enough investment in the scientific divulgation about pathologies like depression that may be caused by CTE? That's a very big social problem.
Yeah, no, of course I don't. You know, depression has many, many causes, and I'm certainly not saying that CTE is the major cause, but I do think we tend to give it a very, we tend to think that it's all mental health, that it's all, you know, something that we can't solve. And I don't think we look enough for other causes of these depression or feelings of loneliness or, you know, isolation or erratic behaviors or impulsivity. We don't take these complaints seriously as a medical profession. We don't look for possible causes. And so these people come to us for help and they're often dismissed. Especially, I've noticed, you know, because of my work, athletes, young athletes, if they seek help, they're dismissed. You know, and I think as a profession we have to have more compassion, more understanding that some of these things can actually have a physical or pathologic cause, and that all people deserve to be comprehensively managed and followed to make sure that these destructive pathologies aren't at the root of their difficulties.
Another question?
Okay. I'm Letizia. Sono Francesca Maschietto, volevo sapere se c'è un qualche esame per l'encefalopatia cronica di origine traumatica e se ci sono delle cure.
Sure, but I haven't heard.
Are there treatments for CTE? Do you know, are you aware of some treatments that can be developed for CTE?
Is there any exam that may allow to pick up this traumatic brain, chronic traumatic brain injury? This was the question of the lady. Exactly. There is something that may help to pick up early. And if I can add, because I have the microphone, which is really strictly linked to this question, for those who have already some very high risk factor, is it acceptable that the young student is accepted to start some sport activity that has a very high risk? For example, if a student is already affected by multiple sclerosis, I will really be very, very careful in accepting to start this type of sport activities.
Yeah. Well, yes, comorbidities. We don't totally understand the effect of medical comorbidities on the trajectory of CTE and these other post-traumatic neurodegenerations, but they are most likely contributory. We know diabetes and insulin resistance, and it's likely that other immune disorders contribute to this disease. There's strong evidence that T cells are involved, CD8 cells, microglial inflammation. So mostly they converge on the same pathways. As far as detection during life, we don't have a clear ability to detect this during life, and that's why there's massive research for blood and fluid biomarkers and PET scans for tau. But the symptoms of depression and behavioral change and impulsivity and attention disorder, those things, those things need to be managed even in the absence of a specific diagnosis. Those individuals need to be cared for and treated symptomatically, no matter what the cause, and followed over time, because time will tell whether they're developing a neurodegenerative disease. The symptoms of early CTE are very similar to post-concussion and impossible to tell the difference. The only way to know if they're going down this neurodegenerative path is to follow them. But I think all
these athletes and other individuals, military veterans especially, deserve this compassionate management of these very important symptoms.
You cited biomarkers, and you know, we have a lot of biomarkers so far, and we develop NFL, p-tau, and many others, S100. So there are so many, you know, way of monitoring the impact of traumatic encephalopathy. So do you have any study monitoring during life all these people that develop CTE?
Yeah, there's all sorts of studies. We have a study called Bank CTE where people actually they qualify over the phone, and then they donate their blood at a Quest laboratory. We started that study. Definitely studies in boxers all over the world and rugby players showing NFL light increases, GFAP increases, but the results are always on a group level. There's no individual change that we can say is adequate for detection. I think we'll get there. I think we're following very rapidly on the heels of what we've done in Alzheimer's disease with clinical detection. The tau PET markers, very, very exciting. The tau PET markers were developed in Alzheimer's disease, so that's a ligand for Alzheimer tau, but now there's many groups in the US and all over the world that are looking for ligands for this CTE tau. So I just think it's a question of time. I think we're very close, and I think it's an exciting time in the development of this understanding.
Do we have time for one or two questions? Yeah.
Okay, I'm a high school student in Germany, and you already said that it's only detectable after death, and I think you partly answered my question because you said you're doing a lot of research in order to be able to detect CTE while the person's still alive. And how do you do it? What's your methods in order to find out how you can detect it while the person's still alive?
So we're following many patients clinically, doing longitudinal neurologic neuropsych evaluations, taking blood, CSF, and doing PET scans. And we're hoping that as these studies progress, we develop these sensitive markers for disease. Now, what I showed you in the young athletes, we did have evidence that there are macroscopic changes in the brain. So while it's not a specific marker for CTE, cavum, septum, pellucidum is often found in the presence of CTE. So it's a suggestive marker. If you see any evidence of ventricular enlargement or fornix degeneration, those would be suggestive markers of CTE.
Was about follow your comment. Is MRI sensitive? I mean, what you show was astonishing from a neuropathological point of view. So you should see something at gross at MRI.
Yes.
So MRI should be maybe the first test that you can do, or
yeah. So we could do MRI. You can see frontal temporal atrophy. You can see thalamic atrophy, third ventricle enlargement. You have to do probably several, right, to see the change. But you can pick up suggestive markers of a deterioration. They're just not specific.
A last question, maybe, Caroline?
Questions for Professor Obaz, and thank you both for wonderful talks, by the way. So the first question is, we found very similar findings in some U.S. population-based, EMR-based populations of misclassification of diagnosis. But one of the things we also found was that there were apparent disparities in the sense that people who had residents or had, fewer, were not diagnosed by a specialist, by an expert, or were much more likely to be misclassified. So this suggestion of disparity and perhaps even underdiagnosis in certain populations. So I just wonder if that's something you've also encountered and how you might think about how we might incorporate that in terms of improving our analyses when we look across broad populations. And then the second question is, we've started, when possible, to start trying to do some verification of diagnosis using video telemedicine, where you actually, especially in Parkinson's, which is pretty visual, you can do a remote exam and come up with a pretty good sense of diagnosis. So I wonder if that's something you've thought about as a way to help to reduce that problem.
So thank you very much. These are very good questions. So for the first one, I completely agree that it's very likely like there are disparities, probably due to things like access to healthcare, for example, in rural regions or in urban regions. It's probably very different how easy it's to see a neurologist or a movement disorder specialist. And so this is also related to socioeconomic characteristics. And I think it's even more difficult for Alzheimer's or dementia in general than for Parkinson's disease, for example, because the likelihood that someone is going to see a doctor, in particular at old age, for a memory problem, is going to be influenced by many things around this person, the support he or she has, etc. So I think that the underdiagnosis of these diseases is likely to be influenced by some socioeconomic variables, and I think this is probably a complicated question to address. Regarding the second point, no, I have no experience in this, but I think it's a wonderful idea to do at least screening, for example, when you have large studies, and I think it's a very good, very interesting idea. Thank you.
Okay, thank you very much to the speakers and for the discussion. We will end this session and leave the room to the next lecture. Thank you very much.
Okay, so welcome to keynote speaker number 2. We're going to discuss climate change and the impact on neurodegenerative diseases. So I'm Roberto Buizza from Scuola Superiore Sant'Anna. I am a physicist and mathematician working on Earth system modeling and climate change. And it's great to be here. So I want just to show you 2 or 3 slides to introduce the speaker. So climate change is a global priority. We are responsible for what's happening. There's no doubt about it. And if we continue to emit as we're doing, we will get to even higher global average temperatures, which means more frequent and more intense extremes. What you see here is a graph from the European Environmental Agency that shows the temperature increase in the Mediterranean region. So given that we are in Italy, I thought to show you this instead of global averages. So the Mediterranean region has been warming about 2.5 times the warming of the globe, because it's a climate hotspot. And if we continue as we are doing with the emissions that all the countries are projected to emit in the next 20, 30 years, we are on the orange curve or on the brown curve. So we are going to hit 4 or 5 degrees warming by the end of the century. And reports by the International Panel on Climate Change or Intergovernmental Panel on Climate Change or the European Environmental Agency clearly show and say that the predicted impact on health is going to be critical to catastrophic, depending on how much we are going to emit. And this is due to heat stress and to pathogens moving following climate change and impact on health system and infrastructure. And I guess the speaker will talk about this, okay? There is a co-benefit in addressing climate change. So we should not delay it by saying we can wait until we have new technologies that are working, like generation 4, generation 5 nuclear power station. We need to reduce emissions now. And if we reduce emissions now, we can also reduce pollution. And this is very important for countries like Italy, for example, which has some of the most polluted cities in the world, as you can see from this report by the European Environmental Agency. So by addressing climate change, we can also, by electrifying transport, for example, we can reduce emission, we can improve health. So this, but unfortunately, we're not doing it. So this is in black, how the global emissions have been going up from 1990 to now. We keep increasing emissions. The last black point is 2022. The dip that you see was COVID. And you see, if we want to really achieve net zero by 2050, we need to reduce emission by at least 5%, 7 or even more percent. But if you look at the last 10 years, emissions, global emissions have been increasing 0.5%. So you see there is a huge change that we need to implement. Okay, so this is just what I wanted to say. I want now to introduce the speaker, who is Andrew Dowan from Cleveland Clinic, USA, who is going to talk about effects of climate change on neurodegenerative diseases. So Andrew is a mathematician and a computer scientist whose research focuses on using translational data science, computational biology, and mathematical biology to accurately diagnose, treat, and prognosticate neurological diseases. He has a very strong interest in understanding the effects of climate change on neurological health, and he was one of the people who wrote one of the first reviews on the neurological effects on climate change. So I'll leave the floor to Andrew Dowan. Thank you.
Thank you so much for the kind introduction. That was a perfect introduction to this talk, and thank you to the Prada Foundation, Professor Comi, for inviting me. It's an absolute honor and a privilege to speak to all of you. This is a pixelated image of our home. It's the only place in the universe that hosts life, and as I was a child, this was what fascinated me, is why are we the only place with life, and how can we support it? So I have no disclosures. Today I'll speak about kind of a little bit about what you already heard, which is why climate change is a major issue, why it's the biggest issue of our time and the biggest issue of all of our lifetime, the mechanisms by which climate affects human health, and specifically with the axis of neurodegenerative disease. I'll show you some references, conclude, and then I'm happy to take any questions. So first, let's understand the scale of the problem, and this was set very perfectly by the kind introduction, but this is how we measure climate. And the first image here is of the Mauna Loa Observatory, where CO2 levels are monitored on a monthly basis by a very dedicated team of scientists. And the second two images are from ice cores, and this is how we actually obtain carbon dioxide measurements from thousands of years ago. Carbon dioxide is trapped in these bubbles in the ice, and you are able to basically, once you have this core, measure these concentrations very precisely. And from this, we know that indeed it is humans who are causing change in greenhouse gas emissions, which are leading to changes in climate. And this is how we know the temperature is changing. So reliable temperature measurements started in the 1800s, and taking the average from 1971 to 2000 as our reference, we can see that before the 70s, things were, relatively on the lower scale, but they have significantly picked up since then. And we are on a trajectory that is on a collision course for major issues. So where are we going? Where is this trajectory? So these are, like we just saw, the graph of global greenhouse gas emissions by the Climate Action Tracker, which is a non-profit. These show us our projections out to the year 2100. And the green line is where we need to go. Okay, that's our 1.5 degree compatible emissions target. You need to hit net zero close to 2050, but like the previous speaker mentioned, we are not even close. There's a massive implementation gap. Even with the most optimistic scenario, we're ending up around 1.8 degrees Celsius. And with the current pledges and targets, even if we were to meet all of those, we would hit around 2.1 degrees Celsius by the end of the century. The 2030 targets don't go far enough, and we will hit around 2.5 degrees Celsius. And really our current trajectory, because emissions really do keep increasing, is headed in not a great territory, between 2.5 to 3 degrees Celsius, at which point there is incredible damage predicted to many species of life, including our own, and I hope that motivates all of us. So why does this matter? Climate health and human health are inextricably linked, okay? The effects of rising temperatures on our ecosystems results in severe weather, which itself causes injuries, fatalities, mental health challenges. Extreme health causes health-related illness and death. Cardiovascular failure has been shown. Increasing carbon dioxide levels are also linked to environmental degradation, which has itself been linked to forced migration, conflict, again leading to mental health impacts. Water and food supply impacts lead to malnutrition, diarrheal disease, and rising sea levels affect our water quality, leading to many infectious waterborne illnesses. Increasing allergens lead to increased respiratory allergies and asthma. And more extreme weather, of course, as we've already heard, leads to changes in vector ecology, infectious diseases, many of which are neurotropic, as well as effects of air pollution, which I will touch on. These are real risks, and these are risks that actually we've been able to measure over the last 20 years. So this is from a 2022 report by The Lancet. And first of all, I can show you here that they have reported that food insecurity is really increasing. We may not feel this. We are the privileged few who don't experience this, right? But the percent of people in the world who experience food insecurity is rapidly increasing, even at a scale we can measure over the last 20 years. Moreover, the suitability for infectious diseases, so the hosts, the mosquitoes that carry these viruses, the climate conditions are more favorable for their reproduction, leading to an increased R0, or reproduction rate, of viruses like dengue, West Nile virus, etc. And the duration of extreme heat waves, as I'm sure all of us have noticed, has increased since we've been able to more reliably measure it. The effects on health are absolutely inextricable. So my team and I, seeing all of this, we asked the simple question of, well, what are the neurological consequences? And we undertook as many pieces of literature as we could get our hands on, included over 3,000 pieces of literature. We dutifully reviewed each of these, and we ended up with a core set of around 364 studies, which we grouped into three major themes. Those that really studied climate, meaning temperature, weather patterns, and its effects on neurologic health, infectious diseases and its effects on neurologic health, and the third category was the most prevalent, which was pollutant impacts. And we included this because the burning of fossil fuels generates pollutants, which of course is quite linked to the climate. And so the first study one of the major studies that we reviewed in this paper came from Medicare data, and it examined 3 million patients in New England over 11 years and really highlighted how climate influences survival in patients with neurodegenerative diseases. It may not necessarily affect the incidence, it does, but we'll talk about that. But even in those who have pre-existing disease, it's quite certain to measure that they have limited survival as a result of climate factors. So over the time period that they measured, 81,000 individuals were admitted to hospital with a discharge diagnosis of dementia. This is just a map of the region they covered. And what they found was that each 1.5 degrees Celsius increase in summer mean temperature was associated with a 12% increase in dementia-related hospital admission. Of course, this study suffers from many of the caveats that we heard about in terms of classification, reverse causation, but I think at this moment, it's probably some of the strongest evidence we've got about how climate influences survival. There's also a study that has come from China in which over 132,000 dementia-related deaths were examined over a 7-year period. And interestingly, this study looked at a much more granular set of data and was able to show that nighttime and daytime heat both associated with increased risk of dementia deaths. However, they differentially associated with the risk of death of dementia, and the effects of hot evenings persisted for a shorter period than the effects of hot days. And this raises the question as to why this may happen. It's unclear. The other part of this study that was quite important is that it really showed this interaction between temperature and pollutant levels. So pollutant levels, as they're measured, can be affected by the relative humidity and the temperature. And so the effect on health also has to take into account these variables. And so however way you look at it, though, in this study, they were able to at least retrospectively show that climate really does influence survival in patients with dementia. Climate change is also affecting our nutrition. Of course, we've heard all about how nutrition affects our brain health. And so really greenhouse gas emissions lead to changes in climate, which lead to changes in what we can grow, lead to changes in what animals can eat that we then consume, and also lead to food insecurity, as I've shown. And quite frankly, these are unmeasured effects that we truly don't know the nature of how all of this is going to affect the ultimate nutritional status of human beings, but we can see these effects in other organisms. So you can see it as, for instance, pests and pathogens increase, that other organisms that rely on those plants and animals for their food supply have altered nutrition. And I think it's not far until humans are affected. No study of climate change is complete without a discussion on airborne pollutants. So when we talk about pollutants, this is largely what we discuss. We are discussing things smaller than the width of a human hair, much smaller, in fact. So we often talk about PM 2.5 or particulate matter 2.5, which is things that are less than 2.5 microns in diameter. These are the products of combustion of fossil fuels. These also interestingly include metals. And for the Parkinson disease researchers in the room, you probably have read some studies on inhaled manganese as it causes Parkinson's. We also talk about PM10, which are larger particles. These include dust, pollen, and molds. And these are incredibly tiny particles that, as we inhale, we believe go into the human, actually into our blood supply, and thereby cause an inflammatory response. People have studied the inflammatory response and shown that it affects neurovascular health. And so that is a major axis of how this may cause neurodegenerative disease. Our review, we put together everything we could find about each of these pollutants and looked at the associations. And what we found was striking. And so first of all, there are many classifications of such pollutants that have been studied. People have looked at PM in different ways, PM 2.5, PM 10, and PM coarse, which is a less precise method of measuring the diameter. We have looked at sulfur dioxide, nitrogen dioxide, and ozone as well. And so what you can see here is that really all of these pollutants associated with a huge variety of neurologic diseases. More specifically, PM 2.5 associated with white matter disease, amyloid PET positivity, and poor neurocognitive outcomes. Coarse PM 2.5, sulfur dioxide associated with cognitive decline, and we'll get a little bit into more of the evidence of that. Stroke mortality, MS relapse risk, stroke risk, TIA risk, dementia risk, migraine headaches, Parkinson disease risk, brain atrophy and vascular dementia risk. All of these have been examined through the lens of airborne pollutants. Now, none of these studies is perfect. They're epidemiological studies that are retrospective with many of the complications that we've already discussed. But I think the fact that you've got so many lines of evidence converging on the same sort of set of diseases tells us something that we simply can't close our eyes to. And so to highlight this effect, I'll show you a couple studies. This is, again, from the Nurses' Health Study, which is a very, very strong, robust cohort of 19,000 women. And in this particular study, they looked at those aged 70 to 81 years old. What they showed was that in those who were exposed to greater amounts of air pollution, the nurses actually had cognitive aging. So by cognitive scores, their level of exposure could be measured. And those who were exposed to 10 microgram per meter cubed higher exposure of PM 2.5 to 10, which is a coarse measure of PM, particulate matter, that they actually had cognitive aging equivalent to 2 years. And many always ask me, well, what about those who moved? Because these are based on zip codes or census tracts or very, more granular regions of air quality measurements. And of course, these nurses weren't constantly outside and they have different levels of exposure, but one can assume it's relatively similar given the job description. And interestingly, they did a subset analysis in which they looked at those who did not change houses or who lived in the same place. And the associations actually were strengthened, again, supporting the fact that perhaps there is this causal relationship here. And actually, this is quite a nice study in which 19 individuals who were residents of Mexico City or Monterrey, which are high pollution areas in Mexico, versus smaller cities in Mexico, which presumably had lower air pollution, were studied post-mortem. No individual in this study had prior clinical evidence of neuroinflammation or dementia. What they showed was that, in fact, in those individuals who were otherwise matched to this cohort, there was higher level of A-beta 42 in the hippocampus and the frontal cortex, suggesting that perhaps there's an association between airborne pollutants and neuroinflammation and therefore AD-like pathology. And in those individuals who are perhaps primed to have neurodegeneration, it's possible that airborne pollutants hasten cognitive decline. So in a study of from Taiwan, and Taiwan is a place which is very interesting because it's a small island, of course, it has some of the best air pollution monitoring in the world. So what we found actually in our review is that one needs high quality data to do these studies. You need air quality data that goes to a resolution of 1 kilometer by 1 kilometer or even less. And you need good data about patients' health. And so you need a nationalized health system. Taiwan has both of these things. And so this study, in which 704 patients were interviewed each year for 16 years and examined for clinical dementia rating score, used annual mean pollutant concentrations across the island, and they adjusted their Cox models for very many factors. And what they showed here is that when you start to get granular at what pollutants actually associate with a cognitive decline, they're able to show is that there are differential associations between various airborne pollutants. Most are significantly associated, but in particular, sulfur dioxide, one that's relatively understudied by other studies, actually may have a much stronger effect. And sulfur dioxide is, of course, produced by heavy industry, and so this is something that could be rapidly targeted, of course, but we have to do that. So switching gears a little bit, I'll talk about how extreme weather limits healthcare delivery. It limits healthcare delivery in the sense that when there is extreme weather, we cannot deliver emergency health services or routine health services to individuals. Unfortunately, those who are most affected by extreme weather are those who are most vulnerable in our society. They're the poor, the people who can't move, who can't afford to move. And we have to think about that as we design healthcare systems that are robust to extreme weather, because I think we're not getting out of this problem. It's only going to increase, and we should think about, should we really have retirement communities in Florida where there's many hurricanes and floods and things like that? Should we think about how we expand teleneurology and dementia care, Parkinson care, seizure care in extreme weather scenarios? And so I think we have to think about how we're going to deliver care. And that was a major facet of what we actually found in our review that was missing. And really no governments, we thought, had really written much about this. And we think it's important that as a community, we think about this very carefully. Lastly, I'll touch on how climate change affects patterns of vector-borne neuroinfectious diseases. And of course, I think most of you are probably aware that mosquitoes and birds are the most common hosts, and they're kind of co-hosts and of very many neuroinfectious diseases, and tick-borne illnesses, of course, come into play. And these go from the vector to the human. And here's a map showing how rapidly, over just a few years, this is 2010 to 2013, so this is just 4 years of data, the counties reporting West Nile virus, which is, of course, a neurotropic infectious virus, whose effects long-term on neurodegeneration remain unknown. What you can see here is how quickly it has expanded across the United States. I think this is being seen in Northern Europe with tick-borne illnesses, as well as Western Europe. One interesting aspect that we found when we reviewed all of these studies related to neuroinfectious diseases was really the fact that climate not only affects the vectors, but it also affects humans. It affects what we do outside. And climate affects economy, which also affects that. So, for instance, after the fall of the Soviet Union, the agrarian cultures actually picked up in Eastern Europe, and there was a rise in tick-borne illnesses. But at the same time, the climate was changing. And so how does one disentangle those effects? And this becomes quite complicated from an epidemiologic perspective. But nevertheless, strong reporting and monitoring of neuroinfectious diseases has really shown that the reach has changed, and the populations affected are those who were not previously affected by these illnesses. So things like vaccination have to be considered more broadly than previous. And so I'll ask again, where are we going? And this is what keeps me up at night. This is a picture of India Gate in New Delhi. The world's top two most polluted capital cities are New Delhi, India, and Dhaka, Bangladesh. These are rapidly modernizing economies. These are the most vulnerable places in our world to climate change. They're close to the equator. Dhaka is a very low-lying city in terms of sea level. New Delhi has 20 million people under the age of 30. Dhaka has 15 million people under the age of 30. By 2050, these numbers will not only increase, but these people will have aged. They will have cognitively aged faster than we have in the West. And we have to think about that. And we have to think about what the impact of this will be. And this is what keeps me up at night. Is there hope? As we heard, there are co-benefits to greening our economies, greening our health systems. And so there are co-benefits to our brain health as well. So this is from The Lancet. A transition to renewable energy can help reduce energy poverty, so it can increase the availability of energy by bringing cost-efficient electricity. It can reduce exposure to indoor air pollution from fossil fuel burning stoves. A plant-rich, low-carbon diet actually can help prevent multiple millions of deaths annually across the world and, of course, improve our brain health. And there's hope. I mean, clean energy generation did reach highest levels actually in 2024. This should be updated. And improving air quality, as we have already shown, will prevent many, many early deaths and hopefully early cases of dementia. And urban design, when considering healthy and green urban design, we can promote physical activity, better brain health, better livable cities, more friendly, better mental health, and better for young people that really need to survive. And so, I leave you with this, that there's really no planet B or plan B. We have only one option, and it's to rapidly decarbonize if we are going to save our planet, save our brains, and really save our future. And some references. Thank you again to the Foundation Prada for making this talk possible, American Academy of Neurology, Cleveland Clinic Foundation. I'm happy to take any questions, and thank you for your attention.
Thank you very much. Will you join me here? Yeah. Okay, so do we have questions from the audience? I cannot see. No questions? You all want one question over there.
Congratulations for your interesting talk. It seems that it's not easy to distinguish between climate change and pollution. We have studied this problem recently, and we find sometimes difficult. Some difficult to distinguish between the contribute to increases of little degrees of temperature and pollution in some country. And I would like to ask what your opinion on this, and if you know some specific mechanism of the little increases of the global warming on molecular mechanism in neurodegeneration.
Yeah, thank you for the question. So really asking about mechanism and how temperature and small changes in temperature, I think, can really relate to neurodegeneration. And this is something that's incredibly challenging to study, right? These are small effects, and you're looking at clinically heterogeneous diseases. Nevertheless, I think the few studies that have actually looked at this have thought a lot about molecular mechanisms related to oxidative stress in the brain and metabolic stress as we struggle with thermal regulation. Now, that's of course at the extreme end and not at the chronic exposure end. So I'm not aware of any studies that really study those small increases, but I suspect from small animal models we may be able to get a better idea, but I haven't seen strong studies in that regard.
Okay, so let me add a point on this. So with climate change, we might get, for example, in situation where you have these very high pressure systems, especially during summer, that leads to very high temperature. But they don't only lead to very high temperature, because when you are under a high pressure system, which is large, I mean, talking about a structure order a few thousand kilometers, then this clearly leads to the temperature at the surface to increase, but also to, since there are no vertical motions, to an accumulation of pollutants. So the two, one reinforces, so the climate change reinforces the accumulation of pollutants in the area, that reinforces the exposure of people to both the effects. So people will experience not only higher temperatures during day and night, but also an increased level of pollutants. So this is how the two reinforce each other, and then you can get, you know, even more increases into the impact of climate change. So do we have any other questions? Oh, yes, there is a question over there.
Yes, thank you for this very nice talk. I have just a provocative question. So you explained that the consequence of climate change is an increase in disease and possibly conflict and wars. And the consequence of that is an increase in mortality. And so if you have fewer humans living on Earth, then you also have a lower level of pollution. So I am wondering whether you include in your projections this type of feedback, that actually is there kind of an equilibrium at the end where the consequence is also part, not only a problem, but also part of the solution.
Yeah, it's a very provocative question. And actually, you know, the global population will peak within our lifetime, hopefully some of our lifetimes. And I think that's one interesting aspect. I think the more important thing, though, is not only to think about humans in a way, because it's actually biodiversity loss. It's this runaway effect of climate change that we won't be able to stop. For instance, when you lose carbon sinks, right? When you lose natural ecosystems, the Earth's ability to resorb carbon, regardless of how many humans exist, can lead to runaway warming, which is, I think, what many of the climate modelers will tell you. And so I think limiting as much as possible is still a reasonable approach, even perhaps anticipating a decline in population. And I think no one would want to see an increase in conflict either way. So I think that's probably the way to think about it.
Yeah, if I may add, on when you see these projections of a future climate, like reported by the IPCC or what I showed from the European Environmental Agency, so these projections are generated using very complex system models that use assumptions on future emissions. These assumptions, again, a possible concentration of greenhouse gases are linked to economic growth, population growth, possible changes in the economies and so on. So in some way, it's taken into account the different scenarios that are used to do the projection, the brown versus orange versus cyan that I showed you, or that Andrew showed, in some way includes this. And second comment is, I mean, the increase in number of deaths compared to the increase in population. I think it's the secondary, if you want. It's a tiny number. Okay, I think I would probably talk more about the distribution of the people who will suffer more. If we can continue like this, there are going to be population especially in the tropical region that already have climate conditions in summer that is close to unbearable, but this will become even more. So you will have a lot of people die in these countries, and compensated by the growth of people in the extra. So this is so we better address climate change. In conclusion, okay. I think we have a question over there. Yeah.
From University College London. Thanks, Andrew. Very nice talk. And because as I know, the temperature and mortality, the relationship is U-shaped. So extreme cold is also a cause. So I want to listen to your comment about global warming. Probably it's kind of beneficial for if you got warmer weather in the winter. So it's really challenging to do the projection and so on, considering that. How do you comment that?
It's a very interesting point, actually, that there is this U-shape to the temperature mortality curves. The other actually interesting part of that data that comes out is that it's not only the extremes of temperature at which mortality from neurologic disease increases, but it's also daily temperature variability. Which implicates extreme weather and kind of these rapid fluctuations in temperature. And there've been like a few studies that showed that, for instance, emergency department admissions for strokes or intracerebral hemorrhage are actually greater on days that are in the extremes of temperature or in the extremes of temperature variability. So while the winter may be warmer, perhaps there are more variable temperatures within the day or more extreme weather events, all of which will negatively impact. So I think you can't look at one of these things in isolation. It has to be kind of considering all of these other aspects.
Okay, thank you very much. I think, thank you, Andrew. And we break for lunch. Okay.
We are going to start in a minute. We are already 3 minutes late, but in Italy, 5 minutes is not being late, so we'll wait for 1 minute. So let's start. Welcome to session number 7, session on brain plasticity. We have learned a lot of things about plasticity directly and indirectly across all the sessions, starting from sleep to rich environment and so on. And now we are going into the mechanism of plasticity and specific ways to enhance plasticity. That is the most amazing ability of our brain, the ability of changing. And that's why we are human, because we have this great ability. So the first lecture is by Leonard Muche, and I'm not reading the entire curriculum. He's very modest, and he asked me just to say that he's a brilliant I'm sorry. He's a brilliant neurologist and neuroscientist, and his work is focused on disease of neuroscience, in particularly pathophysiological mechanism of a cognitive decline in neurodegenerative diseases, and in particular in Alzheimer, and also epilepsy and autism. So we'll now listen to your talk. Thank you.
Well, thank you very much for your introduction. And of course, I didn't tell her to say that about me, but so this lecture is about mechanistic aspects of both adaptive and maladaptive brain plasticity. And so this is quite basic for this audience, but I thought it would be still good to make some of these basic comments about brain plasticity. Brain cells are able to rapidly change their structure and function, and this is really what we mean by plasticity. And they do this in response to environmental changes, but also internal changes. And this plasticity helps us adapt to an ever-changing world, and not just us, but all other creatures who have plasticity, which is across a very broad evolutionary spectrum. And brain cells and their interconnected networks also react to disease in very similar ways as they function normally, but they can respond in ways that are either adaptive and beneficial, but others can be maladaptive and in the long run actually contribute to disease progression, which raises several therapeutic implications, namely that enhancing adaptive and blocking maladaptive processes could be beneficial. It might improve our ability to overcome difficult challenges, like how to cope with climate change, and it may help us slow, halt, and prevent disease. So among neurons, plasticity can occur at all levels of brain plasticity and complexity, from molecules to individual synapses to neurons across their various subcellular specializations in microcircuits and across larger, complex distributed networks. And it doesn't stop there when you think about disease, where a lot is going on. This is a glimpse at an Alzheimer brain where you have disconnections between synaptic connections among neurons happening due to amyloid beta and possibly tau and alpha-synuclein accumulations. And the neurons and networks have to adjust to this kind of disconnection. But then there are also immune responses There are responses by astrocytes, and some of them are well-intentioned. But for example, we showed that adenosine A2A receptors are massively upregulated in the Alzheimer brain, and that process actually interferes with memory consolidation processes. And in a similar vein, microglial cells respond to blood products that come through a broken blood-brain barrier, and this turns them into neurotoxic mediators, pouring out all kinds of free radicals and other poisons onto brain cells. And then there are vascular adjustments that also occur. So the neurovascular junction is another very intriguing component of plasticity in the nervous system. So all of these things are happening, and I think anybody who thinks that there's going to be one blockbuster drug that will take care of this big scenario probably will be surprised. We will need many things to deal with that and to suppress certain detrimental aspects and enhance beneficial ones. So the concept that there could be a problem with plasticity in Alzheimer's disease is quite old, and Marcel Mesulam, really amazingly insightful neuropathologist and clinician, sort of put out this excessive plasticity burden as a concept that may occur in the context of Alzheimer's disease. And I think this is a really interesting concept, which has been tested individually with respect to some of these factors, but maybe not in terms of this sort of exhaustion phenomenon that he postulated, and is not easy to test, but intriguing. Jorge Pelop in my lab and I, we reviewed this field based on a lot of work we've done, and intriguingly, Alzheimer's disease sort of combines different aspects of synaptic pathophysiology. There's clear evidence for synaptic depression, but this is combined with aberrant excitatory network activity to which the hippocampus and other brain regions respond, with very vigorous remodeling phenomena that are probably initially adaptive, but then may run out of steam and ultimately may even become maladaptive. And I just want to illustrate this. So when we looked at non-transgenic mice and at APP transgenic mice that express Alzheimer's disease mutant forms of the human amyloid protein precursor, we showed that certain neuronal activity markers like the immediate early gene ARC in the majority of these mice are depleted, suggesting that there is lower than normal neuronal activity in the hippocampus. But in about 9% of these mice, we saw this massive increase in ARC induction, which almost certainly reflects epileptic activity in these mice. But they don't show frank convulsive seizures. This is non-convulsive sort of subclinical epileptiform activity. So the interpretation of the real data up here in terms of hypothetical implications is that a normal non-transgenic mouse and probably a healthy human brain cruises along in this window of normal neuronal activity, and what happens in the presence of these amyloid beta peptides and mutant forms of the amyloid protein precursor is that there are these peaks and valleys of excessive or suppressed neuronal activity, and that basically deranges the homeostasis of neural network functions. And we showed that this results in really intriguing remodeling in the hippocampus, where, for example, the interneurons in the hilus, they develop these excessive sprouts of GABAergic fibers into the molecular layer, and then the granule cells in the dentate start making neuropeptide Y, which then has aberrant collaterals onto basket cells. So there is a rewiring of the hippocampus, and that is probably in response to this excessive epileptiform activity, which you can see here across multiple channels in an APP mouse on a tau wild-type background. So here in this diagram, the APP and A-beta is the pathogenic trigger, which leads to this aberrant neuronal activity, but it somehow that is linked to the presence of the tau protein, where soluble wild-type tau is needed for APP to induce this activity, because when we reduce tau just in half in this model or entirely remove tau from the mouse, APP was no longer able to induce this aberrant excitatory activity. Now, are these network changes in mice relevant to the human condition? And so Keith Vossel, when he was in my group, pursued this question with our clinical colleagues at UCSF in the Epilepsy Center and the Memory and Aging Center, and we asked the question, how prevalent is actually this subclinical epileptiform activity? So we focused on people with early AD and no known seizure history and age-matched controls, and we brought them into the hospital for an overnight video EEG or long-term EEG monitoring, and the next morning they got 1 hour magnetoencephalogram combined with EEG and some clinical investigations. And what we found is that this subtle form of epileptiform activity could be detected in over 40% of sporadic AD patients with fairly early onset of cognitive symptoms, and it looks something like this sort of central spike you can see here in the EEG, or this spike that cuts across multiple channels in the magnetoencephalography recording that had no equivalent on the surface EEG, and most of these events sort of cluster in the frontotemporal region, as you can see here. Another very interesting study by Alice Lamb and Andy Cole at Mass General Hospital implanted foramen ovale electrodes into the intracranial space, just juxtaposed to the medial temporal lobes. And what they showed is that in 2 patients with Alzheimer's disease, the foramen ovale electrodes would reveal very high numbers of epileptiform spikes, even when the surface EEG was entirely silent. And this was particularly evident during sleep, very much in line with what we had found. Importantly, we showed that longitudinally, the detectability of this epileptiform activity in patients predicted a much faster cognitive decline than in patients where we couldn't detect this activity. And that either suggests that the activity marks a particularly aggressive form of AD or in itself contributes to the progression. Now back to our mouse models, what we showed is that treating the mouse models with an anti-epileptic drug called levetiracetam reversed synaptic plasticity or long-term potentiation deficits in the APP transgenic mice. You can see here the impaired LTP in a saline-treated APP mouse, and when it was treated for 3 or 4 weeks with levetiracetam, that normalized synaptic plasticity in these mice, suggesting that the aberrant hyperexcitability of the network was upstream of the synaptic depression in this model. And this was associated with better performance in learning and memory tasks, such as the Morris-Water maze, where you can see the impaired learning curve of the saline-treated mice and the improvement with levetiracetam treatment. And here in the memory or probe trial, the saline-treated APP mice missed the criterion of favoring the target quadrant, whereas the levetiracetam-treated mice, they make it. Now, Keith Vossel has taken this forward into a small clinical trial of levetiracetam in people who either did or didn't have detectable epileptiform activity, and showed some benefits of levetiracetam treatment in patients with Alzheimer's disease in whom he was able to detect the epileptiform activity. So we're pursuing this a little bit further. This is not true for all anti-epileptic drugs. There seems to be something quite unique about this one. We can talk about what that might be later on. It acts on the SV2A synaptic vesicle protein and may have some very intriguing plasticity enhancing functions, as well as suppressing this aberrant network hyperexcitability. Now, Ina Slutsky at Tel Aviv University published this interesting review where she made the point that neural network activity through what she calls the core homeostatic machinery of the brain is related to a whole bunch of things that are very important in neurodegenerative diseases, including genome stability, proteostasis mechanisms, energy homeostasis, immune homeostasis, and calcium homeostasis, and that AD triggers might influence all of these things, including, of course, as I showed, the neural network activity. And one thing that emerged from genetic studies is that a lot of the risk factors for AD relate to immune homeostasis, which again seems to be linked in some way to neural activity, which we wanted to interrogate a little bit more because it relates to this intriguing relationship between microglial cells and neurons, which seem to regulate each other. And our question was whether that relationship becomes dysfunctional in disease. So Melanie Dawes in our group looked at amyloid-laden mice at a pretty ripe old age for a mouse of 20 months and blocked their epileptiform activity with two strategies, either by tau reduction, which I showed you before, can reduce epileptiform activity, and with an anti-epileptic drug, levetiracetam. And then she profiled the molecular the molecules expressed by microglial cells. And what we see here on the left side is the EEG recordings. You can see here is the MAPT, stands for microtubule-associated protein tau, and this is the genotype of the mice, and they either did or did not overexpress the AD mutant form of APP. And you can see on the wild-type tau background, they get all this epileptiform activity, and this is essentially prevented on a tau knockout background, as we had shown before. Now, the interesting thing here is now we're looking at microglial gene expression, and we're comparing on a tau wild-type background APP versus no APP, and the amyloid deposition shifts all of these inflammatory genes up, so this is increased expression, and the synaptic genes go down, as you can see here. Now, intriguingly, when we remove tau in APP mice, all of these genes shift over where they belong, so suggesting that in association with this suppressed epileptiform activity, we also change very drastically the response, the plasticity of the microglial cells that are reacting to this network hyperexcitability. Now, to further substantiate that this really reflected changes in network activity, we treated mice at the ripe old age of 22 to 26 months with levetiracetam for 4 months. And we looked at their EEG recordings. As we'd shown before, we can suppress the epileptiform activity, and we can improve their performance in learning and memory tasks. Here, the active place avoidance, where up is worse and down is better. Now, when we compared APP-dependent gene expression, mostly in microglial cells that were prevented by tau ablation or reversed by levetiracetam treatment, there was a very nice overlap, 15 gene products that we found by string network analysis to all sort of hang together and be related to this sort of disease-associated microglial profile. And we then wondered whether these changes mostly represented detrimental effects or beneficial effects, and we found a mix. So C1qa, for example, complement factor, is probably mostly detrimental and can lead to elimination of synapses. But quite a while back, Tony Viscora and we published this work highlighting that inflammation is really a double-edged sword, and that there isn't just bad, that there aren't just bad components to it, but also beneficial effects. And that is particularly the case for 2 factors that came out of our analysis here, TREM2 and the very closely related adapter signaling protein TYROBP, or DAP12, which is increased in APP mice, and but only on a tau wild-type background. Tau reduction eliminates this, similarly to TYROBP, and levetiracetam treatment reverses this. So you might look at this and say, okay, well, that's probably something bad, but it could also be a reactive adaptive response. So we wanted to understand this better because there are actually several variants in TREM2 that have a very high impact to the similar level of ApoE4 in terms of odds ratio, but it's a much rarer variant than ApoE4 on AD risk. And so these tend to be all hypofunction variants. So if there is less TREM2 signaling, there is an increased risk of Alzheimer's disease. And the assumption is usually that this may relate to the interaction of microglia with amyloid plaques, which may definitely be true. But because of our interest in network hyperexcitability, we asked whether TREM2 actually helps microglia suppress network hyperexcitability. And Melanie tested this in heterozygous TREM2 knockout mice, which she challenged with the excitotoxin kainate. And she showed that the hypofunction variant TREM2 mice had a much greater response, much more epileptiform spikes in response to kainate, and it took them way longer to suppress this. So you could imagine that if in a brain a microinfarct triggers excitotoxicity, this would be allowed to have a much greater impact on the brain, with much more persistence if there is TREM2 hypofunction. We then extended these studies into mice that expressed mutant APP and that have amyloid in the brain. And we could show that hypofunction of TREM2 also exacerbated the spontaneous epileptiform activity that emerges in these mice. And when, in collaboration with Li Gan, we used knock-in mice that express one of the AD variants seen in humans, we found a similar effect, that the hypofunction variant that increases AD risk in humans exacerbated the development of this subclinical epileptiform activity in the networks of these mice. So our overall interpretation of this is that network and immune cell dysfunction engage in a vicious cycle that ultimately leads to synaptic impairments and cognitive decline, that different people may enter this vicious cycle from different angles. It could be interneuron impairments, microinfarcts, or trauma, as you heard so nicely from Anne, in some cases that would trigger network dysfunction, but also stimulate immune responses. And if these immune responses are dysfunctional through genetic variants or other triggers, that cycle gets fueled. But fortunately, there are some interesting therapeutic entry points, as I showed you. The network dysfunction in AD can be blocked with anti-epileptics, some but not others, and also clearly by tau reduction. And I can talk a little bit more if I have time about these interventions. And then there are some very fascinating antibodies targeting complement, fibrin, and TREM2, either antagonistic or agonistic antibodies that are very close to clinical deployment as well. So I wanted to acknowledge my group of people who contributed to this. Our former lab members, several have gone on to wonderful positions, and collaborators at Gladstone and UCSF. Many people have given us very generously mouse models and reagents and our financial support. And I don't know how I'm doing with time. Do I have time for more? I don't. Okay. I suspected this. Okay. Yeah. All right. I'll stop right here. Yeah. No problem. Thank you.
Thank you, Leonard. Was a great and enlightening lecture. And now the second lecture is about brain reserve and the risk of neurodegenerative diseases, and specifically on motor reserve. And Angelo Quartarone is a speaker. Angelo is a professor from Messina, and he's a neurophysiologist by training who has given the way of different plasticity enhancer methods and markers. So, Angelo.
Thank you, Lice, and thank you for the organization and the foundation, Prada Foundation, and Professor Giancarlo Comi to invited me today. So in this afternoon, I will focus on brain reserve and the risk of neurodegenerative diseases. This is the outline. We will talk about brain reserve, cognitive and motor reserve. But I will specifically focus on motor reserve, because while on cognitive reserve there is a lot of literature published, I have recently went through the literature and trying to write a meta-analysis on motor reserve. And to tell the truth, there are just few papers focusing on that. I scanned many papers from the literature. I came out at the end only with 15 papers. So I think it's something that is neglected and need to be clarified in future studies. Then we will try to explore, to see how it's possible to explore and quantify motor reserve. And we will talk about the use of non-invasive brain stimulation and brain reserve. So the concept of cognitive reserve is widely accepted in Alzheimer's disease to account for individual differences in susceptibility to neurodegenerative pathology. In general, people with greater cognitive reserve can cope better, can endure more AD-related pathology before the onset of cognitive dysfunction, or will have a less severe clinical presentation at similar levels of pathology. The brain reserve, which is the resilience of pre-existing cognitive networks to cope with brain changes in neural compensation, the compensation by using other networks, may underline the cognitive reserve against AD pathology. But what is really curious, is weird, that once clinical dementia become manifest, patients with high cognitive reserve usually show a more rapid cognitive decline compared to those with low cognitive reserve. We can talk about this in the discussion if you like. The concept of motor reserve in Parkinson's disease could explain the heterogeneity of symptom severity despite similar degree of nigrostriatal dopamine depletion on dopamine DAT scan. In line with the concept of cognitive reserve, neural reserve or motor reserve in PD should be closely linked to the motor function network. And the so-called brain reserve may rely on, for sure, on genetic factors. And there are some putative genes that have been indicated as responsible to rule out plasticity in the brain of healthy subjects, but also patients with neurological diseases. You see the connection of healthy subjects compared with AD. And there is an important polymorphism that you probably, most of you know, the BDNF polymorphism, where if there is a substitution of a valine with a methionine, this can produce some profound changes in the level and the efficiency of synaptic circuits. In other words, patients or healthy subjects carrying the VAL amino acid have a better plasticity compared with patients or healthy subjects carrying a MET substitution. And the situation got worse if you can get also some eta4 ApoE genes. So in other words, there are genes that can profoundly affect plasticity in our brain. And this is something that is that we got from when we are born. It's something that cannot be modified. Of course, there are other genes that are less known. For instance, the NT3 gene, the YT1 gene. The YT1 gene is implicated in dystonia. You probably know that dystonia is considered a problem of synaptic plasticity. There are the genes you see, HL1 and TKR, the COMT, and some cholinergic polymorphism. So there is a lot to study also in these kind of genes, because also these genes can contribute to synaptic plasticity. So one important problem in motor reserve in general is how can we assess motor reserve? One first tool could be neuroimaging, for instance, that scan, MRI structural studies, functional MRI during resting state, and EEG. This is a paper which appeared a few years ago on Parkinson's disease, looking at the neural basis of motor reserve in Parkinson's disease. The motor network here was composed by basal ganglia, inferior frontal cortex, insula, cerebellal vermis, which may be the base of motor reserve, according to these authors. And estimate of the motor reserve of each patient may be predicted by baseline UPDRS part 3 score and data availability in the posterior putamen. And motor reserve was calculated as the difference between the actual and the predicted value of the PDRS part 3 score based on that data, with higher values indicating a greater motor reserve. So patients with increased functional connectivity within this motor network seems to have the greater motor reserve. So this is one of the first attempts to try to understand what's going on. And individuals with low motor reserve might manifest Parkinsonian symptoms with mild PD pathology. And when that activity is used in the posterior putamen, is used as an indirect marker for PD pathological burden, individuals with low motor reserve exhibited high PDRS motor scores at similar level of that activity you see here. So which are the premorbid experience shaping brain reserve and possibly contributing to motor reserve in Parkinson's disease? There are some modifiable factor and unmodifiable factor. Let's start from the unmodifiable factor. Age, in general, old age tend to reduce motor reserve because we know that physiologically the level, the amount, the efficiency of synaptic plasticity decline over the age. It doesn't disappear. So even a 90 years old man or woman can express some level of plasticity, but the level of plasticity is for sure different compared to a young people. So age, sex, there are no association with sex for motor reserve, and dominance. The dominant side onset of PD increase motor reserve. Patients with olfactory dysfunction, normal osmic PD have a greater motor reserve. RBD, presence of RBD tend to reduce motor reserve. Depression tend to reduce motor reserve. In generally, high motor burden tend to reduce motor reserve. And also the striatal dot loss pattern is important. Anterior-posterior gradient increase, the asymmetry increase motor reserve. Oh, sorry. And then there are modifiable factors which can be used to build up our motor reserve, which can be, first of all, physical activity. Physical activity tends to, for sure, increase motor reserve. Education, high educational attainment, it is important not only for cognitive reserve, but also for motor reserve. Smoking, there's no association. Body mass index seems to be implicated. Low body mass index seems to be associated with a reduced motor reserve. And interestingly, also the white matter hyperintensity, moderate to severe, seems to reduce motor reserve. This is one example. So you see here the three different color. The highest tertile group, red, show a significant steeper slope compared to the middle with green, and the lower tertile group, suggesting that premorbid engagement in exercise may enhance motor reserve in patients with Parkinson's disease. You see the patients with a higher amount of exercise, which have a lower level of PDRS score. And which are the prognostic implications? The low motor reserve group received higher doses of dopaminergic medication for symptoms controls rather the high motor reserve group. And moreover, the low motor reserve group exhibited an increased risk of developing levodopa-induced dyskinesias and freezing of gait compared to patients with high motor reserve group. This is we have already covered this issue that why matter intensity can contribute to Parkinson's severity. So these observations are consistent with the passive reserve model. In other words, a greater capacity of pre-existing neural substrate rather than an active reserve model, which is a more efficient neural compensation that might result in less rapid progression once Parkinsonian symptoms manifest. Therefore, factors which can enhance passive motor reserve, which are, as we said, education or physical activity, may be protective against PD pathology throughout the disease course and can be used to prevent the development of the disease. Both passive and active reserve may rely on adaptive neuroplasticity phenomena that shape neural plasticity, neural circuits, more plasticity, more resilience to injury. However, as we have heard in the previous lecture, excessive plasticity within motor reserve circuits may become detrimental and maladaptive, such in dystonia and dyskinesias. And we have also seen in Alzheimer's disease that excessive plasticity within the hippocampus may produce epileptic seizures, which can be exert a detrimental effect. So another possibility to assess motor reserve is using neurophysiology, and particularly transcranial magnetic stimulation. With transcranial magnetic stimulation, it is possible to evaluate in a non-invasive way cortical excitability. So we can test, for instance, the excitability of motor area, of primary motor area, and we can build up some intensity curves, some recruitment curve, which are important to give an overall idea of the synaptic strength of primary motor area. But we can induce a different TMS protocol, which can induce artificially can induce plasticity in the cortex. So in other words, we can test the responsiveness of the cortex to these artificial protocols, which are applied, which can be an indirect measure, perhaps of motor reserve, depends on the area we are going to stimulate. And we can test other measures, for instance, using two coils. We can test the connectivity between different areas. So we can test premotor-motor connectivity. We can test the parieto-motor connectivity. We can test the connectivity between the two cortices, primary motor cortex through the corpus callosum. So we have the possibility to test more in the indirect way motor reserve with different protocols. But most importantly, TMS can be induced to enhance motor reserve. I'm not covering this topic because this topic will be covered by the next lecture by Letizia Leucani. So again, this is just to show you the possible use of TMS, assessment of motor system interfacing TMS with other techniques. TMS can be interfaced with functional MRI, can be interfaced also with MEG, TMSG. Again, you can test again the overall synaptic activity of the brain, and can use it, as I said before, to enhance the motor reserve. Those are some of the protocols that can be used to manipulate plasticity and enhance motor reserve. There are protocols which reduce the excitability and protocols which increase excitability. So I don't go through this. I think that Letizia will cover this in a few minutes. And then another possibility of TMS. Do you have another 5 minutes, Licia? Okay. Another possibility is to interface TMS with some behavioral measures. And I would like to show you some work that I've developed with Licia Ghilardi a few years ago. Was a paper, was now published, my gosh, 10 years ago. And where we looked, we used TMS to test the endurance of motor system and during fatiguing exercise. So pretending to come up with a measure of a component of motor reserve. The experiment was very simple. You see, it was done in healthy subjects. You see, the subjects have to wear an engineered glove, and they have to do a very simple sequential movement with the fingers, like that, keeping the rhythm of 2 hertz. And for 5 minutes was the first experimental setting, and for 10 minutes. And then we measure at baseline, motor resting threshold, the amplitude of MEP, and the parameter which is called pre-movement facilitation. Pre-movement facilitation is a very simple parameter, can be tested with transcranial magnetic stimulation. It's very simple, it's based on a reaction time. So once we prepare to do a movement, of course, there are events in our motor areas that are preparing for initiating the movements. So if you test cortical excitability 50, 100, or 150 milliseconds before the start of the movement, there is a huge increase in cortical excitability. And it is logical because our motor cortex is preparing to movement. Okay, so it's a very simple test in which we test this huge increase in cortical excitability in our cortex before the movement. So that's the results of this experiment. You see 5 minutes, this is the behavioral part that was monitored using this engineered glove. You see that between the first and the last minute, there is no fatigue. The subject can keep going with the rhythm of 2 hertz, and there are no errors, there is nothing. There is a very good performance. Let's see what happened in the second set of experiment, 10 minutes. This is the first minute, this is the last part of this 10 minutes exercise. What do you find? That the exercise became more variable, there are more errors because the people start to be fatigued, okay? So what happened in terms of corticospinal excitability? We are talking about normal subjects, okay? So here, the 5 minutes where the subject perform, the normal subject perform well. You see that there are no changes in the motor evoked potential. This is before, this is after the 5 minutes task. And you see that what is interesting, that this pre-movement facilitation, this is before the task. You see that, as I told you before, there is this huge increase compared to baseline of cortical excitability just before the movement, 50 milliseconds before the movement. And this increase further after five minutes of exercise. So 5 minutes of exercise push up this pre-movement facilitation. In other words, this pre-movement facilitation is like a servo mechanism that try to maintain the performance to a good level. Let's see what happened after 10 minutes of exercise. You see that after 10 minutes of exercise, there is a drop in the amplitude of the potential here, the motor evoked potential, and there is also a drop of this pre-movement facilitation. In other words, there is a saturation of this pre-movement facilitation, and this mechanism, this pre-movement facilitation, is not able anymore to sustain, to cope, and to push up motor performance in our subjects. Okay? So can this pre-movement facilitation contribute to maintaining motor reserve, or is it an indirect measure of motor reserve, this pre-movement facilitation? We studied this pre-movement facilitation in a set of patients. Those were patients with multiple sclerosis. We divide patients in patients with fatigue and patients without fatigue. And we applied the same protocol, but without doing any fatiguing task. So we just measured the pre-movement facilitation at rest. And you know that patients with multiple sclerosis are fatigued even when they wake up during the morning. So fatigue is something that is not necessarily related to fatiguing movement. So what we see here is the pre-movement facilitation 50, 100, 150 milliseconds. Those are normal subjects. Those are the subjects with multiple sclerosis with with no fatigue. With these patients with fatigue, you see that patients with fatigue, there is no modulation of this pre-movement facilitation. So this servo mechanism, which is important to maintain motor performance, is not working in patients with multiple sclerosis. Yeah, and we tested also these the same protocol of before again in patients with multiple sclerosis. So again we used this gloved engineer for 5 minutes, and we tested the excitability at baseline immediately after 15 minutes and 30 minutes. And what you can see, this is the behavioral part. Patients with multiple sclerosis cannot perform even after 5 minutes. They are not able to maintain a good motor performance. And what is interesting that, again, there is no modulation of this pre-movement facilitation. Okay? So patients with multiple sclerosis, especially with fatigue, probably have a reduced motor reserve. So which are the factors that can contribute to reserve in the motor domain? We have seen genetics is very important for brain reserve in general, but then these brain reserve can be shaped by different things, different factors, daily life activity, musical training. For instance, we know that musicians, performers have, especially for the motor system, have probably a very enhanced motor reserve because they are retraining, they are doing rehearsal every day, they're training for many hours a day. Sport exercise is another important part for building up a good motor reserve, social context, leisure activity, occupation, and education. And the last slide, so which are the future perspectives? So I think one of the first questions is establish which is the best time window to build up a motor reserve, which I guess is early in life. Evaluate the role of additional genes implicated in synaptic plasticity, possibly contributing to brain reserve. Find out reliable markers for motor and cognitive reserve, questionnaires. For instance, there are questionnaires for cognitive reserve, but they are not validated questionnaires for motor reserve. Neurophysiological markers, neuroimaging markers, exercise, especially with the aerobical component. So we should study some protocols which can specifically enhance motor reserve using the aerobical component. And then empowering motor and cognitive reserve using non-invasive brain stimulation. Thank you.
Thank you, Angelo, for the beautiful lecture. And now, least but not last, Letizia Leocani, professor of neurology in Milan and chair of different committees and president of the Italian Society of Clinical Neurophysiology. She will tell us about how we can enhance brain plasticity with neuromodulation. Thank you, Letizia.
Thank you very much, Lice. I thank very much the organizers, Prada Foundation and Professor Comi, for having me here to this great event. I'm really learning a lot. And my main disclosure is that I'm the chair of the EMA expert panel for medical devices on the nervous system, which is a field that is getting attention on the by the regulators, similarly to drugs. We heard this morning and yesterday that enriching our environment is going to give us a richer brain, richer in synapses, but many studies are in young people. But how about if you're already over 70, 80? I mean, we have great examples of elderly people who do great despite their age. This lady is like now 98, and she's doing things I never did and I will never do. But the issue is that they all started when they were young, but can we still learn something new when we get older? Actually, there are also studies on that demonstrating that even people over 65, they can learn to juggle with the classical 3-ball game. Maybe it takes a couple of months, maybe not all of them, just a smaller percentage of people can learn that, but they can also show a thicker brain cortex related to that. But that's a good news, but the bad news is that it's reversible as anything we learn. We know from our studies at the university, we forgot many things. And so the brain tends to reverse what is not used. Can we do something to improve the percentage of people that will be able to learn something new and retain it, and so also potentiate the retention of what we have learned? We have, I will focus on a couple of non-invasive tools that are the oldest, but of course there are new things around that are already published. But I will focus on the transcranial direct current stimulation and transcranial magnetic stimulation. So thank you, Angelo, for introducing that. I will start with the oldest method. You see, that's 1962, where it was demonstrated that if you induce cathodal currents, you can reduce the activity, spontaneous firing of neurons, that's a cat cortex, and with anodal currents, you can increase it. So you can modulate the propensity of a neuron to fire. So this is a very good method to also increase or decrease brain excitability, and that was shown that you can increase the excitability of the motor cortex. That can be tested with motor evoked potentials, as Angelo mentioned before. So you can increase the responses, the muscle responses to brain stimulation if you give anodal currents for about 20 minutes, and decrease it if you give cathodal currents. But of course, with a single session, the effect will last about like half an hour up to 90 minutes. But the nice thing is that you can repeat sessions and have something that is building up on top of what we learn, because when we learn, we also build new synapses, and we can retain what we learn also the day after and keep it for days. But if on top of our learning, this is a hand motor skill learning in healthy young people, so that's what they can learn in 5 days with sham stimulation. Sham means placebo, so apparent, fake stimulation. But if on top of your training you do also electrical current, the excitatory current, you can learn better and retain better what you have learned. So this could be nice also for like studying a new language or playing a new sports. But the bad side of it is that thanks to our science and publications, the idea came around, and so on Amazon you can also find devices from 100 euros to 400 euros that you can use to deliver yourself brain stimulation, which is a bit scary because these are non-invasive tools if you use it safely. But of course, you can be now free of cooking your brain if you like. So I don't think it's a good idea, and that's why we need regulators to keep an eye on that, and that's what actually they're doing now. And another possibility is that also posing ethical consequences, that doping. Doping is normally done with drugs, but we can do electrical doping. And some teams have already bought some devices to, this is a headset delivering music, but on here, on this side, Angelo showed the homunculus is right here in the middle of our head, so you can also deliver currents on the homunculus. But a colleague of mine went to check what these teams did, and actually didn't win so much yet. So I think if we want to be taken seriously, we have to behave seriously. So what we need is to use a device also with a proper randomized clinical trial and with adequate numbers. So statistics with 8 people are not acceptable. So having said that, we already have a good evidence. I'm showing the field of multiple sclerosis, which I'm most familiar with. And we already have serious randomized clinical trials reducing several symptoms from pain to fatigue and enhancing the effects of cognitive rehabilitation. And I said the backside of these devices is that any people can buy them, but the good side is that we can deliver TDCS at home and so facilitate cognitive rehabilitation now with digital also training. So we should take the positive part of it. I'm showing an example on the fact that being these devices not so expensive, there is not much investment by companies. So the evidence we have are from meta-analysis, again, from small studies, and we need really big public funding because maybe some not so straightforward results like meta-analysis in Alzheimer's disease gave contrasting results, but there is a lot of heterogeneity between studies. So meta-analyses are not the key. The key is to have big fundings and get big data, which is actually the case for transcranial magnetic stimulation. That's also an old device since the 80s, is already around. And it was developed to test the motor conduction. So it is a diagnostic tool if you use it with a single pulse and measure the muscle response. But if you use it repetitively, as Angelo anticipated, you can also induce brain plasticity and change it into increasing the excitability with high frequency stimulation or decrease it with low frequency. By low frequency, I mean less than 1 hertz. And by high frequency, it's stated over 5 hertz, but nobody's using that anymore. It's beyond 10 hertz or 20 hertz. But for this type of technique, we have already big studies because there were investments by companies who sell it now to the psychiatrist because now it has been FDA approved for drug-resistant depression that did not respond to a first drug. And the approvals extended to obsessive-compulsive disorder, but unfortunately we don't have much for neurology and for neurodegenerative diseases. Although, again, efforts performing meta-analysis on that are promising towards the use of the stimulation of the motor cortex to improve dysfunction in people with stroke or with multiple sclerosis. And I'm happy to underline also people in the room like Angelo and Professor Di Lazzaro among the panel of experts that I'm proud to be part of. And here we already have some meta-analysis, but maybe I can be happier about it. One reason could be that the numbers tend to be with the higher doses in terms of number of sessions, not just 5 sessions as traditionally for the TDCS. But maybe one other possibility is that these energies involved are stronger. If you deliver repetitive stimulation to a hand motor area, you don't see any movement if you deliver it below threshold, but you can see it's not below threshold for neuronal activity because if you perform functional MRI, you can see you can activate the whole motor network that we use for voluntary movement. So maybe here we're dealing with a more powerful tool. And but we can also consider that in neurodegenerative diseases, we cannot use the focal coil that we use for depression to target a very focal area, about 2 centimeters. So we need to evolve also the type of device or the way we use it. We had the lucky condition to be able to test a device developed for wider stimulations called the H-coil, that is allowing a wider field and bilateral stimulation, so we thought it was more suitable for degenerative conditions like Parkinson's disease and Alzheimer's disease. But we were a bit skeptical, so we first tried in a very small region. We tried on a patient with progressive aphasia. We did PET with Professor Perani, and we did PET before and after, and we actually saw it did increase the metabolism besides improving the aphasia of the patient. So we were encouraged to try it on other conditions and on a larger scale. So we initially started to find a nice effect on motor abilities in people with Parkinson's disease by using a bilateral and widespread stimulation. But again, this was not a placebo-controlled study, so we have to be careful on these studies, like the one I showed you, although it was ours. It was a pilot to make sure we did something before entering into a randomized study. Because if you just pretend to stimulate the brain of a patient with Parkinson's disease and you don't, you just give the noise delivered on top of the head, you can see that there is a dopamine increase in the brain. So this is very well explaining a huge placebo effect that we see in people with Parkinson's disease. So we had to do, there no way to escape, a randomized controlled trial. And then we found that after a month of treatment, of repeated sessions, just 12 sessions, 3 times a week, there was a placebo effect, so motor performance improved also in the sham stimulation, but that was significantly less compared to the improvement we saw after the real stimulation. And for tremor, the difference is quite even better because there is almost no placebo effect. But then we moved to Alzheimer's disease, again, a big area of stimulation, widespread frontal, parietal, and temporal regions. Again, we invested a month of the patient's time coming back and forth 3 times a week, and we found a significant improvement in cognition. So this was nice to have. We could publish it, but it wasn't nice for the patient because after 8 weeks, so a month after the end of treatment, the patients went back to the same situation as the ones who had placebo. So this is not a very happy ending for a study like that. So we should do something to potentiate the effects of what we do. And maybe we could couple the brain stimulation with training. Like Angelo said, we can improve plasticity by training on the activity we want to improve with the stimulation. And Diego Centonce, who gave a great talk this morning, was the first to show in MS that if you give rehabilitation or magnetic stimulation alone, you have less effects than if you combine the two, which is the key to go. So we tried, for example, hand bilateral stimulation in people with chronic stroke, but we coupled it with motor training, with special manipulations. And again, we were successful even after a month, so we were happier about that. And the same in the lower limb with, again, in chronic stroke, we were capable of doubling the meaningful change for people with chronic stroke if we combine just the stimulation with just the static cycling. So again, the combination of training and brain stimulation seemed the way to go. And we moved into people undergoing intensive rehabilitation. So on top of rehabilitation, patients could be randomized in a pilot study on 23 people. We were able to improve much better and significantly better motor speed and motor endurance in terms of gait compared with the placebo stimulation. So that was on top of rehabilitation. And then we moved to a bigger study on 100 people with MS, combining rehabilitation and brain stimulation. And again, here we didn't find a significant difference at the end of treatment. So again, 3 weeks of intensive combination. But when the patients went home, at week 15, actually the ones who had the sham stimulation came back to the initial condition, while the others with the real stimulation, the real combination, kept improving. So maybe we have a tool not only to improve plasticity, but to retain what we have learned. But the bad news is I heard a question about respondents this morning. I think it's very interesting because maybe the respondents to that are those with sufficient corticospinal reserve. What we found in our patients was that patients with already absent motor evoked potentials to the lower limbs behaved actually like people undergoing a sham stimulation, while the real respondents were those with some residual corticospinal motor response after a single pulse brain stimulation. So we also repeated on an open label fashion the combination of rehabilitation and brain stimulation. And again, even neurodegeneration that we could pick in the retina was enough to predict that if it was beyond a given level, the patients would not be able to respond even to the combined approach. So measuring the reserve could be also a predictor of response to this combination. And also from the literature, if you see the literature on the retainment of the good effects of repetitive stimulation in Alzheimer's disease, the best results, the best lasting results are in people with early Alzheimer's disease or MCI. So maybe this is the way to go to select the most likely to respond. And also, as we already saw from the two previous lectures, there is already abnormal plasticity in these conditions, like demonstrated in Alzheimer's disease. There is no enhancement of motor responses to 5 Hz stimulation in Alzheimer's disease. And there is hyperexcitability in Alzheimer's disease that can be also picked with the transcranial magnetic stimulation that can separate Alzheimer's patients from healthy subjects, but also correlate with cognitive tests and with the tau levels in the CSF. But maybe we can use these methods also for other purposes, also to directly target the disease. To test that, we implemented the TDCS, which is easier to translate into mice, reverse translation, and we were able to use a non-invasive stimulation to increase or decrease motor responses or visual responses, visual evoked potentials. And the most predictable result was that if you give excitatory currents, not only you can improve plasticity as demonstrated in several pieces of literature, but also in models of demyelination, like the Cuprizon demyelination, we can see that not only the delayed visual evoked potentials can improve after a cycle of direct current stimulation, but we can also improve motor behavior, like the beam test in the speed of crossing the beam. And we can improve the myelin content in the optic nerve that well correlates with improvement in VEP latency. But that's, again, the excitatory approach. By the way, it took us 15 years of projects and 2 years of Elena's work to get this. So this is not easy as it looks like. But maybe the story is not so easy when we deal with inflammatory conditions, because we saw that there are accompanying neurodegeneration. So maybe going to excite the brain is not always good. We saw there is epilepsy also in Alzheimer's disease. So maybe we should use inhibitory stimulation. And actually, that's what we did, for example, in the hyperacute phases of stroke. The protective currents were the inhibitory currents. And again, in the initial stages of EAE, the inflammatory counterpart in animal models for MS, the way to prevent from the delay in visual potentials, to prevent demyelination and the abnormalities in the node-paranode complex was to give, again, inhibitory currents that prevents from developing the demyelination and delay, and also reduced inflammation. So maybe this is something we have to consider. But beyond what I showed you for preclinical studies, there are people who already went and translated into real practice and clinical studies this possibility. That was Professor Di Lazzaro that applied static magnetic stimulation. But of course, it took 6 months, and they're following people with ALS for over 1 year now. But they were able to delay the need for tracheostomy in these patients. And again, it was an inhibitory approach and not, to counteract the hyperexcitability. So to conclude, I think in the future we will be able to not only promote brain plasticity by combining technologies for training and for delivering magnetic and electric currents, but also we will need to early detect the initial dysfunctions to better targeting of those and improve our reserve by a combined approach. Thank you so much.
Thank you, Letizia, for the great lecture and the perspective. So I'm inviting now Leonard and Angelo together with Letizia. And we can start the discussion. Are there questions in the audience? There.
Thank you, Elena from San Raffaele Hospital. Thank you all for the great session. I had a question in regards of the epileptic seizure-like spikes in Alzheimer's, and I'm not an EEG expert, but I was just curious. There is both evidence in these terms, but there's also evidence that, for example, induction of gamma fast spiking electrical activity can reduce Alzheimer's load, and on the contrary, that reduction of gamma waves is associated with the onset of plaques. So I was wondering in these terms whether this high spikes that you see that are sort of related to enhanced disease, how can they be explained in comparison to the fact that gamma can actually sort of be...
Yeah, let's see, is this on? Yeah, so, you know, gamma oscillatory activity isn't epileptic spiking. So this is a very different thing. This is a brain rhythm at that particular frequency. And so I think, you know, that the 40 hertz stimulation that Li Wei Tsai has showed to be beneficial, I think we're still sort of waiting to see this play out in replicated larger studies in the clinic, whether it really modifies in a beneficial way Alzheimer pathology. But I think these are two very different things. In fact, people with epileptic activity tend to have oftentimes impaired gamma oscillatory activity, which may relate to interneuron dysfunction. And we are actively trying to enhance parvalbumin cell activity with NAV 1.1 enhancers, for instance, in order to boost up the power of the gamma oscillatory activity. So I think there are two different phenomena, but they could be linked by interneuron impairments, which would be predicted to make way for hypersynchronous network activity and spikes while diminishing the power of gamma.
While we wait for another question, I want to ask all of you if you consider sleep in all the experiments that you did in terms of plasticity, or comments about it.
I don't, so I welcome Angelo to say.
Plasticity is for sure is a very important component, because as we already heard this morning, it's the crucial part of the day where the homeostatic mechanism, the depotentiating mechanism, the downscaling mechanism take place. And of course, to build up a correct motor reserve or cognitive reserve, you have, I mean, plasticity must be regulated very carefully during night. So I think we haven't done this in our experiment, but I think it's very important part that it need to be considered in future experiment addressing motor reserve.
As there are young people here in the audience, I would like to say we don't work on sleep, but there are data on brain excitability after sleep deprivation that show that the sleep deprivation, like in young residents who stay awake all night, so the morning, their brain is hyper excitable. So yeah.
The
After
hospital.
being on call, yeah. So, I mean, this is very important to keep in mind.
So in terms of the outcome, for long-term outcome with a patient study, maybe sleeps, it will be something that should be monitored in future.
Yeah, I mean, in terms of sleep, we certainly see most of the subclinical epileptiform activity during the sleep phase in people with Alzheimer's disease. And there is so much evidence for sleep disruption in AD and in Parkinson's disease early on. And there is good evidence that that does really interfere with the purging of protein accumulations through the glymphatic system. That, one would argue that good non-REM sleep in particular may facilitate the clearance of proteins that accumulate in AD and in PD. So one should, have a very close eye on sleep hygiene, and anything that interferes with that would be expected to backfire.
Patients with PD that have a hard BD have a lower motor reserve. There are several studies showing that.
And also, another approach is the accelerated interventions, like multiple sessions in a day to reduce the number of days. And I think this is important because if we need sleep to consolidate plasticity, we really need to compare whether it is better to interspace sessions with a good night's sleep and find the ideal, not only dose, but the ideal interval. I was intrigued by the BDNF dosing, like, that it will last a couple of days and then you need to exercise again. So these data are very important also to know also the interval for these. And maybe also to know that to train every day is not needed. Maybe every other day will be enough. So that's also good news for people who don't like to train too much.
Can you hear me? So, sorry, this question is for Angelo. The motor reserve mechanism, are you taking into account compensatory mechanisms that might occur that engage different brain areas into sort of this idea that these established motor reserve circuits involving motor control might also engage these other brain areas?
Yeah, that's a good question. For instance, taking the example of Parkinson's disease, we can take other examples. But taking the example of Parkinson's disease, you know that the disease started many, many years before. Once there is a depletion of dopamine in the substantia nigra. But the disease becomes manifest only when a certain amount of depletion reach the critical level. So, and during this period, there are several plastic phenomena, adaptive phenomena that are going on, in which there is either there are phenomena locally at the level of the basal ganglia circuits, but also in remote area, which try to compensate the deficit. So there is a mixture also of compensation of network, which try in this case to delay the onset of the disease.
I wanted to make the comment, to what you were pointing out in terms of numbers. I think this is really important because when we look at the pharmacological trials for Alzheimer's disease, we're now talking about enrollment of 1,850 plus patients in order to get conclusive results, right? And a lot of the attempts at either behavioral study modifications or the device-related studies are nowhere close to those numbers. So I think it's going to be really important to build up, and I'm not entirely sure how to do it. I also wonder, what would be a better way to go through different stimulation protocols, because I could imagine that it's just hard to guess what will work the best. And so how can one test many different stimulation protocols?
Yeah, this is very important. Actually, you make me think we could be optimistic because even the trial on major depression that gave FDA approval was on 600 patients. So, I mean, we are speaking about effect sizes that are larger compared to drug. But I have to say, to get larger numbers and compare different protocols, again, we need funding, which is not the case for, I mean, drug treatments. It's easier to get.
About the use of algorithm, lecture yesterday about it, and so we can use that type of approach to decrease the variability and so on, or you think that we need hands-on experiments, or artificial intelligence and different algorithms can help in decreasing the number and making everything speedier?
May start. I think it's a great idea. Actually, I asked yesterday about this possibility. But that's if you want to target neuroprotection, so you need to wait years. So having an algorithm with prediction model would be great. But so far, and so it maybe would be great for the measuring the effects of reserve. But so far, when we target a sort of symptoms like cognitive performance, and we want to have immediate effects, this is not enough, because we would like to see short-term changes after a month or two. And so these models are not ready yet for that.
I have another question for Professor Quartarone. Since he gave the hint regarding the cognitive reserve, I would like him to expand on the concept that higher cognitive reserve sometimes can lead to a faster decline once dementia is manifested. And another question for Professor Locani regarding non-invasive neuromodulation technique. As I am a psychiatrist, but also head of research at Parasime, which is a leader nanotechnology company in non-invasive VNS, since we have seen cortical NIMS, but there are also other potential non-invasive neuromodulation techniques, if you have some data, for instance, regarding VNS and neuroplasticity.
Okay, thank you for your question. There are two possibilities. One is it's obvious. So I start from the most obvious, because I mean, if the people have a high cognitive reserve, presumably they will get the disease when they are 80s, 85s, 90s, and you know that at that age it's easier, because but then the plasticity collapse, and it's easier to get a progression which is more rapidly. So I think it's the most obvious. The other possibility is that perhaps, as the colleague has underlined, there are not only adaptive phenomena, but there are also maladaptive phenomena. So there are parts of the hippocampus which may have a higher synaptic activity in developing seizures. So it is likely that these people with a good, very high plasticity, this would be more prone to develop a maladaptive plasticity, which could get the Alzheimer's disease worse. I don't know which one you prefer.
Thank you for the question. I did stay away on purpose from other types of techniques that do not give me enough data for meta-analysis, but of course, not only vagal nerve stimulation has been shown. Also this morning we show examples of effects of the vagus nerve on inflammation. So we definitely need to explore this part besides symptoms themselves, and among which there are already preliminary promising data on depression, for example, or epilepsy. But other approaches that I skipped could be related to ultrasounds, or neuromodulation, so low intensity, or modulation of brain activity with different frequency in the auditory and visual modalities, and that's being tried already in preclinical models of Alzheimer's disease, and that's currently tested in Alzheimer's disease. So We need to learn more about the mechanisms, of course, and about, as suggested, not only comparing different approaches, but also why not considering their combination, because I don't see brain stimulation in competition with vagus nerve stimulation, and is not in competition with therapeutic approaches with drugs.
I would like to ask you in some way give a quite relevant importance to the epileptiform discharge. This is because you think that is in some way one of the aspects that may help to promote this level of instability, let's say.
So I think, my initial question experimentally was I wanted to know whether it was upstream or downstream of the cognitive or the let's say the synaptic impairments, which I think are, the best correlate for the cognitive decline in AD is the impairment and loss of synapses. And it wasn't clear to me whether you start with the synaptic loss and then that gives you the network instability, or the network instability is driving the synaptic impairments. And I think the experiments with both the tau reduction and the levetiracetam treatment suggests that the network hyperexcitability can be upstream of the synaptic impairments and then the cognitive loss. So I think it is a rather pragmatic issue where it could be either way, but it turns out that when we block the network hyperexcitability, it does seem to be beneficial in this very complicated context of both too little and too much of something in a very finely tuned network. And I don't quite know, how transcranial stimulation and so on comes in. I would have never predicted, quite frankly, that deep brain stimulation is as effective in Parkinson's as it is. It seems like, you have these finely tuned circuits, and how can we figure out what we should be doing at what time and preserve all the communications? But there is good evidence that there are clearly beneficial effects from it.
So one question I have for you is, what is, in your opinion, the percentage of people with Alzheimer's disease that can have epilepsy?
That's a very difficult thing to answer because of the findings from Mass General with the foramen-ovale electrodes, which are invasive and you can do it, but it clearly shows that we vastly underestimate the number of people. In our study, using very highly sensitive combinations of magnetoencephalography and EEG and long-term EEG monitoring, it was over 40% of patients, but that is probably an underestimation, I would think, and when you talk to people who take care of patients with Alzheimer's disease, it says, well, my patients don't have seizures. That may be true, but we're talking about very subtle things. It can be psychic phenomena, it can be speech arrest, it can be confusional episodes, it can be various things that would escape the sort of gross screen for epileptic activity. So my hunch is that it is quite a substantial number, but nobody can be sure because we're not able to get invasive enough to be sure.
We should treat them.
Yeah, I think, there are, again, trials that people have conducted, but they weren't then pre-selected for people who have or don't have evidence for this, which makes it a little bit hard to know what you get. Did you have a... Sure.
And these epileptic seizures are one of the counterpart of the glutamate excitotoxicity.
It's not clear whether glutamate toxicity is the main cause of that, right?
regulation of GABA activity?
I think that that would be my suspicion. I mean, it could be both, right? There is clear evidence in AD for excitotoxicity from, for example, microinfarcts, and also A beta can lead to that. But there is also an abundance of evidence for interneuron failure in the disease. So you could have a combination of those two, that there are, excitotoxic phenomena in combination with interneuron failure and GABAergic deficiency.
Yeah. Last question, and then you.
Thank you, Eugenie Suter at Roche, and I'm lucky in that these last couple questions have gone exactly in the direction I was hoping. I wanted to come back to the beginning of Professor Mucke's talk and that epileptiform activity, and you took us through, your story progressed from the treatment with Keppra, levetiracetam, and improvement cognitively, and through to inflammation and TREM2. And I wanted to get your view as to how much of these effects are mediated via inflammatory pathways versus directly through things you just mentioned, like the GABA signaling.
Yeah, so I think that's interesting. They're clearly not mutually exclusive. And initially, we were very much focused on interneurons and GABAergic systems. And we're very strongly pursuing this with NAV 1.1 enhancers that look very good. We, screened, and Jorge Pelop in our group now has some that are highly brain available and can be given in chow and clearly enhance gamma oscillatory activity and do precisely what we want, which is quite nice. So this would be a sort of interneuron enhancement strategy that I think should enhance memory encoding and, suppress the network hyperexcitability. So all good things. And it's very clear that microglial cells and also astrocytes regulate neuronal activity in multiple ways. And I think microglia in particular seem to curtail network hyperexcitability, and they use TREM2 to do it, at least in part.
Okay, I think we should go now. Thank you to the audience. Thank you to the speaker for the beautiful talk and the nice discussion. Thank you, everybody.
Well, we continue the program. It's a pleasure to introduce the next speaker who will deliver the keynote 3. My name is Peter Hartung. I'm a neurologist from Düsseldorf, Germany. Now, we had an extensive review of risk factors in neurodegenerative disorders yesterday, and we'll now turn to risk stratification. Now, this is, from a clinical standpoint, a very important concept for a number of reasons. It implies a prognostic assessment of a given individual. It probably allows to make a diagnosis early on, even prior to clinical manifestation, and will enable the design of a personalized management program. Risk stratification is also important on a more mundane but important level in determining an appropriate and justifiable risk-benefit profile for any treatment that you are considering for that individual patient. So a prerequisite for personalized medicine. Now, Professor Sergio Baranzini, a biochemist and molecular geneticist by training, has worked extensively over the past years on immunogenetics and biomarkers in multiple sclerosis. He's professor in the Department of Neurology at UCSF, and more recently turned his attention to translational data science. I mean, how could we grasp better and in a more comprehensive fashion the information available, interrogate multiple databases, various layers of information, molecular, clinical, in our area of interest, multiple sclerosis, MRI, to get a better overview of the complexity of the disease. So he will talk about creating a biomedical knowledge path, a sort of super integrated approach utilizing the information retrieved from multiple sources. So without further ado, I would like to ask Sergio to take the floor, please.
Thank you very much, Hans-Peter, and thank you very much to the Fondazione Prada for the kind invitation, and Professor Comi. It's a great pleasure and really a privilege to be speaking to all of you here today. I think that so far it's been a fantastic conference, at least personally for me. I've learned a lot of topics that are not normally in my realm, and it has been a fantastic display of collective human intelligence. Now we're gonna switch to the artificial kind. And this image, for example, was generated by artificial intelligence, and the prompt that I provided was a fact, actually, that in 2017, humanity had achieved more information than in the history of humanity prior to 2017. So in 1 year, we've generated more information than the prior history of humanity. And this is what it came up with, which I think is a wonderful representation of that. So where am I going with this? We're inundated by data. Some of the data may become information, and that information may become knowledge. And I'm going to explain that a little bit more in more detail. And so then we require the use of computers to help us understand and help us digest that and help us remember. Computers are much better than us at certain tasks, particularly they don't forget. And they can integrate things in a much more systematic way. So if we can only build systems that can understand that and can help us make better decisions, why not? So what I would like to start with is with this concept that it's not a new concept. This paradigm of data, information, and knowledge was brought up first by Russell Acuff in 1989. But the concept is that data is primary results of an experiment. Data is devoid of any contextualization, it's just numbers. Data can become information if it's replicated, if it's synthesized, if it's contextualized. And information can become knowledge if we can start drawing connecting those dots. So creating a cognitive network, if you wish, of all that information. And therefore, knowledge in that sense is an emergent property of that complex network of information. Then we can extend this to insight if we know exactly how to go from point A to point B, and wisdom. And some people take this to a very extreme, which we're not going to deal with that today. But the whole thing is that AI is quickly approaching human intelligence, and why can't we use it for biomedicine? And I will argue that the reason why it hasn't really been as successful, its implementation in biomedicine yet, is because its complexity, which sets it apart from the banking industry, the e-commerce, even the physics. The rules in biomedicine are not clear, are murky, or we're still trying to figure them out. But at some point of abstraction, we do have certainty of certain processes. So why not put all what we know for sure, or that we're certain, into a system that can integrate all that for us? And this is what happens when we're in a biomedical context. Today, patients that come to our clinics are assessed with a multi-modality, if you wish, from clinical neurological examination to laboratory assays to imaging, genetics, even immunophenotyping and many other biomarkers. All this could form what we call the endome. So these are all variables and measurements that are done from the skin in. And then we also have the ectome. So these are all, the microbiome could be half and half, but then we have demographics, we have social determinants of health, exposures, food. So why not integrating everything that we know about a patient when we're trying to assess its state? So I talk about the complexity of biomedicine, but let me walk you through what I'm talking about. So if one tries to build a hierarchy of biological complexity, one might start from where the information generates or initiates, and then how it flows. From the bottom up, we will agree that all the information is contained in the genome, or now metagenome as well, as our second genome. But then the most immediate phenotype will be that of the transcriptome. So that's the most immediate phenotype that we can think of. And then mRNA will be translated into proteins. Proteins will interact in deep and complicated networks, which will form pathways which, one level above, cells will interpret. And cells are great integrators of that information, because cells need to sense their surrounding, their environment, and then they need to produce an output. And a cell is a great integrator because despite hundreds or even thousands of stimuli, it needs to decide what to do. And a cell can decide to proliferate or differentiate or die of apoptosis, but there's not a lot of things that a cell can do at that level. Then if we continue going up, we have cells organized into organs and systems, and all the way up there we have the organism, which is where we actually diagnose, which we will actually see. But then I will claim that most of the research happens in each of these strata and rarely across different strata. And this is a really complex system in which spans 4 orders of magnitude in space, even 5 orders of magnitude in time. And when I'm talking about complex, this is just one layer. This is the metabolic map. So how can we possibly integrate everything into these layers and take it into account when we're seeing a patient? So one possible solution is that of a knowledge graph. And a knowledge graph is a relatively novel idea in the sense that it takes information that is known from curated databases and links them all together via semantic relationships. And semantic relationships in this case means this is what is called a metagraph of a knowledge graph. So a metagraph, because I'm not displaying all the 50 million nodes that it contains, I'm only displaying the few dozen node types that it contains. So when it says gene, well, it contains every gene in the genome. When it says protein, it contains every possible protein variation that is known in humans. Diseases contains more than 9,000 diseases classified in the disease ontology, more than 5 million compounds, millions, hundreds of food items, protein domains, protein families, metabolic reactions, pathways. So diseases are connected to organisms in case they're pathogenic, to symptoms, to pharmacological compounds, and so on. So the idea is to put together in a single system all what we know about everything that we go through when we try to study human physiology, human molecular biology, human physiopathology, and so on. So this is just a quick demonstration of visual imaging of what this knowledge graph has. This knowledge graph, by the way, if you try to go to this website now, it will give you an error. We're updating our server, so please wait one more day. It's a bad day to update the server. But this is a visualization, for example, of all the 9,000 diseases in the graph. They're colored by the disease type. You can color by whatever you want. In this case, neurological diseases are in pink, immunological diseases are in green, cancers are in blue, and so on. You can zoom in and out, and then you can find the disease of interest or the group of diseases of interest, and once you find them, then you can jump to anything else that you might be interested in. Bring me the symptoms of this disease, bring me the therapeutic drugs, or bring me drugs that are in clinical trials for them, and so on. So this is an example. You can start a search in this graph, for example, from any molecule or any gene, any drug, your favorite gene, in this case, let's say interleukin-2 receptor alpha. So you can bring all the, for example, all the polymorphisms, the single nucleotide polymorphisms that are associated to diseases that map to the interleukin-2 receptor alpha. And as such, you can see that there's many immunological diseases that have polymorphisms that associate to their susceptibility, rheumatoid arthritis, multiple sclerosis, and so on. And then you can say, okay, I want to know what is the gene that encodes, what are the transcription factors, for example, that modify the expression of this gene. Well, you have a number of proteins, in this case, in green, that are known transcription factors that modulate the expression of the gene interleukin-2 receptor alpha. And this comes from yet another database. This comes from a database called Transfac. And then you say, okay, NF-kappa B is one of those transcription factors. What else do you know about NF-kappa B? So you can keep going from one thing that you're very familiar with to something that I guarantee 2 clicks away, you're going to learn something, because there's no possible, it's not possible that you will know everything about this, but a computer can. And then NF-kappa B, in this case, for example, is related to a number of complexes. For example, that in this case is a complex with the subfactor P50 and P5065. And then going back to the gene, this gene is part of these pathways, interleukin-7 signaling pathway, apoptosis pathway, RAS signaling, and so on. So you can navigate from one area where you're very familiar with, you might be the world expert on, to something that you're really likely to be unfamiliar with, and you're going to learn something. And this is human exploration. Then we're going to go to machine exploration. So another example is here, the bacterial taxonomy. So there's more than 400,000 bacterial organisms in the database, each of them with their genome. So each of them with the proteins that they express. And there's every metabolic pathway. You can concentrate here, they're grouped by hierarchy, and here's the central carbohydrate metabolism. One of this is the glycolytic pathway, and then you can zoom in and see every single reaction that goes from glucose to pyruvate, and you can, again, know what is the enzymatic reaction that converts each of these compounds. How about the ectome? Well, if we go to the ectome, we've talked yesterday a lot about the importance of food, the kinds of additives and the kind of compounds that food contains. Well, there's the entire food ontology in the in the database, thousands of food items. For some of them, not for everybody, not for all items, but for about 1,000 food items, there is their metabolic composition, so the metabolomic composition of these food items. So in this case, for example, I'm highlighting tomato, and then each of these connections between a food item and in this circle, I'm representing a number of metabolites. Then this comes from databases that actually did mass spectroscopy on thousands of food items, and there's a quantitative assessment of how much lycopene is there in a regular tomato that you buy in the grocery store, and so on. So another example of ectome is prevalence of diseases. This comes from yet another database from the CDC in the US. So it can tell you what is the local prevalence in each county at the county level in the US. Another interesting piece of information from the exposures. So we talked also a lot about exposures and why it is important. The contaminants that are found at each zip code, this comes from water contamination from the Superfunds. Then social determinants of health. This comes from different questionnaires and different agencies that have this population-based surveys. In this case, many social determinants of health by county level. So where are we going with this? Well, towards explainable AI, and explainable is a key word here, because it's not just we're creating a sophisticated model, but we're creating a sophisticated model that we can understand, that it can be explained. It's not a black box. So how are we doing this? One of the students of bioinformatics in my lab developed an algorithm that is called an embedding algorithm. Here on the left-hand side, there's the information from one patient, and on the right-hand side is the graph, spoke itself. So spoke, the graph does not contain any patient information, but then we have access to patient information from our research studies or from the electronic health records of the entire institution at UCSF. We have more than 6 million that have been de-identified and available for research. So what we do is then we embed, we take variables from the, in this case, from the structure portion of the electronic health record, and we map them to nodes in the network. If the patient has a genotype, we can map that. If the patient has a lab test, we can map that. A diagnosis, a medication, so everything that can be mapped is mapped, and then we employ a diffusion algorithm into the network that it gives an importance to each of the nodes in the network based on the input data. And then we can do that with the next patient and then with the next patient. And what this generates is sort of a health barcode, which is a representation of the state of the patient at that point in time in the context of the network. So with this, we tried an example with multiple sclerosis, this disease that I'm interested in. So we looked at our 6 million patients at UCSF, and we identified 5,000 of them that have a diagnosis of MS. Not one diagnosis, but 4 diagnoses. So at least we wanted to make sure that these patients had a MS. And then we went back in time and collected the information up until 1 year, 3 years, 5 years, or 7 years before diagnosis. And then we trained a machine learning model with only this data prior to the diagnosis and compared it to a much, much larger population of individuals without a diagnosis of MS. And then we asked the question, are they different? And then if they are, how can we predict who will develop MS? And this is the area under the curve. At year -3, we achieve an 83% accuracy, meaning that 3 years before the first diagnosis code was given, there was an 83% accuracy in the prediction. We did the same with Parkinson's disease, and I'll show you here the explainability part of it. So the mechanism was exactly the same. We identified the cohort of about 5,000 patients with Parkinson's with multiple diagnosis codes. we went back in time, we selected our epochs or windows, and then we compare them with a much, much larger number of controls. And here's the explanatory part. So these are certain concepts in the network and their importance compared to a control population as time to diagnosis approaches. So if we focus on the Parkinson's disease node itself, the information flowing through that node becomes more and more important and more different in individuals who will develop Parkinson's than individuals who will not. And this is the p-value. The y-axis is the p-value of that difference. And as you can see, as time to diagnosis approaches, then this node becomes more and more important for the algorithm. Then other nodes that are related to Parkinson's, such as gait apraxia or tremor, also have the same trend. In contrast, terms that have nothing to do with the disease are not any more important in individuals who will develop Parkinson's versus individuals who will not. And when we do the, again, when we train the machine learning model, we see that at year -5, we have a 72% area under the curve, which is not ideal, but then it improves as time to diagnosis up to 77%. And again, this, remember, it's only using the structure portion, so we're not using the nodes yet. That's next. But then, of course, the gold standard validation is what happens if we give exactly the same information to a clinician and ask the clinician to split and to tell us which ones are going to be the patients. So we did this with the clinician specialized in Parkinson's, and the clinician did not do better than the model. In fact, the model did a lot better. And again, this is not fair because the clinician probably has more information that we're not able to embed at this time, but still, given the same information, then the clinician was almost 50-50. So we haven't talked at all about ChatGPT yet, and can we use ChatGPT or those large language models to inform our decision making? Well, there's a catch to those models. They're great in terms of generating text, but they're not very good at telling you why they know what they want, what they know. And there's no way that these models can tell you what they know and how they know it because it's a black box. So what we developed in the lab is a method called KG-RAG, which stands for Retrieval Augmented Generation based on a knowledge graph, which essentially takes the user prompt, extracts the words, the concepts that are understood by the graph, in this case, genes, MS, and so on, and then it brings the context of those from the graph and infuses that back into the model, and then it produces the output. In that way, we're essentially teaching these models biomedicine, which without this infusion, they cannot provide an accurate answer. So I'll end with just a quick demonstration of how this model works. We created a chat bot, if you wish, that can interact with the user and actually does this in real time. So this has audio, but it's not coming out. But imagine that there's audio. There you go. So it's asking about the genes associated with phenylketonuria. Please give a short answer.
The genes associated with phenylketonuria are PAH, SPR, QDPR, PTS, and PCBD1.
So it tells the genes and tells you where they come from.
HPO.
And then continue, and it keeps the memory of what you're talking about.
The gene PAH encodes the protein phenylalanine 4-hydroxylase.
Has a little bit of an accent.
Provenance uniprot.
So it tells you where it knows, and then
yes,
you can
the protein
keep asking,
encoded by the gene PAH,
navigating the graph.
Phenylalanine 4-hydroxylase, PAH, is an enzyme. Provenance uniprot.
So all of these ChatGPT does not know. You can try it and it won't tell you. It's now
the enzyme phenylalanine 4-hydroxylase, PAH, catalyzes the following reactions. 1. Reaction.
I won't make you listen to all four, but that's the idea. And we're implementing this in a UCSF so far closed shop implementation of ChatGPT called Versa, and this is going to be tested now internally within the UCSF community. But the idea is to then see how we can help clinicians to better inform their answers when the answer is really technical and it needs an explanation. So I will close with this, and I will like to say that the challenge of modern medicine really is to go from something like this, where we really don't know what the whole is, to something like this, where we can be explanatory and predictive. So I would like to thank my multiple collaborators and our funding agencies, and thank you so much for your attention.
Sergio, for this fascinating view, spoke the power and beauty of AI. Are there questions to Sergio? Yes, please.
Hi, Sergio. This is really nice and impressive. I've actually a lot of questions. I will not ask all of them now. But first question is, how is this spoke being updated? Because of course the system is as good as the data, and they are really nice, but also not complete and not always right. Maybe that's the first question. And the other question is this usage to also develop prediction models. How does that work? The data that you enter, or you have it, of course, in your institute from the patients, but does that become part of the system, like with many of these training models? And also, is this also open to external users, and how might that work?
The first question, how do we update it? And you're right, that's a challenge. The minute you download a database, is old. So what we do is we write scripts to get the databases downloaded periodically every week, and then the graph is assembled every Sunday. So the data is at the most a week old. So the scripts run on a schedule. On Monday, certain databases, and Tuesday another databases. So by Sunday, we rebuild the whole graph. In terms of the graph doesn't have any patient information. It's general, established, almost textbook information, if you wish. The patients are embedded transiently, so that we can create those vectors that we can use then to train the models. And the system is open in the sense that you can go tomorrow to their website and then you can navigate it. You can create visualizations, you can navigate, you can download up until 10,000 nodes, large networks, and so on. So, and there's also an API, an application programming interface, that if users want, they can interact via computation directly.
Yes, please.
I have a question about, you say that ChatGPT can't reply to these questions, but what about an LLM model fine-tuned on medical data, for example, like MedGemini by Google or something like this?
Well, yes. You can, not everyone can fine-tune a model, and so only Google or Facebook and OpenAI can. This is an extremely intensive process that requires multiple GPUs of the likes that no academic institution has. And there hasn't been yet there are several models that have been fine-tuned for like PubBird and so on, but they are not fine-tuned with primary data from these databases. So the training data that these models use is from the corpus of publicly available data from web pages mostly. So these models are based on text mostly, and they're great at predicting the next word. But when it comes to downloading tables from papers or databases from the string database or the human proteome atlas and so on, these models are not performing well.
Final question, please.
Another question about, for example, clinicians that will use this tool. If they rely too much on this kind of technology, could be some negative implications?
Yeah, well, of course, I'm not implying that these methods will replace the clinician. I always equate this to a modern airplane. Modern airplanes can take off, navigate, and land without human assistance, yet there's always two pilots in the cockpit at all times. So I think it will be a co-pilot, if you wish. It will enhance and help in the decision-making. It won't replace clinicians.
Final question, short, please.
Yes. The question is, in this huge representation, really fantastic work of knowledge, how do you decide that a link is accepted by the scientific community or not? Because as far as I know, the link between a gene and a pathology is can be debated.
Well, that's a good point, and we spend a lot of time not only within our group but also bringing in external experts for every database that we add. So databases make the hard work of curating data, and in the case of genetic, for example, we use the GWAS catalog as one of the genetic databases. The GWAS catalog only reports associations that are above a certain p-value, that are with a minimum of certain number of patients involved in the group. So we take a lot of time and effort into validating that or vetting that the database we're going to use is scientifically accepted and it's experimentally based. So there's no prediction, there's no literature based, no NLP or human imagination into the relationships. Every relationship in the graph is a semantically defined relationship that has an experiment behind it.
Thank you so much again, Sergio, for your excellent presentation. We now break for coffee.
Okay. Good afternoon. Welcome back. So this is the session number 8, and is dedicated to secondary prevention. It's very important topics, and I think it's very important that is a topic that will be treated at the end of this symposium. So we have to consider that in the last 2 decades, we have the opportunity to recognize some pathological pathway that in some way we consider specifically related disease pathway. And so let's open up the opportunity to us to consider prevention, and especially a secondary prevention. That is very important, because now we call this phase as a preclinical phase, but probably has been demonstrated that the diseases, neurodegenerative diseases, are already started. So I would like to invite the first speaker. We will have a 4th speaker, and the first speaker is Professor Maria Pia Amato. She is Director of a Neurological Rehabilitation Unit, responsible for the Multiple Sclerosis Center at Careggi University Hospital, but a full professor of neurology in Florence. And also she has been PI phase 2 and 3 clinical trial on MS, and developed, she participated at the acronyms EAN guidelines for MS, and participated in the development of the first European acronyms, EAN. So I give the floor to Maria Pia, please.
Thank you very much for your kind introduction. Thanks to Professor Comi, thanks to Fondazione Prada for inviting me. It's really a pleasure and a privilege to be here, having to discuss the identification and treatment of prodromal multiple sclerosis. So our understanding of prodromal MS is by far less advanced than in other neurological diseases such as Parkinson disease or Alzheimer's disease, where we have better biomarkers, better markets. So it's still a field of research in its infancy, but I'm trying to recapitulate some preliminary findings, but I would say promising findings. These are my disclosures. And of course, multiple sclerosis is the most frequent and important demyelinating disease of the central nervous system, affecting young adults, particularly females, and it's a leading cause of disability in this class of age, and it is estimated that approximately 3 million people live with the disease worldwide. So it's a multifactorial disease deriving from a complex interaction between genes and other extragenetic factors at large, involving infectious agents and in particular EBV. We heard this morning the importance of this virus, and other environmental lifestyle factors, potentially modifiable factors such as smoking, low levels of vitamin D, obesity, and others that are under investigation. So the existence of a latency phase where the exposure to these different environmental factors during different periods of life, long before the clinical onset of the disease, already suggests the possibility of a prodromal phase of multiple sclerosis. And I'm going to review the salient evidence in the field. This is a well-known and often cited paper published by Alberto Ascherio and his group, already discussed this morning, showing that the EBV seroconversion is associated with a very high hazard ratio for the clinical onset of the disease, and in a subsample, EBV infection also precedes the elevation of neurofilament light levels in the serum. There is also a mounting epidemiological evidence. This is the first study in Canada combining administrative data with MS clinical data on what you can see, very large population of MS cases and matched controls from the general population, basically showing that an increased use of healthcare before the first attack, the first clinical event of MS, in cases compared to controls between 5 years and 1 year before the clinical onset of the disease. This was in terms of hospitalization rates, physician visits, prescription of medications. Similar results also from this UK study showing again increased healthcare usage 10 years before MS diagnosis and based on primary care data. And this study focused on the reasons for physician visits 5 years before the clinical onset of the disease. These were very specific symptoms such as fatigue, sleep disorders, anemia, and pain. And consistent findings, this is again the Canadian group, were also shown in pediatric MS cases compared to controls, where healthcare usage was increased 1 year before the clinical onset of the disease, and particularly psychiatric, physiatric, and ophthalmologic visits. Interestingly enough, again in this Canadian study, mood and anxiety disorder were seen more frequently in the mothers of children with multiple sclerosis already before the diagnosis and the first clinical event in the child, indirectly suggesting the existence of a prodromal phase in the child. This is another well-known nested case control study performed in Norway, focusing on young men undergoing conscription examination and cognitive examination, and the data were linked with the MS registry and for a nested case control study, and basically the result showed that decreased cognitive performance preceded by 2 years the onset of relapsing remitting MS, and even by 20 years the onset of primary progressive disease. So in this review, collectively, the clinical profile of MS prodromes shows a high burden of mental disturbances, psychiatric and cognitive problems, fatigue, but also anemia, headache, pain, and bowel and bladder and sleep disorders. So we can say a mixture of really specific symptoms, but also some symptoms pertaining to the disease. So we can hypothesize that at least a proportion of patients with prodromal MS are in fact already MS cases with a missed diagnosis. And of course, due to this specificity, we need to identify and validate biomarkers. Probably the most compelling biological evidence of a prodromal phase of the disease is, again, this study published by the group of Alberto Ascherio, based on the serum repository of US military personnel, showing in cases compared to controls increased levels of neurofilament light in the serum at a median of 6 years before the first clinical event, with a brisk increase at a median of 1 year before the onset, the clinical onset of the disease. Very promising also this very recently published study by this group of authors, and Professor Baranzini is among them, that pointed out an autoantibody signature that can predict the initiation of the disease. The study is based on an incident cohort that was assembled during the Gulf War era in the US military personnel, and the results were replicated in an independent cohort. In brief, the results showed that the authors were able to find a subsample of patients that exhibited a stable antibody autoantibody profile, longitudinally stable and consistent before, during, and after the clinical onset of the disease. The antibodies were directed against a common motif that shared a number of similarities with many human pathogens, including Epstein-Barr virus, not only, and there was also an elevation of the serum levels of neurofilament light. So this can represent the first step towards further immunological characterization of this subset of patients, and also probably a first step towards the characterization, the identification of immunological biomarker that is antigen specific. Lastly, this is a very recent paper focusing on monozygotic twins. One of the twin is affected by the disease, the other one is not clinically affected but shows signs of what the authors call subclinical inflammation, and in the unaffected co-twin the authors found signs of metabolic and immunological dysregulation of CD8 T cells that were also found in the affected twin and in an independent sample of MS patients. So there are, of course, specific groups that can be particularly informative. I mentioned the monozygotic twins, but also first-degree relatives that have higher risk to develop the disease, and pediatric onset MS, where the biological onset is deemed to be closer to the clinical onset compared with the adults. And I will focus briefly on this radiologically isolated syndrome, group of subjects with incidental MRI findings highly suggestive of MS, but without typical signs or symptoms of the disease. These are the last criteria. It's a rare condition, but very informative. It can be very informative one. There is mounting evidence that risk subjects compared with healthy controls can exhibit or not a number of prodromal symptoms I have just discussed, and using digital tools they can exhibit very subtle motor, visual, and cognitive impairments. There is also rather extensive evidence that they can have altered biomarkers, such as increased levels of neurofilament light, apparently with a dose-dependent effect in relationship with the risk to develop, to convert to the clinical disease, OCT changes, and a number of neuroimaging findings. This is an old study, probably the first one, where we found subtle cognitive changes in these risk subjects that were associated with decreased normalized brain volumes and particularly neocortical volumes, and the decrease was similar to what we saw in clinically isolated syndromes and patients with established relapsing remitting disease. More recently, the volume of the choroid plexus was found to be increased in pre-symptomatic multiple sclerosis and also in the earliest stages of the disease. These are clinically isolated syndrome with optic neuritis, and also in pediatric MS, particularly in females. A few risk subjects also exhibit some biomarkers, imaging markers, that are highly specific for the disease, such as these slowly expanding lesions that are literally lesions that increase their volume over time, and this central vein sign, so the location of the lesions around a small vein. In the large international cohort of risk subjects of the International Risk Consortium, the rate of conversion, of clinical conversion over a 10-year follow-up was 51%. And the risk factors for conversion were younger age, the presence of oligoclonal bands in the CSF, the presence of infratentorial lesions and spinal cord lesions at baseline MRI. 11% of the subjects converted to primary progressive MS over the follow-up period, and these were in particular more men with older age and more spinal cord lesions compared to the subjects who converted to relapsing disease. So the publication of these two phase 3 trials on risk patients marked a first step towards preventive medicine in multiple sclerosis, and in particular secondary prevention, but also raised a number of questions and ongoing debate about who should be treated, which are the threshold for intervention, when during the preclinical stage, where general neurologists or most probably highly specialized centers, and how just counseling about healthy lifestyle, vitamin D supplementation or drugs, which drugs. However, the ARISE trial found that in the dimethyl fumarate treated group compared to the placebo, there was an 80% risk reduction to convert to the first clinical event, and a 62% reduction of new or enlarging T2 lesions at MRI. Similarly, the TERIS trial with teriflunomide showed in the treated group a reduction of 72% of the risk to convert to clinical disease. However, secondary MRI and PRO's endpoint were not met. Due to the small sample size in both trials, no risk stratification was, however, possible. The inclusion-exclusion criteria and the primary and secondary endpoints are quite similar in the two trials. However, you can observe that in the ARISE trial, mean age is higher. Also, the disability level is higher, from ranging from a score on the EDSS of 0 to 3. So probably this sample also included the patients who were in a more advanced phases of the disease, not the pure risk. And this is the CELLO trial, a phase that is ongoing, a phase 4 multicenter randomized double-blind placebo-controlled study with using ocrelizumab, 3 courses of ocrelizumab. And notably, the trial included a screening program of the first-degree relatives of patients in order to boost the recruitment that was rather slow and problematic in the previous trial and included a pre-planned analysis of a number of CSF, blood, and other biomarkers, and the follow-up is going to be up to 3 years. So this is the current view of multiple sclerosis as a continuum from the at-risk phase to the biological onset of the disease to prodromal stage to eventually the clinically manifest disease. And we have a long way ahead. This is a suggested roadmap for research into the prodromes of the disease. I am aware this seems rather simplistic or perhaps rather complex compared to the elegant algorithm based on artificial intelligence that Professor Baranzini has discussed. Anyway, it starts with the pre-test probability of multiple sclerosis based on prevalence rates according to age, sex, geographical area, characterization of the MS prodromes, and in particular identification and validation of biomarkers focusing on informative population, then quantification of the relationship between prodromal markers and the probability of the disease, and eventually development and validation of criteria for prodromal MS to be used in research similarly to what has been done for Parkinson's disease. With this was my last slide, and I thank you very much for your attention.
Professor Amato has to catch a train, otherwise she will be staying in Milan. And so I suggested that we can have the question right now. Please, Professor Comi.
Thank you very much, Maria Pia. If you like, a quite simple question. Do you think that if we combine together the different type of factors that predict the risk of MS, we may really be in a condition to have the possibility to organize a trial in a very, very early prodromal phase of the disease? So we have enough data to combine together so that we have enough large risk that we can start a trial.
Thank you very much for your question that focuses on the critically important point here, that is to stratify the risk and to be able to identify those risk subjects that are at the highest risk to convert in relatively short time. And I do think that we have enough information from international data sets that have been collected with relatively long follow-up to effectively stratify the risk, and based on this risk stratification plan and design a trial for these subjects. But so I think this is feasible. The problem is how to extend these results to the general community of neurology, of neurologists, because I wouldn't be surprised if whatever drugs would work in this population. So I wouldn't be surprised of the opposite. So here our issue is how to communicate the results, how to extend this possibility that is a real possibility of secondary prevention to the general neurological community.
Maria Pia, would you, how would you assess the duration of a preclinical phase of MS? I mean, do you have any information on how long it may last?
So I don't know. The true answer is that nobody knows which is the duration of the prodromal phase. In the epidemiological study, it can range from 5 to 20 years. So so I think that with the current and available information, it's not possible to establish an exact duration. Even for risk subjects, you don't know for how long they have these lesions in their brains or spinal cord.
A question over there, please.
Thank you, Professor Amato. René Carvajal from SEMCAT, Barcelona. What do you think about that? We usually consider the index date or the date of the onset of disease based on databases of population studies and according to ICD codes. Maybe we really are assessed not prodromal phase instead of maybe diagnosis delay. I don't know, I may be concerned about that because maybe this is an issue.
This is another really critically important question. I just briefly, quickly mentioned this, but if we consider the pattern, the profile of the prodromes, some of these called prodromes can be in fact, the first symptom of the disease, for instance, sphincteric problems. So also, if you look at the mean age at onset in the epidemiological study that I shown, the mean age tends to be higher than usual, around 45 years. Which generates the hypothesis that at least a proportion of these subjects are not in the prodromal phase, they are MS, in fact, with a delayed diagnosis, and with a missed diagnosis or a delayed diagnosis. And I have also to say in this regard that in the current revision of the diagnostic criteria for multiple sclerosis, a few risk subjects with specific characteristics are considered, in fact, already multiple sclerosis.
Other question? Okay, thank you so much. So I would like to introduce Jill Livingston, the professor of psychiatry of older people in University College London, and Professor Livingston leads the Lancet Standing Commission that just recently published the report on prevention, intervention, and care, and so I am happy to introduce her.
Thank you very much for inviting me. It's been an amazing experience being here and having such a wide array of knowledge and experts in different subjects. I'm not sure when secondary prevention of dementia is. Is it mild cognitive impairment, or is it as soon as anyone has any amyloid in their brain? Is it decades before? And I don't think anyone is sure when secondary prevention. We're pretty sure when people have dementia, but I think that we're very unsure when secondary prevention takes place. So I'm just going to talk really about dementia prevention, and some of the things have been covered by other people, and I'll skate over them, but I'm trying to put it all together and think about how we can use it in prevention. So the commissions that the Lancet do have people from many different countries in the world and different disciplines, and their names are all there, are all here. So I'm not going to talk through them, but I'd like to say that some of the work here, some of the calculations and the systematic reviews were done in part or almost in whole by my colleagues Andrew Summerlad, Sergi Costa-Frida, and Nahid McAdam, but the whole group spent a lot of time working on this. I'm going to talk a bit about why I consider dementia preventable, but not in any detail, because people have talked about that, a life cohort model and how to compute it, the new information we have about risks and what it means, a little bit about cost and benefit and what we should do. So people have talked already today about a bit about dementia being preventable. There's more older people in the world as fewer people are dying at a young age, and that should mean that there will be, and does mean there will be more people with dementia. Despite that, there's been a decrease in some high-income countries in the last 20 years in US and Europe, but in some other countries, dementia has been stable or increased. And that means that dementia is potentially at least partially preventable, but it's certainly not inevitably preventable. And the mechanisms of prevention are broadly thought to be a cognitive reserve, which enables us, and which has been talked about today, but which enables us to withstand neuropathology, and a reduction in damage. Even in the US and the European countries in which dementia has gone down, it's gone down in higher income, more educated people, and unsurprisingly in men more than women. And that shows that there's still a huge amount that can be done even in the highest income countries, and much more that can be done elsewhere. So I'm just going to skate over this, but because people have talked about it, but what this graph shows is that while on average the amount of neuropathology you have is related to your cognitive function, many individuals with high cognitive function when they die have a lot of neuropathology, and other people with considerably less neuropathology can die with severe dementia. So what we have done in the Lancet Commission is we have tried to consider what risk factors, and I have to say it's a very controversial thing to do. We've taken a high bar and tried to only consider risk factors which are biologically plausible, which have consistent high quality evidence many years before dementia to avoid reverse causation, which changing the risk factor shows a change in dementia. And with that, we have calculated the population attributable fraction, which is the fraction theoretically prevented by eliminating the risk factor. And approximately, it is related to the relative risk times the prevalence of the risk factor. We used all-cause dementia, and we've used international data to look at worldwide population attributable fraction. But we know, and we have published, many of us have published about that varies between countries. So depending on the prevalence of the risk factor in that country. And in general, low-income countries have a higher population attributable fraction, so have more possibility that of reducing the number of people with dementia. International data, which we've got from systematic reviews, tends to have a bias towards high-income countries because more research goes on there, and there are more data, there are more cohort studies. We've looked at 14 risks, and we've adjusted for communality. So people are not like rats. They often have more than one problem, and in fact, problems tend to cluster. So the person who is obese is more likely to be hypertensive, is more likely to have high cholesterol, is less likely to exercise, and more likely to have diabetes. So we've adjusted for communality. And while I'm going to talk about these 14 risks, we know that there are other risks. I'm going entirely the wrong way. So this is a table which shows the population attributable fraction. I don't want you to try and read it, but it shows the population attributable fraction of all 14 risk factors with the prevalence worldwide as much as we can find it. And I'm going to talk more about this, the relative risk of each risk factor. And then what I want you to look at is that we have weighted the risk to the fact of communality, and the weighted risk is much, much less than if you just look at a risk factor by itself and don't consider what else other people might do. We've used a database with 14 risk factors to do so. And from this, we have calculated and drawn this infographic of the population attributable fraction of potentially modifiable risk factors for dementia, and looked at the time in life when they are tend to be most important. And I'd like you to notice a couple of things. They make up slightly more than 45%, suggesting that almost half of dementias are at least theoretically preventable, that and that they are hearing impairment and high cholesterol are the two biggest ones. But each of them are important, and the small numbers of percentages, but very high number of people. And most of them are in midlife, a time when we think that people are perhaps developing dementia, but rarely, unless they have a genetic predisposition, have symptoms. So I just want to talk through some of them and talk about the things that we've learned in the last few years about them. It's not a surprise to anyone that educational activity, attainment and cognitive activity appear to be somewhat protective, but it's only recently, this elegant study led by my colleague Mika Kivamaki, that we've been able to show that that high cognitive activity in midlife is also protective. So if we consider people who have little education and little cognitive activity in their job as a risk of one, then having high cognitive activity at work without education reduces the risk. And if you then have to about 80%, if you have high education and little cognitive activity at work, it reduces it to about 70%. For all of us here, who have both of these things, it reduces it to about 60%. Moving on to hearing loss, which is the biggest risk factor in midlife, we performed a new meta-analysis in the Lancet Commission, and there's been more studies on risk of dementia for people with objectively measured hearing loss, looking at them between 6 and 12 years later. And overall, we find that the relative risk of dementia is 1.37, so the people have got 40% increased risk. It's dose-related risk, so it's worse if your hearing is worse, and it is attenuated if people wear hearing aids. Both cohorts and a randomized controlled trial finds that wearing hearing aid reduces dementia risk in people who are at risk. Studies of people with mild cognitive impairment show that people who have hearing loss and mild cognitive impairment who wear hearing aids have an average of 4 years till they develop dementia, but those who have hearing loss and without hearing aids have an average of 2 years. Moving on to depression. For a long time, I think we were unclear whether people who had depression were more likely to get dementia or whether people who had dementia were more likely to be depressed. And the answer is both. But this is a meta-analysis done for the Commission, finding that midlife depression increases risk for dementia, and it increases a considerable amount, and that we're over twice as likely to get dementia 10 years later. But there's also been a UK Biobank cohort study which finds that treatment with antidepressants or psychotherapy or spontaneous remission of depression decreases the risk of dementia, and those who treated had only 70% risk of dementia versus the untreated people. Hypertension is one of the few, so hearing and hypertension both have randomized controlled trial data. Hypertension trials are always abandoned as the control group always does very much worse. But putting them all together in individual participant data, the risk of dementia is decreased in the treatment group in in several studies, but one Cochrane review, so in several meta-analyses, one Cochrane review did not find a significant difference, but they mainly followed up people 1 year later, and it probably takes longer than that for the effect to come through. There's been a lack of direct comparisons of antihypertensives, but a network analysis found angiotensin II receptor blockers and calcium channel blockers were associated with decreased dementia risk versus other antihypertensive. Smoking is a sort of thing that all doctors reflexly say, bad for you to smoke. And it obviously is. What is quite remarkable is a couple of studies that show that even over 2 or 3 years of quitting, smokers have risk goes down to the control group. And if they don't have dementia then, they reduce their risk. So there's no increase in former smokers. I'm not going to talk much about traumatic brain injury after that remarkable talk we had earlier, but apart from sport, it is common in low and middle income countries from road traffic accidents, which is also potentially very preventable with seat belts and safer cars, and in high income countries from falls or violence. And again, things cluster, these tend to be related to alcohol. As I said, there's much more evidence of link with some sports and head contact, and what sport you play makes a difference. People have also talked about physical inactivity, exercise and fitness. And interestingly, and a good thing to be able to tell our patients, is that reduction in risk is greatest when moving from being extremely sedentary to some physical activity. You don't need to run an ultramarathon. Any exercise at any age seems to be helpful for cognition in cohorts, but the randomized controlled trials have not really found very much benefit. And I think we don't really know about the duration of exercise that you need. Trouble about randomized controlled trials is by their nature, they tend to be quite small, quite short. There's diabetes with new evidence showing that midlife diabetes is a particular risk rather than late life diabetes. That may just be a time thing. There's not been that much time, but it may be that things change just as they do with hypertension and obesity. And there's randomized controlled trial evidence that people who with diabetes given GLP-1 receptor agonists, which we were talking about last night at dinner, are the new wonder drugs, but they're associated with a decreased dementia risk for people with diabetes, and we don't know about people without diabetes. It's common, cholesterol is common to have high cholesterol, and there's increased evidence that high LDL cholesterol is in midlife as a risk for dementia. That wasn't terribly clear before, though some studies found it, and it seems to make a great deal of sense. But now we have more evidence that statins used in midlife decrease the risk of both dementia in general and Alzheimer's disease. And there's a clear opportunity to prevent dementia with statins for high cholesterol. Obesity, studies adjusted for hypertension and diabetes still find increased risk of dementia. Intentional weight loss associated with cognitive improvement. If you look, and we have been looking at trials to try and decrease dementia, and the things that the trials are really quite bad at doing is increasing weight loss. It's something that's very difficult to change. Andrew talked this morning about social isolation. There is further evidence that less frequent social contact is associated with dementia, and that is many decades before, but it's really very difficult to have studies about increasing social contact. And alcohol, again, nobody's surprised that alcohol is related to dementia. I think we're maybe surprised that the level of drinking doesn't have to be that high. So 12 US units or 21 UK, I'm sorry, I don't know what the Italian units are, related to increased risk. Many studies say that people who don't drink are also increased risk, but then if you follow up the people who don't drink, quite a lot of them die of alcohol-related diseases. So they're quite often ex-drinkers, ex-heavy drinkers. And again, there's more evidence from Korea, South Korea, that reducing alcohol to moderate level reduces the risk very rapidly. Air pollution has also been talked about in more detail, but there's a lot of evidence now that changing air pollution decreases the risk of cognitive impairment, cognitive decline, and dementia. And a new risk from our Lancet Commission is untreated visual loss. It's common to have untreated visual loss around the globe, although less common in high-income countries where people frequently have their eyesight tested, and wearing glasses is usually not particularly stigmatized. But it's still really quite common. And untreated vision loss of any type is associated with increased dementia risk. Untreated visual loss is higher in minority groups and in low and middle income countries. And again, it's a dose related. People who've had cataract extraction are no longer at increased risk. And again, there's clear opportunity for dementia prevention by treating vision loss. So I've sped through, because I don't have a huge amount of time, about risk factors. And the question is, changing risk factors, does it change the outcomes? And the answer appears to be yes. If we look at longitudinal studies, there's less evidence because it's difficult within randomized controlled trials. But use of hearing aids decreases risk in the group who are likely to develop dementia. But in the United States, Frank Lynn's trial, one group were healthy volunteers, and they really didn't deteriorate over the time of the trial, and it didn't make any effect. But in another slightly older group who did deteriorate, the hearing aids made a great deal of difference. When air pollution reduces, cognition is better than in comparator areas. Now, we can't individually necessarily do much about air pollution, but governments can do a great deal about it. And in Britain, there's been a lot of debate about decreasing air pollution and having areas where highly polluting vehicles can't go on. I think the government is going to, we now have some areas of low emission zones, they're called, and I think the government is partly because of dementia going to roll that out. Treating hypertension gives you less dementia. People without much education are relatively protected by jobs with high cognitive demands, but probably if you just do Sudoku, it probably just makes you good at doing Sudoku rather than anything else. Stopping smoking reduces risks. Reducing alcohol changes risk. Treating vision impairment reduces risk. Treating high cholesterol in midlife helps. Treating depression helps. It helps with diabetes, keeping under not too tight control, but control helps. It's important to make changes in people who are at risk, who are the people who are often not in trials and don't see doctors, and they need to be targeted. So on the same day that we put out the Lancet Commission, we also published a paper in Lancet Healthy Longevity in which we modelled the cost savings in England. Now, many of you are probably not very interested in England, but when you do cost health economic analysis, you have to put it in a place because things cost in a particular place. And it's probably true in other things. So we used interventions which had changed what happened in the population. So remarkably for England, changing the formulation of food to decrease salt so that blood pressure drops is projected to reduce the dementia cost by 2.4 billion saving. Reducing sugar, which will reduce weight, and that's not asking individuals, but reducing it in formulation of food is expected to, in models, to decrease by 1 billion. And less money, but still making a difference, is having minimum price for units of alcohol low emission zones, and increasing smoking price. And in a very long term, I don't think I could say it's secondary prevention, compulsory helmets for cycling for children aged 5 to 18 is also expected to save money in the long term. So dementia is the most feared disease. It affects people with dementia who don't want to get it, their families who look after them and are very stressed, and the economy. Preventing dementia is what we all want and is what and should be economically possible. So overall, what does it mean? New hopeful evidence about dementia prevention, and we're getting it all the time. Almost half of worldwide dementias are potentially preventable from tackling 14 risk factors, and we know there are more. The evidence is unclear now about some of them. There's much, much more evidence that interventions make a difference and people continue. There's potential for risk reduction at scale through policy changes, and these changes are often cost-saving and help individuals avoid dementia and give them more illness-free years, compacting morbidity for those that do develop dementia, and some people will continue to develop dementia. It gives what individual wants with a healthy life and a compaction of morbidity. And the measure I am with this conference. Let's be ambitious about prevention for our patients, for academically, and with our governments. So thank you very much for your attention. That's UCL in the win.
Thank you. We move to the question at the end of the two remaining lectures. So now we have a professor of neurology, Ronald Postuma. He is a professor of neurology at the Montreal Neurological Institute at McGill University in Montreal, and he focuses his interest in Parkinson's disease, especially the early detection of Parkinson, diagnosis and treatment, and sleep disorder, including REM sleep behavior disorder. And so he will talk about how to prevent Parkinson. When does Parkinson's disease start? Please.
Thank you. And thank you to the organizers for having me here. So I'm going to give a bit of a whirlwind tour. I'm talking here about secondary prevention, prevention of progression of people who already have some Parkinson's pathology, unlike the first talk. And I want to just give a couple of essential basics of Parkinson's, just understand why some things will work and some things won't. I'm going to give I know this is a mixed audience, so I'm going to give a very, very quick summary of secondary prevention, things that are ongoing right now, and illustrate the potential possible pathways that are out there. And then we'll really focus on the understanding the early stages of Parkinson's disease. And finally, I'll undercut it all by talking about how biological diagnoses may change everything. You can't prevent a disease if you don't understand it. And if we were in the 1970s or the 80s or maybe the 90s, if I was talking about Parkinson's, I'd be talking about this. This is the substantia nigra. This is the absence of substantia nigra. It's a dopaminergic lesion. This is what Parkinson's is, tremor, rigidity, etc. However, it's become entirely clear that that is actually false completely. Everything on this list is experienced at some point by about at least half of patients with Parkinson's disease. So there are motor abnormalities unrelated to the dopaminergic system directly: drooling, choking, falls and freezing, depression, anxiety, some visual changes, a whole host of autonomic abnormalities, sleep disorders, I'll talk about RBD in particular, psychiatric complications like hallucinations and paranoia, and ultimately dementia are seen in most patients with Parkinson's disease in some combination of these. And this is really the major unmet need in Parkinson's disease. We can do a good job of taking care of your dopamine system. It's the everything else that's the problem. And this has an underlying pathological basis. I'll come back to this slide, but just everything that you see in color are areas that are affected by Parkinson's type synucleinopathy in these various staging systems. And so we think Parkinson's is a synucleinopathy, and just illustrating that it's a relatively diffuse synucleinopathy. So with that, I'll just change and let me give you a bit of an update into what's happening right now in the field of Parkinson's disease. Again, this is not a pipeline talk. They tend to be 45 minutes. I'm just going to tell you a bit about what's happening. So there's a few answers that we can give in terms of secondary prevention. I'm talking about neuroprotection, so slowing down the underlying neurodegenerative process. Do the current treatments for Parkinson's disease, for example, levodopa, make a difference in neuroprotection? This is actually 100% clear now. As near as we can tell, there is no difference that is measurable. And these are studies on patients that are being followed for 7, 8, 10 years, randomized and follow-ups. Stem cells, like an implanting stem cell to replace a dopamine, well, if you saw the last slide, you'd realize the problem. We can't put stem cells all over your brain. They have to be injected. So this sort of thing is really not, I think, a neuroprotective strategy. Now, in terms of the lifestyle and non-pharmacologic things, there are certainly lots of potential things we could do. It should be noted that, in general, the lifestyle factors are not very strong. We're looking at relative risk 1.5, 1.8, 2, so that does limit a bit the scope. The one that has the most advance is probably exercise. We should note, of course, that we're already telling our patients to exercise, but there is an ongoing study of exercise in prevention of Parkinson's disease and REM-sleep behavior disorders. There's no question in Parkinson's disease that are clear benefits. We don't really know why. Is it truly neuroprotective, or are people just better asymptomatically? And so, yes, I think it's certainly promising. We do have to have some realistic expectations. I mean, I've had many people who were marathon runners telling me about the first sign of their Parkinson's disease being the intorted foot on kilometer 12. Healthy diet, microbiome, these, we don't really have any idea yet if those are being used. In terms of the pharmacology, pharmacologic targets, so the most obvious target, of course, is synuclein. Parkinson's is a synucleinopathy. It unifies at least 90% of Parkinson's disease. It also catches dementia with Lewy bodies. Probably the most advanced right now would be the antibodies, and the one that is the most advanced is prasinezumab, which has a single study which showed a possible readout on a secondary outcome, but it was a big secondary outcome, is the motor exam of Parkinson's disease. And this is now being confirmed in the study called Padova. Pasadena was the first. And so the primary outcome was negative, but the secondary looked kind of positive, so we'll see. There are many aggregation inhibitors, synuclein blockers, other ways to reduce or prevent synuclein from aggregating. They're in phase 2. Genes are a wonderful target because they really get directly to the cause. If you have a gene that causes Parkinson's disease, targeting that gene should help you. By far, the most important gene in Parkinson's disease numerically is GBA, glucocerebrosidase A. It's a risk factor found in 10% of Parkinson's patients, generally a risk factor with sort of relative risk of around 5 or so. And it's probably a loss of function, and there's trials of GCase activators. There was a failed trial of substrate reduction therapy. LRRK2 is the other big common gene. It's in special populations, so it's less worldwide, but it's a kinase and it's druggable, so there are ongoing LRRK2 trials. This one would be of interest to a lot of people here because you'll recognize the echoes, the GLP-1. There was a positive trial in exenatide at phase 2, a positive trial of lisexenatide at phase 2 as well, and there are talk about Ozempic, but only these two are the only ones that cross the blood-brain barrier. They seem to possibly work in young Parkinson's disease specifically, and I just found out last night, unfortunately, that the phase 3 of exenatide looks like it has failed. We have no details on that, so we don't really know where this is going. We think the GLP-1 agonist may be working on anti-inflammatory. Inflammation is also common in Parkinson's disease. There is one prodromal trial that will start with adalimumab, anti-TNF, in the next little while in prodromal disease. Okay, so that's the pipeline. Let's go back to those things that I showed you earlier. So these, I'm sure you're aware of these. This is the Braak staging system of Parkinson's disease. This is another one that combines PD and DLB. The essential point of all of these, and they actually have been found to be mostly true, is that Parkinson's starts in the anterior olfactory nucleus, in the dorsal motor nucleus of the vagus, and it turns out, way over here, in the peripheral nervous system as well, and does a semi-geographical march through the brainstem up into stage 3 in the Braak system anyway, starting to affect the substantia nigra. At only stages 4, 5, and 6 is when you show up in a doctor's office with Parkinson's disease, by which point it's already in the cortex. That doesn't account very well for dementia with Lewy bodies, so there are other systems that posit that it goes to the anterior olfactory bulb and goes either down into the brainstem or around into the cortex. One is PD, one is DLB. They're overlapping conditions. There's some controversy over these, but the general principle, I think, has been relatively well established. So non-motor features should, in fact, be the first features of Parkinson's disease. So when should we be using our secondary prevention? Well, we understand that any treatment for secondary prevention, neuroprotective treatments, are not going to cure the disease and stop it in its tracks. They're going to slow it down. And so it would be, of course, very useful to apply these in mid-early stages of Parkinson's disease. But if we could apply these same slowing down treatments into the prodromal and even the preclinical stages of diseases, we could prevent them from ever happening. And this is obvious to everyone in the world. And so there's been an immense amount of effort to try and figure out what are the markers. I don't have time to go through these, so I'm not going to be able to talk about color vision, somnolence, constipation, erectile dysfunction, depression, anxiety, subtle motor loss, cognition, or any imaging. But I've just selected out a few of the most important and most interesting and newest markers of prodromal disease. In other words, identifications of the early processes of Parkinson's disease. This is one of the first that was ever known. This is olfaction. There are now, I think it's 12, there's probably more now, prospective studies demonstrating that if you lose your sense of smell, you will have an increased risk of developing Parkinson's or being diagnosed with Parkinson's in the future. And the relative risks are quite consistent across studies, and they're quite high, so sort of 4 to 5-fold increased risks. And that's higher than most of the clinical markers. It is a very good predictor of dementia, particularly dementia with Lewy bodies, and to a lesser extent, Alzheimer's disease. The lead time is extremely long. I'll show you that a little bit later. And what's really promising is that it's very easy to test. You can do it on a population scale with these things, which you can use thousands of times, or you can put these things in the mail and test people's sense of smell. This is one that's very new, so you may not be aware of it. This is primary autonomic failure. So we've known for forever that constipation and urinary dysfunction and erectile dysfunction can predict Parkinson's disease. But if you're in a laboratory with orthostatic hypotension that's been documented to be neurogenic, that's a much higher level of specificity. This was published in Brain only this year. Just pay attention to the bottom line here. And this is the Kaplan-Meier curve of people in the lab with primary autonomic failure. And at a rate of 12% per year, overall on 4-year follow-up, about 33% of developing disease. They convert to a disease. If you're a lumper and not a splitter, they convert to one disease, and that is neurodegenerative synucleinopathy. 20 of them got Parkinson's disease, 17% got DLB. Again, all of the predictors of Parkinson's predict DLB almost equally, and 11 got MSA, the 3 neurodegenerative synucleinopathies, and nothing else yet in any of these studies. This is what most people would be anticipating. This is a gentleman who is dreaming. So as he's dreaming, he's out for a walk with his wife. Someone is threatening him and his wife, and he's defending him and his wife from this attacker. Now, if you would have this dream, you would not be able to do this. You would not be able to do any movements at all. You might be able to twitch and maybe make a little bit of a moan. This is REM sleep behavior disorder. So this is present in 1% of the population, well established now, 1% of the population, so not rare at all. The people who present to doctors' offices tend to be men in the ages of 50 to 70, but when we look on population basis, it's much more equal between men and women, a little bit more men than women probably in reality, but not much, probably about the same prevalence as Parkinson's disease sex distributions. And this is one of the more dramatic curves you get in prodromal disease. So this is a 1,300 patient study. This is a multi-center study. Just everyone's combining their own data together. 24 centers from the International REM sleep behavior disorder study group. And what you can see is the same sort of Kaplan-Meier, and this is disease-free for survival now going out to about 15 years. And at about a 7% rate per year, it's a bit of an underestimate. It's probably a little bit higher than that if you follow them carefully. But over 15 years, we get down to essentially no people who have not developed a neurodegenerative syndrome. Well, I guess you could probably say 20%. 55% of them get Parkinsonism first. 45% of them get dementia first. They all tend to overlap with each other a couple of years later. And so the bottom line is that these idiopathic RBD patients essentially are in the early stages of synucleinopathy. This has led to a lot of research, and we've done a lot of prediction studies and trying to stratify and come up with neuroprotective trials, because everybody realizes, of course, that this is the ideal neuroprotective trial population. I just want to show you a few things that are a little bit newer. This is when you're watching people develop Parkinson's disease, and I've been watching them sort of personally for about 20 years, and we're following RBD patients, we're doing a systematic protocol, and then we're seeing how they look as it evolves over time. And what you can do is you can take the person who now has Parkinson's disease or dementia with Lewy bodies, and you can track backwards and sort of try to model directly what does prodromal disease look like. This is with an earlier version was in Brain. This one is submitted for publication, so you haven't seen this yet. But this essentially goes back in time. So this is, let's go here. This is the motor score. This is the motor signs of Parkinson's disease, a physician examination. And these are normal controls, age-matched controls, and these are people. And this 0 is when they're diagnosed with disease. And if you go backwards over time, you can see that there's a bit of an exponential takeoff that starts around 4 to 5 years before, and then some subtle grumbling symptoms that are probably present about 7 to 8 to 9 years before. And this is consistent across all of the motor tests. I mentioned all these non-motor tests. This is how they change. I'll call your attention first here. This is the earliest thing that we see. This is the upsit, which I showed you earlier, those little scratch and sniff pads. And what we can see is these are RBD patients testing abnormal and remaining, even 14 years before the disease, remaining clearly abnormal. And then what we start to see next would be these autonomic things, so constipation, sexual dysfunction going back around 11 to 14 years, orthostatic hypotension, similarly 11 to 14 years, color vision and cognition, then going at around 5 years before. Another striking feature is that the DLB patients and the Parkinson's patients look exactly alike on almost every single one of these measures, and we can talk about them in the discussion if you like. If you put that all together, and I'm just to walk you through this, this is where we're starting here. This is a patient who's now developed Parkinson's disease, dementia with Lewy bodies at this time. This is their future. This is, on this scale, we have the symptom severity. So whatever this measure is, the worst you can possibly get, basically, advanced Parkinson's disease and normal. And this is time. And what you can see is that, for example, around 22 years before diagnosed with Parkinson's disease or DLB, we start to lose our sense of smell, and it actually reaches 65% of the maximum gone, the highest score that you can get on this, by the time you're diagnosed with disease. Next, followed by rhamatonia loss, next followed by autonomic dysfunction, and next followed by motor and cognitive dysfunction, which do a much steeper and almost exponential takeoff. And if you think about the BRAC staging model and the Mayo staging model, this is exactly what they predicted based on autopsy. And we're seeing this. This is real data in REM-seq behavior disorder patients. I want to finish with what's the newest advance, which is really challenging our concepts of what we're even talking about when we're talking about prodromal disease, and some of the transitions toward biological definitions. So everything I showed you so far were clinical tests, but there have been 2 massive advances in diagnostic tests for Parkinson's disease and dementia with Lewy bodies, biopsies and fluid assays. So we diagnose Parkinson's currently with an autopsy, but if you could do an autopsy during life, of course, this would be a 100% way of diagnosing Parkinson's disease. Well, it turns out, I showed you earlier that there is skin abnormalities in Parkinson's disease, and you can see in this one paper published in JAMA just this year, 93%, it's a 6 biopsies, so it's a little more work, 93% of Parkinson's patients have a positive biopsy compared to only 3% of controls. You also see it in multiple system atrophy. You also see it in dementia with Lewy bodies. What's very interesting is you see it in 100% of these pure autonomic failure patients that I showed you. And this is our sample that we've done. This is a biopsy. It's seen in 82% of our REM-seq behavior disorder patients. And overall, there's no study that gets really less than 75%. And so we're seeing a biological autopsy during life in the earliest prodromal stages of disease that we can come up with. This is the other one. These are the seeding assays. And so the principle here is that we have abnormal Parkinson's patients and dementia with Lewy body patients are having this abnormal synuclein seed that is in their brain and is therefore in their cerebrospinal fluid. You add a cerebrospinal fluid sample to a tube, you mix it up, a tube full of synuclein, you mix it up, and the abnormal conformation, prion-like, if you will, aggregates the synuclein. And you can see that the synuclein starts to aggregate. This is done in CSF and has now become entirely clear that the large majority of Parkinson's patients are positive on CSF. This is probably the best study. This is a sporadic Parkinson's disease, non-familial. 93.3% were positive, and controls are around the 3 to 5% range. And you can think about what that might mean if you wish. And in our REM sleep behavior disorder study patients, we see the same proportion. We see between 70 and up to 90%. And there was one study that did REM sleep behavior disorder patients, and the concordance between skin biopsy and CSF synuclein was 100%, and they found it in 90% as well. So this has led to a challenge in the way we're thinking of what exactly we mean when we're talking about prodromal and how we might want to think about diagnosing these diseases, not really for clinics, but in research now. In clinics, we can do it all perfectly well. So two groups were thinking about this independently without knowing. It's kind of a little bit sad, actually. Started working on biological classifications of Parkinson's disease, and they published in the same issue of Lancet Neurology just this year. One of them is, I was involved in this one. This is the Synergy. It's very analogous to excuse me, to ATN, amyloid tau neuropathy. This is synuclein, neurodegeneration, and genetics, because genetics are quite important. And this is one by the Michael J. Fox Foundation, which is actually quite similar. It's synuclein, dopamine, not all degeneration, and they add a staging system, and they combine DLB and PD in neurosynuclein disease. There has been a lot of discussion about this. I don't want to get into it. It's like a lot of controversy. But I think just to share what these things share, to tell you what these things share, is that it's like where Alzheimer's is we're starting to make biological definitions of the disease. And in both of these classification systems, one of which I was an author on, there is no distinction made anymore between the prodromal and the clinical stages of Parkinson's disease. Prodromal Parkinson's disease is Parkinson's disease. It's also dementia with Lewy bodies in many cases. And maybe we should be abolishing the distinctions so that we can move earlier and earlier and recognize that these people, in fact, have Parkinson's disease and had it all along. So with that, I'll stop. So just to summarize, Parkinson's is not just a motor disease. The non-motor stuff is prevalent, and it starts first. There's lots of targets. I went through those a bit. We have many, many prodromal markers. Olfaction's amazing because how scalable it is, and autonomic loss so strong but relatively rare, and REM sleep behavior disorder, the sweet spot of both common and the strongest one that we have. The path of evolution is really becoming much clearer. We're starting to actually see patients develop Parkinson's disease. The biological diagnosis is coming. Again, this is not replacing clinical practice at all. And finally, I showed you a study of 1,300 REM sleep behavior disorder patients. We now have 5,000 REM sleep behavior disorder patients in the centers of the RBD study group. We could run 3 or 4 neuropreventative trials starting tomorrow if we could. Thank you.
Your lecture. Now we're moving on, and I call Professor Silani, that is Professor of Neurology and Director of the Department of Neuroscience and Laboratory of Neuroscience at the Institute Oxologico Italiano, and Professor at the University of Milan. He has a very deep clinical research experience in neurodegenerative disease, and today he will talk to us detecting and treating prodromal amyotrophic lateral sclerosis.
Thank you, Massimo. I'd like to thank the organizers, and specifically Giancarlo and Fondazione Prada for this invitation. My duty is try to solve the issue of how to detect and treat eventually prodromal ALS. Those are my disclosures. Actually, the ALS community is looking to what's going on in neurodegeneration, specifically in Alzheimer's disease, in Parkinson disease, as you heard before, because obviously we are impressed about the achievements, namely related to Alzheimer's disease and related to the MCI, the pre-MCI. So the ALS community somehow is questioning if there's any way to come out with a MCI or better an MMI, preclinical motor impairment. And this is a very difficult task. In other words, means understanding phenoconversion, understand the phenotransition eventually of the patients. The ALS community focused initially on pre-symptomatic mutated ALS patients, and SOD1 population represent the referral. And the rationale for that is coming from the transgenic. Actually, this is probably the transgenic that was more useful in neurodegeneration, the gene ID3A SOD1 mouse. And we contributed back in 2001 to demonstrate that the biological changes are happening early on in the transgenic, vacuolization, mitochondrial swelling. So that means that the biology, at least of the SOD1 patient, is starting early on during the course of the disease. Related to humans and related to Heiko Brack, you probably know the last part of his life, he devoted all his effort to try to define the evolution of in ALS, but actually Heiko is not providing any data related to the preclinical period of ALS. So coming back to reality, in the United States, the pre-fall studies start in Miami related to Michael Benatar that start to collect the pre-symptomatic mutated SOD1 patients, try to define all the clinical and instrumental characteristics of this population, look at, for example, MUNI, looking at cortical excitability, looking at the brain NMR, and there is a nice example on the right of magnetic resonance spectroscopy defining early on in SOD1 mutated patients the changes. And according to a long period observation, Michael Benatar has the privilege to come out with the concept of mild motor impairment that just come out in this landmark paper in 2022. And we will use this paper in order to try to define what are the possible early clinical features related to the prodromal ALS. And obviously, this is a very tough for the clinicians. The findings, which represent a departure from the spectrum of healthy physiology, should be insufficient to permit to an experienced neurologist to declare the unequivocal emergence of a clinical manifest of ALS. It should not obviously be attributable to another cause. So what we have a reduced exercise tolerance, very generalized term, cramps eventually, very mild asymptomatic focal weakness, eventually hyperreflexia, especially if new and asymmetric, scattered ongoing denervation with fibrillation of positive sharp waves in muscle restricted to a single peripheral nerve or root. Walking test, that's an option for the future. Weight loss, this is very important, we come back on that, and evolution over a varying length of time. Obviously, the fairer conversion means that the patients start from something apparent to something more clear with emergence weakness, and because he has accumulation of symptoms, signs, and eventually neurophysiological findings. ALS somehow is a surprising disease because in the early, I would say, beginning of the century, we started to define the cognitive and behavioral changes. We come out with guidelines. This is the last product, the so-called strong criteria that we defined in 2016. Actually, we know the 50% of ALS patients show mild cognitive and behavioral changes. So we can use somehow a mild cognitive impairment and a mild behavioral impairment in order to define the preclinical period of our patient according to specific neuropsychological testing. And for example, Benatar proposed a decision tree for classification of MCI, MBI in ALS that can be eventually very useful in order to define the prodromal patients. What about genetics? Again, ALS is quite peculiar in neurodegeneration because now we know that more the 4D genes are at play, are responsible for the disease, and the three most important somehow are C9, SOD1, and FAS, not just in familiar cases, but also in a large number of sporadic cases, 10% of sporadic cases. And actually, we know from those genes what are the possible pathogenetic mechanisms. And this is obviously very important in trying to come out with a therapeutical strategy. But even more surprising is the fact that 90% of the ALS patients with mutation or no mutation beside SOD1 and FAS actually have a common aggregate that is TDP43. So the whole ALS patient has a common target to be treated. This is again Michael Benatar, and this is the first series of pre-symptomatic patients that he was able to follow along the time. This is a very busy slide, but basically the message is out of 12 FOV SOD1 mutated patients, 67% have a limbic onset, 100% with lower motor neuron signs, and 67% with upper motor neuron signs. And the onset of the disease, in other words, the phenoconversion was abrupt. If you look at on the right side, you see we are talking about few months and totally unpredictable somehow. But even more interesting is the fact that all those patients were characterized by the increase in neurofilament light chain in the plasma. And the average is between 5 years to 0, basically. We will come back on that. Related to other mutation like I117 and L106V, you see few patients, few informations, and the same is related to FAS and related to C9. 3 patients are being reported. Now, each center, even in Milan, we have a large series of pre-symptomatic C9, obviously, and related to the original paper of Benatar, actually, again, there is a variability in phenoconversion related to C9. What about non-genetic biomarkers? Well, we have a neurofilament light chain is the main biomarker used in defining the preclinical situation in the mutated patients. And again, the increase of a neurofilament light chain has been described even 5 years before the onset of the disease, before phenoconversion. In 2014, we described with Petruccelli the dipeptide in the ALS patients, C9 mutated, but also in the pre-symptomatic. And we know that the dipeptide are increased long time before phenoconversion. And we don't know exactly when they start to be increased in the CSF of the C9 patients. What about modifiable factors in before phenoconversion? Obviously, this is the most attractive part. And I'm very pleased to have Professor Livingstone here, because obviously this slide is very attractive for all of us. So what I try to do, obviously I'm here to be criticized. I try to put the risk that we know and try to figure out if we can come out with something. Obviously, we are far away to know what's going on. And actually, a large part of the slide is devoted to BMI and nutritional status, and I will come back on that. I like to say that related to modifiable risk factors, I think we are quite lost because we are long list of elements like heavy metal exposure, like lead, iron, cadmium, selenium, mercury, associated with polymorphism involved in determining genetic susceptibility. Large number of reports indicate pesticide has a possibly responsible for ALS, and we describe polymorphism. PON1 polymorphism has a partially responsible susceptibility of the patients. Then magnetic fields, low frequency, but nothing conclusive. Oxidative stress, but this is nothing. And then BMI and nutritional status. Again, this is very important because apparently better survival BMI between 30 and 35, and the increase in primordial BMI predicts better result in the evolution of ALS functional rating scale. And increased BMI in early life is associated with apparently decreased incidence of ALS. This is the last meta-analysis that just come out. This is, again, very basic slide, but basically it's telling us there is a class 1 and class 2 elements, and in class 2 you see all the elements that I mentioned, but there are also protective factors, and sometimes we are surprised to notice some factors like anti-diabetes, we mentioned anti-BMI, related to class 2, for example, there are many irrelevant factors like coffee drinking, smoking, alcohol consumption, and so on. Because I like to mention that some of them has been reported in a no confirmatory way by some authors. On the right, you see another major conclusion, namely that the risk factors somehow are more responsible for the late expression of a disease, and early factors like genetic factors are obviously responsible in the more primitive part of a disease. But let's go back to metabolism. We know that ALS is characterized by decreased energy intake and increased energy expenditure. There is a lot of reason for that. We don't have to go in detail. Obviously, we have to consider dysphagia, but also altered microbiome. I'd like to mention the Aaron beautiful paper that has been released yesterday related to microbiome. And I don't go in details on that, but obviously this is very important. But on the other side, you see hypothalamic hypertrophy. And this seems to be very attractive and very promising. The next two slides are courtesy of Albert Ludov. He's a good friend in Ulm. Actually, there are two studies, epidemiological studies in Germany and Sweden, telling the same story, namely that between 20 and 62, the individual usually gain 12 kilograms. This is not the case for ALS patients, that apparently in the 10-50 years before disease onset don't gain weight. Albert was able to come out with the preliminary data related to hypothalamic hypertrophy. This seems to be some kind of selective, pre-symptomatic, and obviously hypothalamus is very important, and in terms of metabolism, is in between the sleep defect, weight loss with melanin concentrating hormone, or relaxin, and so on. So this is probably the beginning of a long story. And increasing the diet of a patient with 500 calories per day, actually the same group was able to demonstrate the increased survival of the patients. So the dietary income and the BMI and hypothalamic dysfunction are critical in this disease. Independent, indirect evidence related again to BMI are coming from the tofersen treatment. Tofersen is the first effort to treat ALS mutated for SOD1 patients with antisense, and actually the treated patients not only show clinical variability, clinical evidence of positive evidence, but also they gain body weight. So it's a direct, indirect evidence that somehow the body weight is very important. But I'd like to take another direction. In other words, we know for many years that the ALS is affecting, unfortunately, more male than female, but this ratio is changing somehow according to the last data of the epidemiology. But we know also that the clinical presentation of a disease is different from male to women. For example, the bulbar onset is much more frequent in female, and the bulbar localized expression is more frequent in female. So I was impressed about this review that just come out recently, looking at different risk factors, but specifically pointing to gender and sex. There are many evidence that actually we are probably taking the wrong direction in terms of genetic penetrance, for example, of C9 according to sex. The different clearance of TDP43 seems to be related to the different sex. We know the SOD1 patient have a slower disease progression when female, and the transgenic SOD1 male is more affected. We know that the remodeling of a neuromuscular junction in SOD1 to SOD1 toxicity is stronger in female, and we know that the skeletal mass has a different characteristic according to the different sexes. We don't know anything about the cerebral flow, cardiac abnormality, vascular abnormality in ALS according to the different sexes. Obviously, we have to admit that there are different immune responses, and the microbiome is different. And again, Aaron mentioned yesterday castration doesn't affect apparently what he was showing, and but we have to remember that TDP43 mice show early lethality in male due to gut obstruction, just to say how microbiome is important according to the sexes again. I don't go in details related to sex hormones, but related to biomarkers, I think we have to be sensitive enough to know the biomarkers can be different according to sexes. So we have to be more selective in collecting our biomarkers. So the review is somehow suggesting that we have to look at sex differences. They are totally not studied in ALS and probably can provide answers to some unknown question, unsolved question they still have. Final topic, treatment. Actually, probably you are aware that ALS SOD1 mutated patient can be treated successfully with tofersen. It's an antisense specific for SOD1, and SOD1 is decreasing the CSF, and the light chain of neurofilament are decreased in the plasma, in the serum, and the tofersen has been demonstrated to have a clinical efficacy on different parameters. Well, there is an ongoing trial, the ATLAS trial by Biogen in pre-symptomatic mutated for SOD1. The end of the trial will be in 2027. 150 patients need to be reached in order to close the trial. And this is obviously very promising because this is the first neurodegenerative disease that has a pre-symptomatic treatment ongoing. We are obviously very anxious to see what's going on. I need to mention C9orf. Actually, 3 trials have been finished and stopped. This is a failure story, and I'd like to focus your attention on BII-B078. This antisense has been designed to specifically to counteract the gain of toxicity. In other words, has been raised somehow to decrease the dipeptide and try to reduce the RNA foci. And actually, the preclinical evidence in the transgenics were quite impressive. But the trial was a complete failure, and actually the trial had been stopped. The data just appeared last month in Lancet Neurology. You see here no efficacy on the functional rating scale, no efficacy on the slow vital capacity, on the mega score. decrease in the dipeptide. So the treatment reached the primary goal to decrease the dipeptide somehow, but the neurofilament actually increased, and the plasma, both in the CSF and in the plasma. And this was one of the main reasons they stopped the trial. So the take-home message here is that we must be very careful how we design the antisense versus C9, because probably we have to take in full consideration the gain of toxicity, but also the loss of function. I'm mentioning C9 specifically because obviously this gene is affecting the largest population of LS, but not just LS, also frontotemporal is the most frequent gene. So this is a crucial point. And obviously we are collecting worldwide preclinical patients. We have almost a couple hundred preclinical patients in this country ready to be treated. Reaching the conclusion, I like to say that mild motor impairment is difficult to be defined on the clinical level in ALS, but at the same time, ALS is unique because prodromes may derive according to different axes. Obviously, we share the motor neuron axis, but we must be aware about the frontotemporal axis and extrapyramidal axis. There are more and more evidence that ALS patients has also extrapyramidal features. So somehow we can use MMI, MCI, MBI, and here I'm putting the minimal experiment extrapyramidal eye in order to define the prodromal clinical markers. Related to the study that I mentioned related to gene carriers, the SOD1 Benatar, obviously this is very good, but is this predictive for sporadic patients? Some people don't believe that it can be applicable, so we have to come out with a different story in sporadic patients, and we are looking to the Gold Coast criteria as a new avenue in order to try to define the prodromal situation. Obviously, the question is if hypothalamic abnormality are therapeutically important for prevention, and obviously we are very impressed about the possibility to define new biomarkers related to the hypothalamus. The brain-body mechanism contributing to sexual dimorphism in LS may be relevant to further identify risk factors. And I think that this is a very important new avenue that we must fully consider. Finally, obviously, we are very anxious to know what the Atlas trial is showing, and the C9, obviously, approach needs to be completely targeted. I'd like to thank my collaborators at the Department of Neurology at the University of Milan. Thank you very much.
Thank you, Vincenzo. I invite the speaker here at the desk, and we have 10, 15 minutes left. So for some question, over there, I see one ends up over there.
Hello, Chiara Liberati, Golgi Neurosciences. So, Vincenzo, this question is for you. Beautiful talk, thanks. And also thanks for mentioning C9orf, which is a beautiful target with a very intriguing pathological mechanism there, still unknown. And I'm very happy that you pointed that it's not just the gain of function, potentially the cause of the disease, but also the loss of function. And this could be one of the reasons why the BII antisense failed, but I would like you to comment another hypothesis that I have here, because this is an antisense which is by definition CNS, because they inject it to go to the spinal cord and then to the brain. So the periphery is completely neglected, but we know that C9orf could be also peripheral disease through immune cells. And for me, this is also one of the reasons why the trial failed, but please, I'm happy to hear your comment on this.
Yeah, I think you make the point, actually. it's quite. I asked the same question when everything has been presented at the Gordon conference in July. But that was the first presentation. Well, first of all, we don't have any information related to the length of amplification according to the different patients, and that can make a major difference. Secondly, and this is quite clear, something can be strictly related to the periphery. Somehow was more attractive to design something for the gain of function. But we definitely need to take full consideration of loss of function as well. I'm far away to understand how to deal with both with one single molecule. Maybe we need two molecules at the same site at the same time. I don't know.
So, hi, Carly Tanner, UCSF. Thank you to everybody for these really great and very hopeful conversations. I have a question for Dr. Postuma. As you know, in the Sidoroff paper that you cited, the seeding aggregation assay was identified to be abnormal in a few people who had hyposmia and nothing else. And theoretically, at least, that would mean the pathology was in olfactory bulb, maybe tract, whereas if you got RBD, you got brainstem pathology already. So at least theoretically, maybe aiming for hyposmics with the endpoint being not getting RBD might be another way to think about prevention. I just wonder what your thoughts are about that.
A couple of things to say about that. So first of all, I mean, the concordance between hyposmia and the synuclein assays in Parkinson's disease is almost 100%. And so it's been pointed out that a poor man's synuclein assay is a smell test. I don't think that works outside there. And the other thing is, how early do these things become abnormal? I think you're alluding to that. They might be abnormal already at the anterior olfactory nucleus stage, but there's also those other 3 to 5% of the people walking around. Some studies have found 8% who have abnormal synucleinopathy on these assays. And so what does that mean? Is it possible that this is very early prodromal? Is it possible that we're having assaults on our CNS by synuclein all the time, and we're successfully clearing them most of the time, and so every once in a while you catch it? I don't think we really know, but the long-term studies of these asymptomatic people with positive assays might help us a lot.
Would you like to ask you a question, you all? So, do you have any concern about potential ethical implication, since we are dealing with a preclinical phase, and in the future probably we have to consider DMT therapy, these modified therapies eventually, as adding obviously the prevention on risk factors.
I have concern about.
I mean, risk misdiagnosis, for example.
Yeah, I have concern about several things. So one of my concerns is that if we label everyone with amyloid, not ATN, but just amyloid, as having dementia, most of them will never develop dementia, and I don't see this as ethical. It isn't amyloid. Having amyloid is not the same as having Alzheimer's dementia, and I don't think we should erode the barrier between them and terrify people when the majority of them will never develop it. In terms of the things that we are thinking about that might help prevent dementia, so increased stimulation, cognitive, social, physical, looking after things that might be related also to cardiac risk factors, looking after your hearing and eyesight to help, which would also help. It seems to me that there is no ethical problem with this. It will give people almost immediately a better life, save money, and for some people, and we'll never know who, it will stop them developing dementia. And for other people, they will develop dementia, but it will be considerably later than they would have, and they'll have more years of good life. So I'm not worried about these interventions ethically, but I'm worried about labeling people as having a disease when we don't really know what it means.
Oh, I'm sorry. Were you going, Ron? Were you going to answer that one? I have another question for you. Okay. I have a question for you, and in a way also for Gil. We showed a while back that amyloid can clearly enhance synuclein aggregation and accumulation. And there are plenty of people who have both synuclein and amyloid pathology. And now we have a way of getting rid of some of the amyloid. What is your experience with treating people with overlap conditions with lecanemab or other emerging anti-amyloid antibodies? Would you be in favor of it? Are there particular risks related to ARIA, or what do you predict?
Personal experience is zero because we don't have it yet in Canada. But I will tell you something that we found. So it's always been a mystery to me why people who get Parkinson's first versus dementia with Lewy bodies first, and they are so indistinguishable in those early phases of disease. It's submitted now, but we did the plasma markers for amyloid, P181 and 42/40 ratio, and that appears to be the answer, in fact. So if you have a normal plasma amyloid ratio, you will not get dementia with Lewy bodies. You will get Parkinson's instead. The P181 actually worked quite well. We haven't done P217. So I think I think what's happening is RBD is a synucleinopathy, and it's spreading in its synucleinopathy way. And if you are one of those 45% or whatever of the population that has a tiny bit of amyloid in their brain, the two of them it just starts a fire and wipes your cortex out first before your brainstem. I used to think it was spread. I think it's now comorbid pathology that counts. And I would be very interested in doing a lecanemab or donanemab trial in a RBD patient with a positive amyloid. I think that might be quite interesting.
So how much confident are you with biomarkers that you mentioned?
So every biomarker is different, I guess, right? So, I mean, RBD is almost 100%. And so RBD plus anything is basically 100% is how it goes, I think. Olfactory loss, that doesn't have another explanation. The biomarkers are new. We don't really, we don't really fully understand what these normal ones, these normal people with positive biomarkers is just like the amyloid story a little bit. I don't think we quite understand what they mean. But the fact that 80% of our RBD patients are positive on a skin biopsy and 0% of our controls are, I think it really does change the way that we think about these.
Well, in my vision, we had to understand the biology of the biomarker. For example, what we know about neurofilament coming from axons, coming from what is the real metabolism of neurofilament. We need to go in details about the biology of a single neurofilament, the single element eventually to come out with something. I'm personally very positive about biomarker, but still we must understand exactly where are coming from, what is the metabolism, what is the turnover, and so on.
Yeah, I haven't, and I don't think anyone has, given the lecanemab. It's not approved in UK for the National Health Service, but it has been in trials. But in the trials, people who had a DLB or wouldn't have got it. And I think it would be very interesting to look at it in trials. And I think that what we're really learning about brains in general is that a lot of things are impure and people have more than one thing, and more than one thing tends to be synergistic, not just additive. And we saw with DLB that the cholinesterase inhibitors worked a bit better than in Alzheimer's disease. And maybe that's why, because they've got both, which nobody really thought about. So I think it would be very interesting. And in fact, I was thinking when Ron was talking that he should be setting up an adaptive trials program rather than just an ordinary randomized controlled trial? I know you're not supposed to ask other people about it, but what do you think about, on the panel, what do you think about that?
A platform trial makes a lot of sense. You can share the placebo group. We tried to get one together. Actually, we had two that we could have put in, but it just didn't work out. I think the difficulty is the front end loading of how you have to plan those things, and so you need sort of you need multiple companies or funding agencies all willing to go, and it can be done for sure. I will mention that there is a platform trial that will be started up by the Michael J. Fox Foundation, and it's targeting prodromal. They have not announced their partners yet, but it's exactly what you're talking about. So it should happen at some point. This is not just RBD, this is hyposmics and other prodromal Parkinson's patients.
Okay, I think it's over, and time is over, and I would like to say thank you to the speaker for interesting lecture, to the audience, and I would like, I think also on behalf of all participants at this symposium to say thank you, Professor Comi.
Of do intense days. We opened yesterday morning with the president of the European Academy of Neurology and with the WHO. I think it was really appropriate because we contextualize what we did in these 2 days here. So the brain health is really the referring point of even these 2 days, even if, of course, here we have focused on the neurodegeneration, which is, in any case, one of the more relevant epidemiological problems in terms of brain health. The intent of this roundtable is now to listen a bit at different bodies of the society in order to have a bit definition of what you consider a priority, what has to be done, at least as an intention. We must be ambitious, but I mean, I think it will be very interesting to listen to different views and different perspectives. Let's start introducing the participants. We have Professor Harris Ayre from the Rice University's Baker Institute for Public Policy from Houston. Frederic Destrebeck from the European Brain Council from Brussels. Catherine Sear from the WHO, please, thank you so much for being with us. Monique Breithler, who was already
presenting something the day one. Thank you so much. Is a representative, one of the most important network in terms of neurodegenerative disease from Germany. And then two very other important person, Pavel Swieboda from the Neuro Century, it's correct, no? Neuro Century, exactly, also from Brussels, Belgium, and at the other extreme, Professor Mario Battaglia, who is the president of the Italian Foundation for Multiple Sclerosis. So we have really the different bodies. I would like to start asking Professor Harris, if you want to start.
Thank you so much. Okay, my slides are there, and I guess we just advance them with one click. Is that how? Yeah. Okay, thank you very much, Professor Comi. Thank you for having me and my co-panelists. Thanks to everyone. So let me go big to get everyone re-energized. I'm going to paint for you a big canvas. I was thinking it's like the Da Vinci canvas. It's big, but we need to fill in the canvas. So this is an unfinished thing. What I'm going to talk about is this new global economic transformation that colleagues and I are working on. It's called the brain economy transformation. And it's very big. We think it's akin to the size and scale of the green transition. And I want to give some ideas for what Italy could do with Italy's national brain economy. And this is all about economic growth, which of course we know, as Mario Draghi has said, that economic competitiveness for Europe is a top priority. So that's the sort of gist. First of all, to say that this brain economy transformation is technically called the brain positive economic transformation. It is very transdisciplinary, and it's inspired significantly by the Medici effect. I was very touched by this book, and of course it's very Italian. So this has influenced me, and I share it back to you. This talk is like my ode to Italy, if you will. Anyway, transdisciplinary science is a wonderful thing that has very much inspired this work. So let's talk about the nature positive economic transformation. I want to frame this up for you. I am absolutely leveraging hard from the climate and environmental sector. There is this new economic asset called natural capital, which is a way of measuring biodiversity and species and environmental health. And so natural capital is being depleted right now. We all see it in the news. We have forest destruction, dirty oceans. We have species being lost. You can see in this diagram, this is a nature negative economy, right? And if you look down in the blue, what we could end up with by 2100 is a real big problem with complete degradation of our natural capital. So we need to go from a nature negative economy to a nature positive economy. That is undeniable. It's critical. So that's the nature positive economic transformation. This for you guys is like a 101 of economic transformations. So our brain capital is really problematic. This is data from Shayla's group, the IHME. We've talked about it all. The global cost of mental and neurological conditions is 2.2 trillion to the global economy. This data will be published later this year. This is a massive cost. It's burdening the economic security of nation states. And it's two-thirds of the cost, it's two-thirds of the gross national product of India. So this is big stuff. This is why a brain-based economic transformation is at the scale of the green transition. And our brain health is particularly poor right when we need our brains the most. The three most important brain skills are analytical thinking, creativity, and resilience, flexibility, and agility. And creativity is very important in the context of Italy because you guys are super creative as a country with your cultural creative industries. And of course, the Prada Foundation is a great example of a very creative organization. So this is particularly poignant for Italy. How do we give more horsepower to Italian creativity through the brain economy is an interesting question. So our brains are in the red, they're in debt, right when we need them in the black to be profitable. This is a very simple phrase that was given to me by a business person in the US. So brain capital is what I talked about. It's brain health and brain skills. It's two things. If you remember nothing else from me, that's what brain capital is. And that's what we need to really build in our countries. So brain capital is not just a cost. We need to get away from just talking about costs. The politicians are no longer receptive to talking about cost. They want some hope and optimism. So we need to talk about assets. So brain capital is an asset for a nation. Brain capital should be as important as GDP and road and bridge infrastructure. And we need scientists like you guys all to help us figure out how we're going to measure this, because what gets measured really matters, and what gets measured gets invested in. And the UK, of course, is doing great work in this regard, so we can certainly leverage from them. And I think they were at the G7 last week with Fred in Ancona talking about brain health. The Tony Blair Institute was, at least. So this is the brain positive economic transformation, or the brain economy. It's to stop and reverse the global loss of brain capital. And in the Italian context, how do we do this at the national level? So we do it through some things I'll get through really quickly: valuing, financing, innovating, and strengthening public sector capacity. So I'm originally an MD PhD, but now all I do is think about economic policy and economic strategy. So this is like a playbook that I'm showing you. We have a global brain capital dashboard, which looks at the determinants of brain capital: social, digital, environmental determinants. It looks at brain health and brain skills. This is a global dashboard, country by country. We can easily repurpose the methodology in the Italian context. Brain bonds, Pawal Swoboda will talk about, so I don't want to steal his thunder. It's really, he's leading the project. This is a really big deal. Green bonds were pioneered in Europe and is now a $2.2 trillion financial sector to power clean energy investing. So what could be a brain bond? And I'll let that linger, and Pawal can talk more about it. Brain economy missions. Interestingly, from Marianne Mazzucato, who's a British-Italian economist, she has really pioneered mission economies. So how do you get government that many people have lost trust in, how do you get it going again? Well, you give government a very specific target, like go to the moon was a target that the US government had that they nailed, and it was transformative for the country. So what could be a brain economy mission? Could we reduce dementia by 10% in 10 years? And imagine how that, if the Italian government just had one focus or a couple of foci, it could really rally the bureaucrats, rally the private sector, the philanthropy, the NGO sector behind a very precise target. So interesting question for all of you is, what could be some brain missions that we pioneer akin to getting to the moon? What gets going in your mind when you get asked that question? We need to think about a brain economy lab. This is about sector by sector activity. You don't just say motherhood statements. You need to build brain capital in Italy. You need to think, what are you doing in the digital sector around social media? What are you doing in the environment sector, in the aging sector? On and on and on. We need to think forensically sector by sector. And the Bezos Earth Fund from Jeff Bezos, the Amazon founder, is a philanthropy that's specifically focused on it. It has a systems change lab for the environment. So we need to learn from that, again, cross-pollinating with the Medici effect into the brain. It's all laid out there for us. We just need to really spend the time to go through this sector by sector. As I've noted here, down the bottom is the foundations of a house, is the brain transition. And at the top is these skill-based transitions, digital transition, green transition, bioeconomy, etc. These are skill-based transitions, and they won't happen unless we have a brain transition. If kids in school can't learn, if adults in the workplace can't upskill and reskill, we won't have these skill-based transitions that Europe loves so much, that are high priorities. And interestingly, Mario Draghi is a famous Italian. He just released he was the president of the European Central Bank. He just released a report for the European Commission about European competitiveness. And interestingly, the brain and mental health was not mentioned anywhere. So my little thinking was, we need to brainwash Mario Draghi. That's a bad dad joke. But that's what we need to do. So that's what we're thinking about. How do we make ourselves relevant, all of you in the audience, to the European Commission's economic competitiveness plan? How do we put you at the heart of the engine of the economy, because our brains are the engine of the economy? This work is all gaining traction globally. It's wonderful to see in the global south and the global north, Sweden, Finland, et cetera, et cetera. So there's lots of countries that are coming together because this is a big pan-human issue. It's global, and all these people want to work together. So they are there. And the question is, should we do an executive education course on this kind of work? I recognize this is complicated, so question for all of you guys is, should we develop like a 4-day course Rice University in Houston is interested to do that at their Global Paris Center. So I'd love for you guys to tell me, do you think that makes sense? And what would you want to see out of such a course? So in conclusion, we need to do this transformation. We're at the foothills of this transformation. You guys are all now at the foothill with us, ascending the mountain. This is a very large mountain, as you can imagine. We need to develop a shared vision and mission. We need to identify champions. You're all champions. We need to align initiatives. We need to pilot things and then monitor and evaluate. And finally, I think we need to think about Viviana's Milan Longevity Summit as a next place to convene to talk about an Italian brain economy. So Viviana, maybe you can tell us about that at the cocktail party. And we need to, I think, drive this towards the G7 in Canada next year. Fred will talk about the success of the G7 in Italy with getting brain health on the map. Next year, it's in Canada. So let's look at that as another way of getting sort of global political attention. So thank you very much. And I look forward to interactions and questions. Cheers.
Thank you. Thank you, Harris. I will start where you have started. Brain is a capital. All our brains are a capital. Each of us can do something for that. Because if we give always to the others the responsibility to do, that doesn't work. And here, as we have heard in these 2 days, by the way, the problem of a neurodegenerative disease is an individual problem. Touch each of us. So, I mean, it is then very important that we think about what can be done, and no doubt education. That's really a fundamental point. Starting from the elementary school, starting from the first step, because it's something new that we have to be able to, let's say, activate in the brain, and understanding that each of us has a very big responsibility. I would like to ask now Frederick Desterbeck, who is the Executive Director of the European Brain Council, is a bit at the crossway of many different activities. So, I mean, we are really very interested to listen which is your view and where you push.
Thank you, Professor Comi. And actually, I don't see my slides on the platform. No, that's Pavel's. Oh yeah, not these ones. Otherwise.
This is Godard. Because there's a different email, so I can. Comandare le slide. Le slide. Okay, so in the meantime that we try to find the slide, I don't know where they are, but I would like to ask to have an intervene. Catherine's here, if you want to just to cover this time. So Catherine, no, I mean.
I'll try my best. It's live. Okay, good.
Thank you so much.
I want to follow on from what you just said, Professor Comi, and that is we all have a role to play. It's important to understand and know what each other's roles are, and kind of then stick to our roles and do what we're all placed best to do. And what I mean by that is that Harris and his advocacy for brain capital is really kind of like opened doors and speaking or involving different sectors. Coming from WHO, we have a slightly different perspective in that we're going in parallel. And what I mean by that is if we're just looking at the sheer number of people who currently live with nervous system health loss, and you heard Amy on the first day, we're talking about over 3 billion people worldwide. We know that these people need access to services somehow, and that we're currently in no position to provide these services in any way that would be appropriate and kind of be responsive to their needs. So we're really committed at WHO to our member states. They've adopted the Intersectoral Global Action Plan. We have 2 other relevant action plans, one on dementia, one on meningitis. And we really want to, over the next 10 years, try our best to help member states increasing the service, improving and strengthening the services so that they can deliver the care that's needed. And in parallel, I totally agree, we do need to work and invest on creating for future generations, but also for us at the moment, the most resilient brains possible so that we can take on the challenges that are before us.
Well, in the last session, the problem of biomarkers have been really introduced. Of course, we expect a lot from biomarkers. We expect to be able to act also in poor countries and to bring there something easy to use, something that may be not too much expensive, something that may really be in a condition to allow us to have also for poor countries the adequate quality of assistance, at least for some certain level. Which is the strategy that the WHO think about to use in order to arrive in some condition where there is very little until now, and we need so much to do to align with the more evolved countries.
At WHO, we have a long history or long standing with doing biomarkers, doing or coming up with vaccines, with new treatments for infectious diseases and rolling them out. Now, more recently, that also shifted or attention was brought to dementia specifically, and that's something new for WHO. So dementia is the first one that has a blueprint and the first one that we're now releasing preferred product characteristics for biomarkers. Now, what these do is they're sending a strong signal to industry. We do need industry to develop these tests, but in doing so, they need to keep in mind from the outset what are the needs of the most vulnerable and the most disadvantaged communities, and what do these tests need to have in terms of properties so that we can use them equitable and reduce the gap that we currently see rather than broaden it.
Well, thank you. Thank you very much. Dr. Desselbeck, your slide there now. Perfect. Please.
Thank you, Professor Comi, and apologies for that hiatus, and thank you for the supporting team in helping me out. It's just like I don't know where to find it. Okay, fantastic, great. Okay, yeah, somehow it's not appearing on my screen. Okay. So I'm speaking on behalf of the European Brain Council, as Professor Comi introduced. So we are an advocacy organization established in Brussels, and we basically run our advocacy towards EU institutions, primarily the European Commission, the European Parliament, but then as a consequence at country level, as well as at the global level also. We do represent patient organizations, clinical and scientific societies, as well as the private sector, to promote brain research primarily, but with time, our agenda developed to really embrace the whole of brain health, of brain innovation, but also entering into what Harris presented on the brain capital and brain economy transition. So our latest strategy was revolving around the fostering of innovation in the brain space, but also how to improve patient outcomes for those living with a brain condition. And obviously, the environment in which we operate is a very complex one. So this is a summary of what it looks like. It's our beautiful European democracy. I won't go into the details of that, but it very much looks like a gas engine. What I wanted to draw your attention into is like the current developments of the life and the cycle of this institutional landscape. So we all were invited to express our opinion and express our vote back in June. That means we have a new parliament that is now up on its feet and ready to work. But we also have now, and this is, let's say, the natural next step in that life course, the nomination of a new commission. And it was mentioned earlier on that the Draghi report had really set a landmark into the field of competition, competitiveness, and innovation as being the new priorities for the EU. And this is now translated in the allocation of the portfolios, but also the structure of the commission that President von der Leyen has designated. There is still a bit of process to go on there because all these candidates will be heard by the European Parliament. They will have to present their priorities and demonstrate that they are fit for the job. But what is very interesting is to have this understanding that there is a great drive towards restoring Europe on the global stage in terms of its capacity to really compete with China and the US in particular. But so we have a momentum also to defend what is making Europe relatively unique. I also would like to report on a certain number of achievements and in terms of context. So there is a very interesting and important work that is relevant to our discussions today, which is the emergence of a brain health partnership. So in the context of Horizon Europe, there is a lot of discussions between the national funding agencies to see how they can prioritize and strategize together brain research and then share the load of how to fund a program and different calls in that area. EBC is actively engaged into this. We had the seminal idea of creating such a platform, and we were running the predecessor of the Brain Health Partnership, which was called EBRA, the European Brain Research Area, and was really important in terms of setting a common agenda. Another piece of action that is current and ongoing is the Non-Communicable Disease Initiative launched by the Commission a certain number of years ago, but it's only now that they are going to target mental health and neurological disorders, and that's the next area on their list of action, and we are actually looking forward to them being ambitious in what they are going to propose and using all means to influence them in that capacity. All means that in the context of the election, EBC published its manifesto under the label No Health Without Brain Health, and very concretely, it contains very concrete recommendations, and I'll summarize them as under these two. The first one is to create in the European Parliament what we call an intergroup. That means a non-political platform to address topics that are then decided as being key priorities, and we hope to see brain health, brain research, and brain capital recognized as such. And we are in the very last days of being able to get that proposal on the desk of the Secretariat. And I've been texting with the office earlier today, and we are getting to very close to the threshold of support. So the level of excitement on our side is quite big in that respect. The reason why we need that European Parliament intergroup is that we would like to have a longer term agenda, which is the development of an EU coordination plan. So it's a way by which we would develop a strategy at the European level that connects all the dots that I mentioned. I mentioned the partnership, I mentioned the NCD initiative, but there is way more to that if we want to see things moving. So of course, we would like to see a surge in the investment for research, but we also would like to see a proper thinking along the lines of what Harris was mentioning a minute ago. So we have obviously a huge timeline ongoing. We are extremely busy, as you can say, but I'd like to flag the day of today within this milestone, not only because we have this beautiful event in Milano, and congratulations to Professor Comi for gathering it, But I need to convey an apology officially on behalf of our Vice President, Professor Claudio Bassetti, who couldn't be there because he was speaking in the Health Committee of the European Parliament this morning and presenting all of these priorities. And we are now hopeful that it is now going down the policy trail and getting to the highest level. I won't repeat what Catherine was saying because internationally we've got a momentum. Would like to really see the European Union fitting into this and playing its role of coordinator and helping out member states in developing their national strategies. I think it's really important that we seize the momentum in the current EU mandate that we are now having, because loads of these WHO action plans have as a target 2030. So if you make the maths, that's exactly the duration of the EU mandate that is opening now. I'll just finish off on that global outreach, because EBC is present now every year in the context of the General Assembly of the UN. We had a beautiful event where we launched the Global Brain Coalition, and I invite you to look at the recording of that event. But as Harris was saying, there are no further opportunities that we'd like to identify with the Brain Awareness Week, the United Nations, but also the G7 and possibly the G20. So all of that is in the making, and any insight or any contribution or comments would be more than welcome. And our next step and our next milestone are the Brain Innovation Days in Brussels. You are all warmly invited. I invite you to consult the braininnovationdays.eu website for all information and registration. And obviously, we have a fantastic program awaiting our audience, and we look forward to meeting you there. Thank you.
Thank you very much. Together is much better. I would like to ask Professor Brettler, well, in many countries, in Germany, you have this very fantastic network of neurodegenerative diseases. In Italy, we started with the network of neuroscience. In France, they have a network of neuro. So that's because we need to move to a different dimension. When you have to face big problems, you have to have appropriate level of organization, economically, but also scientifically, so relevant. How critical is this? How relevant is that we work more together?
Yeah, well, absolutely. I could not agree more. And you mentioned the example of Germany. I think there it was recognized in the beginning of this century that, for example, neurodegenerative diseases were really one of the upcoming grand health challenges for society, right? So this idea that let's invest in that, let's bring our or join forces and build a translational research institute, really translational from the beginning. The idea that we need fundamental research, that we need clinical research, that we need population research, that we need healthcare research to cover this entire spectrum from healthy people at young age, throughout their age, toward disease status, towards, well, basically, how do we take care of them until that, and that supported by research at all those levels. I think that is critical to have an impact. I think we get better research. I think it's also the way to speed up research, because if I think how I started last century in 1990, I mean, in the 1990s, a lot of the observations in population research that we made at that time are sort of have been rediscovered this century, right? And I think part of that is because basically we had these silos of population research, fundamental research were in their silo, and I think what we now see is that there's much more converging together. It's still a challenge, I would say, because we have to learn each other's language, be open to different approaches, but it's also extremely rewarding and it's more impactful. I mean, in that sense, I totally agree with the first speaker that if we can leverage more brain power, I mean, that is capital to reach solutions.
Thank you, Monique. Professor Battaglia, the person with the disease is taking more and more a central role. We have left a bit as a patients the passive role to be an object of interest, and we are becoming protagonists in some way of our health. This is particularly true for our brain because of the complexity. So what is your view about the possibility to try to promote as much as possible this campaign of brain health and particularly of neurodegenerative diseases?
I had some thoughts to share during those 2 days that came up to my mind. First of all, I want to say that we have heard for all the conditions the sentence saying prodromal illness is the illness, as well as we have heard the wording secondary prevention and primary prevention, as well as risk factor and causes. So this has a lot of implications. Implications for the rights of the people, not only at European level, of course, but at world level. So this means that every person has the right to a good health, to concrete answers to the situation in which they live. Of course, this means nowadays that we need to have a network, a team. And in this team, there are, of course, the professional societies, the researchers, of course, but also the patients' organizations with a specific role. That is not to be passive, of course, is to sit at the table, because the experiential knowledge, of course, but also the vision of patients, that means not only individuals, but patients' organizations, so a coalition for each illness that has the power and the knowledge to present all the diversity in each illness. And there is an issue that is fundamental, that is that all those organizations have to work together. Have to work together because we have so many commonalities in neurological disorders and especially in neurodegenerative diseases that we need to work together on all the issues. That is not, of course, only advocacy, it's also funding or promoting research. So I think this is a fundamental, and we need to work at world level, European, of course, continental level, and the national level. For this reason, we, and I'm not speaking about MS, I'm speaking about many disorders. We are working together with the Neurological Society for a plan and an activity that should be taken and discussed, of course, with the Ministry of Health at the national level. So, of course, now we have only 3 patient society in the start, that is ourselves, Alzheimer, and rare disorders. But we are involving other societies. There will be a meeting at the Neurological Society annual convention, Congress, with all the 36 organizations of patients for working together for these results. So I think this is important. Of course, some of us has also the power, and I mean the power in inverted commas, of promoting and supporting research. That is quite important because in the neurological field, not many organizations like us has this possibility, but this is fundamental for pushing research and addressing the needs of the persons and not just general research, because the research needs to have concrete results for the people.
I take the opportunity just to inform all of you that in this 4 months when the exhibition, scientific exhibition will be open, we will organize 8 days with 8 different type of patients organization, because we believe that it is really fundamental to transfer to the person, to common person, the problem that we have to face. It's not just our problem, it's the problem of everybody. And I think that and this was the reason why we organized such an exhibition, because with the language that is understandable, everybody has to feel to be on board. Now, excuse me if I go back to the economy, but you know, economy is really fundamental. If we are ambitious, then we have really to take into consideration even where to find the resources for the campaign that we have to promote, for the education that we have to try to bring everywhere. I would like to ask you now, that is your role, then be prepared, because then I will also come back to you. Just to tell us a bit, you have, again, also you have been at the center, at the crossway of many different initiatives. Give your view.
Thank you so much, Giancarlo. And if I could maybe just start by alluding to the last segment of the discussion, because the Italian scientific societies, yourself, Giancarlo, and the MS Society have been really pioneers in Europe of driving this science of patient research and patient inclusion. And it now needs to be embedded also in the research agenda. So what I would like to do, if I could have my few slides, don't worry, it's only a handful, and I prepared them before the session. I prepared them before the session to basically draw conclusions from, or try to draw conclusions, because I wouldn't dare to do a fully fledged comprehensive job on the subject, from the discussions yesterday and today, which have been absolutely impressive. So our focus in this session is on the priorities and future actions in prevention of neurodegenerative disorders. So I will have 8 points to make very briefly, but I would like to sort of entitle them the brain economy blueprint to prevention, using the term that Harris already described, because a lot of the interventions which are needed on the basis of this completely impressive science that was presented over the two days, this is really anchored in something broader than brain research, right? In order to make this now actionable, we need a broader approach that is embraced at the societal level. And for that, the brain economy concept is very conducive. So my first point is that clearly we see a lot of areas where further research is needed, right? You will know this better than anybody else. The good news is, as Fred already said, that the Brain Health Partnership is now emerging. The Brain Health Partnership is significant because it will be the most impactful single funding program of the European Union in the years to come, at least. I was myself Director General of the Human Brain Project in the final phase. This was when the EU invested quite a lot in brain research, but it ended after 10 years. All good things come to an end, and that project also ended last September. And currently there isn't really a single sizable ambitious project in the field of research. The European Brain Health Partnership will fill that gap once it comes online in 2025. And the good news for prevention, which has been our subject today, is that there is in the Strategic Research and Innovation Agenda that was just presented after consultations this summer, there is Pillar 1 on actually preventing brain disorders. So that's great news. But when you look at national research programs, and we have to remember that national research is the bulk of research being done in Europe, 80 to 90%. European is only part of it. It's important because it does leverage a lot of important developments, but it's a fraction. So when we look at the national research funding, it often follows a generic approach, right? There's less and less focus on specific disorders, be it cardiovascular, being brain disorders. And the attempt is to be as generic as you can. And of course, that's fine, right? For prevention purposes, a lot of what is good for the heart is good for the brain. But there are some brain-specific aspects which also require further research. So what I would plea for is that we need to conceptualize this add-on which concerns the brain, namely what needs to be researched specifically on top of the more generic research undertakings with regards to the brain. My second point is that we need a reservoir of knowledge on prevention that policymakers also can use. And I say that because I looked at EU for Health, so Europe's main program for action in the area of health. And last year, there was a call for proposals in the area of prevention of neurological disorders, so all good. Very, very, very well. But the starting point is to map. They said, this is a quote, so needs assessment and subsequent gap analysis. So in a way, it felt to me like starting from scratch, right? Rather than going to a certain reference place where you would find all this wisdom, the call was asking people to start from the beginning. So can we actually have such a reference base of information for policymakers on the modifiable risks? We have heard during those 2 days concerns about the scientific validation of a number of risk factors. So perhaps there could be a Prada initiative or a Milan initiative to work on the methodology with regards to modifiable risk factors. And this initiative would give an answer to the question, how much we can draw on observational data where trials are desperately needed. My third point refers to the question of systemic investing, and this has been raised already, and you just alluded to this, Giancarlo, because we know that the needs are enormous, and the $2.1 trillion US figure that IHME has or is coming up, because I think this is still to be published before the end of the year, for the costs of brain disorders globally is, of course, absolutely overwhelming. So huge investments are needed, and therefore a group of us, including Harris and Fred and others, have conceptualized over the past few months how a systemic approach to brain economy could be worked out and what specific financial instruments are needed. It's systemic because it needs to draw on engagement in different fields that have to be mutually reinforcing. That's the nature of systemic investing. And we have a discussion paper on this subject if you're interested. There's a QR code up there. And indeed, one of the financial instruments could be a brain bond. This concept was, in fact, floated by a high representative of the European Investment Bank in one of our discussions, because the bank is Europe's climate bank. It has a lot of experience with green bonds. Sorry, I already mixed the two. And therefore, we can draw inspiration from how the green bonds were developed in order to have impactful interventions in the area of health and in the area of skills as well. My fourth point is that we need a place-based prevention, because what I also heard and what we heard over these 2 days is that the risk factors have a high degree of variability, and it is showing in different data that we need to have consideration for the local characteristics, and we need to have tailored interventions. So it's very good news that also a range of national brain health plans is now being developed, or brain economy plans in some cases, which are, again, broader, more encompassing. And there are also good practices in a number of countries on co-creation with the local stakeholders. So the Finnish brain health plan is a good example of that. It was really worked out in conjunction with a lot of actors locally and regionally. So you could say that this is place-based prevention that really responds to the requirements of a given area, given territory. My sixth point, I think, I stopped, I'm not sure if I'm counting correctly, but anyway, coming too close to the end, is scaling up personalized prevention. This was very powerfully explained also yesterday with regard to the AI, because we are at last being able to offer personalized interventions, including personalized prevention. So clearly risk stratification would look differently in those circumstances. And we could also dream about having a brain health tracking system that would cover the duration of our life. This would help because quantification of brain health is more efficient than looking at individual markers. And we could also improve anticipation through these predictive models, which were presented yesterday. And since, as I said, I've past experience of working in the Human Brain Project, a lot of the attention of that scientific endeavor was focused on coming up with predictive brain models. Prevention everywhere is also what I think comes from our discussions. I remember Dr. Elena Sant telling us about the wellness center which she set up in her clinic. This is really about having an overview of best practices in the field of prevention, being able to draw on them, share them, and also pilot the more innovative approaches when needed. And then we come to the business models, which are quite essential because we should not rely in prevention only on public sector intervention, right? We need to be able to build a market in this field wherever possible. And this is happening. There are insurance models which are incentivizing engaging in prevention. There are also a lot of initiatives, and the Baycrest Center in Canada is, I think, a flagship example of that. So there are initiatives with regard to prevention-friendly workplaces, so incentivizing employers to actually think hard about how to support the cognitive health of their employees. And my last point will be about supporting good brain health, because we have largely discussed brain disorders, and we had even a plea to conceptualize a new category of disorders, which, I mean, I completely understand the logic of that proposition. But as we do that, I think it's really important to also keep in mind that people are more and more interested in good brain health. And I'll give you that finding from a study 2 years ago, where over 91% of respondents said that they were ready to take a brain health test. And 98% declared a change of lifestyle if required. So if I were to have an optimistic message at the end, I think a lot of people are ready to embrace the messaging coming from this amazing conference. I think a lot of people are ready to take brain health into their own hands. Thanks so much.
Thank you. Thank you very much. Harris, I would like to ask you if you can make some final comments from the budget point of view, particularly.
Okay, this is on. So how do we get more money, more finance, more resources to this field that we all care so much about? I have some thoughts. I think we need, when we're engaging the finance community, we need new narratives. We need to do something that speaks to their emotions and then also their analytics. So things like brain health are very nice. These are new things, brain capital, brain economy. So that's the first part. And then making friends with finance people is important because there's no easy answer here. A lot of this is dialogue backwards and forwards. And so a lot of my time I spend learning very humbly from finance people. We need to recognize that there is, tens of trillions of dollars of private capital in the world, pension funds, sovereign wealth funds, that is sitting on the sidelines, not engaging with us. But so we need to look at things like the climate sector and the green sector. They've managed to engage trillions of dollars. So what can we learn from them? You guys probably see, like, there's a loss and damage fund for low middle income countries that have had climate disasters. You know, so high income countries are paying into loss and damage funds to pay low middle income countries to reconstruct with climate damage. So is there an equivalent with, you know, climate in the brain that, you know, is there a fund that high income countries could fund to low middle income countries around climate related brain damage, sort of subtly, that's just an idea. There are ESG and sustainable development goals. These are ways of thinking, trying to make money more impactful for the good, make money that doesn't just, money doesn't want to make more money. Money wants to make a little bit more money, but also to do good social impact. That means environmental, social governance, ESG, and it means the sustainable development goals. So we need to, as a brain community, we need to engage with these impact investment frameworks. This is technical stuff, but we're happy to explain it more. That's what we're doing with the European Investment Bank, is we're thinking through these different ways that we could engage in the financial sector. And then let me make the last comment, and then I'll stop. Private money, private bankers and finance people, we also need to engage them with other big corporate interests at the table. And I was having a great dinner last night with two architects. You invited architects, which is wonderful. So what about if we thought through redesigning the architecture and design of big swaths of Europe, to go from architecture and design that is not interested in thinking about neuroscience to turn the new page? Let's design all new buildings for, you know, low middle income people, high income, commercial, residential, with neuroscience inspired architecture. That is where you can deploy tens or hundreds of billions of dollars. And that's where the bankers will come alongside the big developers. So these are some new ideas that significantly percolating from dinner last night.
Thank you so much, particularly for this last comment, because it is clear that we have to change everyone in our own type of activity, not just one. So that's really fundamental. Okay, we are at the end of two, at least for me, very interesting days. I want to thanks all of you for the participation at this roundtable, and let me extend also my gratitude to all the speakers. All the talks have been really extremely excellent. Many thanks also to all those who have worked behind the scene. We had more than 1.5 years of activities in order to be able to build such a day, such a couple of days. Let me express my gratitude particularly to Cornelia, to Andrea, to Fabiana, and all the others that have really worked deeply. And a very, very special thank to Miuccia Prada, because everything is because he is really in love with the neuroscience and the world. Thank you so much. Thank you.